di ekosistem pelagis Kepulauan Sangieta Laut. Sulawesi Utara Indonesia. Indonesia itu adalah pusat dari keanekaragaman hayati laut di dunia. Kalau untuk di darat ada di Brasil, tapi untuk di laut itu ada di Indonesia. Namun masih banyak perairan Indonesia yang belum kita kaji, terutama dari segi keanekaragaman hayatinya maupun potensi sumber daya hayatinya. Dengan tujuan ada 3. 1, mengungkap keanekaragaman hayati laut di perairan Sangieta Laut. Yang kedua, mengungkap potensi sumber daya hayati laut di perairan Sangieta Laut. Tapi yang membedakan kedua ini, untuk biodiversitas itu lebih ke eksplorasi, mencoba mengungkap apa-apa biota yang belum kita tahu, yang bisa memperkaya keanekaragaman hayati perairan Indonesia. Dan untuk bioresource-nya lebih ditekankan pada biota-biota yang bernilai ekonomis penting. Kita berharap nanti kita akan targetkan minimal ada 2, ikan sidat dan ikan tuna yang memang benar-benar memiliki nilai komersial di dunia. Dan yang ketiga, tujuannya adalah melihat keterkaitan antara biodiversitas dan bioresource ini dengan kondisi fisika, kimia, geologi, dan perairan di sekitar Sangieta Laut, mengingat perairan ini banyak dikelilingi oleh gunung laut, dan kondisi fisika kimia oceanografi di perairan yang memiliki gunung laut itu sangat spesifik. Secara fisik, kimia maupun geologi itu sangat menarik untuk dikaji. Banyak sekali tantangan di awal kita mau memulai kegiatan ini karena memang lokasinya itu rentan. Tapi buat kami untuk bisa mengkaji potensi keanekaragaman hayati laut Indonesia, apalagi di wilayah-wilayah perbatasan, kita memang harus punya keberanian ya, harus punya tekad yang lebih kuat karena ya itu tadi begitu besar tantangannya. Nah, terutama tahun ini ya saya kira ada banyak kejadian gempa bumi dan gunung meletus, dan barusan adalah kejadian gempa bumi di Palu Koro. Nah, patahan atau sambungan dari sesar Palu Koro ini sebenarnya mengarah ke Laut Sulawesi, dan kita sempat mengkhawatirkan itu akan sampai ke perairan Sangieta Laut. Sebenarnya dari perairan selatan Sangieta Laut sampai utaranya itu di Miangas ya, itu adalah gugusan gunung laut dan merupakan bagian dari Ring of Fire dunia ya. Jadi cincin api bawah laut dunia yang melingkar dari Samudra Hindia hingga ke utara Pasifik. Jadi melewati Sumatera, Jawa, Nusa Tenggara, terus ke utara hingga ke Sulawesi, dan dia tembus sampai Sangieta Laut tentu saja, dan ke Filipina hingga ke Jepang. Makanya daerah-daerah ini selalu sangat rentan, dan salah satu yang menjadi target kajian kita adalah Papatua Seamount ya, atau gunung api Papatua yang ada di depan kita sekarang. Sebenarnya kita ada 5 jam di depan gunung api tersebut, jadi kita akan mengarah ke situ malam ini untuk melakukan kajian.
Untuk pelayaran ekspedisi Widya Nusantara 2018, KR Baruna bertolak dari Pelabuhan Bitung menuju Tahuna. Untuk stasiun jumlah 35 stasiun seluruhnya. Setelah stasiun 17, KR Baruna Jaya 8 akan masuk di Tahuna. Untuk acara open ship, kita membuka untuk semua masyarakat Tahuna. Dari masyarakat umum, pihak sekolah, akademisi, silakan mengunjungi KR Baruna Jaya 8.
sains itu ke bidang pengembangan genetika, ya, kelautan di Indonesia. Jadi salah satu cara adalah dengan melakukan multi takson sekaligus, dan itu disebut dengan metabarcoding. Itu sekarang sangat atau ilmu yang terbaru di bidang genetika molekuler, dan ada salah satu anggota laboratorium saya, Bapak Oni Marwayana, yang memang fokus ke situ. Saya minta untuk mengkaji environmental DNA atau DNA lingkungan dari ikan yang ada di perairan sangita laut. Nah, terkait environmental DNA untuk mikroalga yang dihandle oleh Pak Tegar ini, sebenarnya ini tidak 100% untuk biodiversitas, tapi juga untuk bioresource tadi, ya. Karena mikroalga sekarang punya banyak fungsi, tidak hanya untuk ke arah budidaya sebagai pakan, ya, tapi juga berpotensi sebagai bioenergi. Satu lagi kita punya saudara Charlie Esther de Freitas. Beliau fokus ke kajian mikrobiologi laut yang tidak hanya mengarah ke pengungkapan keragaman bakteri yang ada di perairan sangita laut. Jadi dan banyak kejadian tumpahan minyak di perairan Indonesia lewat teknik yang akan dikembangkan atau riset yang akan dikembangkan oleh grup peneliti mikrobiologi di Pusat Oceanografi LIPI. Saya kira ini bisa membantu untuk memecahkan masalah tumpahan minyak tersebut. Program ini adalah merupakan program unggulan nasional dengan adanya dana dari pemerintah, dan untuk bisa menjadi program nasional, unggulan nasional, salah satunya kita harus bisa melibatkan stakeholder ataupun lembaga-lembaga riset yang ada di Indonesia.
Di atas kapal ini saya diberi kesempatan untuk mengkoordinir observasi larva invertebrate, di mana kita ada 3 researcher, Ibu Erna untuk krustase, Ibu Ana dengan moluska akan bertanggung jawab dalam analisa molekuler genetika bersama-sama dengan Ibu Diah. Bagiannya cephalopod juga akan observasi lebih lanjut untuk molekuler genetika.
Saya berpartisipasi dalam penelitian larva ikan pelagis. Dan saya pikir ini adalah kesempatan yang luar biasa bagi saya.
Pada kesempatan ini saya bergabung di bidang planktonologi, khususnya fitoplankton. Jadi kita bisa menjadikan informasi ini sebagai referensi di bidang ilmu pengetahuan, khususnya biologi laut, bagaimana perbedaan antara plankton, sebaran plankton yang ada di laut dalam dan daerah kostal.
Saya terlibat di dalam tim plankton, terkonsentrasi pada alat CTD untuk mengambil planktonnya di kedalaman 600 meter. Data yang akan didapat akan saya gunakan untuk tugas akhir saya, yaitu tesis. Saya sangat berterima kasih sudah dilibatkan ke dalam salah satu ekspedisi yang sangat, menurut saya adalah sangat luar biasa, yang sudah diselenggarakan oleh LIPI dan sudah melibatkan mahasiswa seperti saya yang sangat membutuhkan keringanan biaya dalam melakukan penelitian seperti itu.
Di tim ini saya diberikan tugas secara khusus untuk meneliti tentang ikan laut dalam, baik stadia dewasa dan juga stadia pada awal hidup ikan atau larva ikan laut dalam. Dan harapan saya adalah melibatkan lebih banyak lagi institusi perguruan tinggi maupun lembaga-lembaga riset, bukan hanya yang ada di Jawa, tetapi berkolaborasi dengan universitas yang di daerah, sehingga ada nanti transfer knowledge, transfer skills, sehingga kita boleh bersama-sama mengeksplorasi dan juga bisa mengembangkan ilmu pengetahuan terkait dengan kelautan, marine, untuk ikan laut dalam yang ada di perairan kita di Indonesia.
Setelah mendapatkan ikan pelagis, saya bersama Bapak Elvis Bataragoa akan mengidentifikasi sampai ke famili untuk larva ikan pelagis dan ikan kara. Mudah-mudahan bisa mendapatkan ikan komersial yang bisa menambah perekonomian dari masyarakat Kabupaten Kepulauan Sangihe, karena sebagian besar adalah nelayan.
Saya terlibat dalam penelitian tentang ikan sidat. Dengan terlibat Ewin ini saya bisa menambah pengetahuan tentang bagaimana mengadakan atau melakukan riset di atas kapal penelitian yang dimiliki oleh LIPI dan bekerja secara tim dengan peneliti-peneliti dari LIPI maupun di luar LIPI.
Ini adalah kesempatan saya yang terbaik di ilmu pengetahuan perikanan. Apa sebab? Karena ekspedisi ini bisa membangun bank data untuk sumber daya dan potensi hayati di perairan pelagis dan laut dalam di sangie dan talaut. Kesan saya, P2A LIPI on the top. Apa sebab? Yang membangun dan mengeksplorasi potensi sumber daya Indonesia seharusnya anak-anak bangsa Indonesia.
Berawal dari cita dan asa untuk membangun negeri, memberikan segenap rasa dan tenaga untuk menghasilkan pemikiran terbaik bagi negeri tercinta, kami ada untuk Indonesia. Mewujudkan diri sebagai Lembaga Ilmu Pengetahuan Indonesia yang andal, profesional, inovatif, dan berintegritas dalam pelayanan untuk mewujudkan visi Presiden dan Wakil Presiden. Indonesia Maju yang berdaulat, mandiri, dan berkepribadian berlandaskan gotong royong. LIPI memiliki berbagai pusat penelitian tersebar di seluruh Indonesia yang berada di bawah kepala LIPI, Deputi Ilmu Pengetahuan Hayati, Deputi Ilmu Pengetahuan Kebumian, Deputi Ilmu Pengetahuan Teknik, Deputi Ilmu Pengetahuan Sosial dan Kemanusiaan, Deputi Jasa Ilmiah dan SDM Pendukung Iptek di bawah Sekretariat Utama LIPI. LIPI berperan dalam proses penyusunan dan kelahiran berbagai regulasi dan kebijakan pemerintah terkait aspek ekonomi, politik, sosial, dan budaya. Selain itu, LIPI juga mengelola data dan timbangan ilmiah dalam rangka konservasi keanekaragaman hayati. Menerbitkan tanda pengenal unik publikasi ilmiah terbitan berkala, membuat repositori ilmiah skala nasional, dan memiliki kewenangan sebagai pembina Jabatan Fungsional Peneliti Nasional, serta melaksanakan proses pengukuhan profesor riset di Indonesia. Kini, LIPI juga mendukung percepatan penanganan COVID-19, baik melalui riset berupa kajian maupun produk, maupun pengembangan SDM melalui program Indonesia Memanggil. Untuk mendukung riset, LIPI memiliki 5 kebun raya sebagai pusat konservasi flora ex situ. 2 museum yang memiliki koleksi flora, fauna, dan kesejarahan alam Indonesia, Indonesia Culture Collection yang menyimpan koleksi mikroorganisme terbesar di Indonesia, infrastruktur riset strategis, dan beberapa pusat berskala internasional, yaitu Asia Pacific Center for Eco-Hydrology atau APCE. LIPI telah menghasilkan hasil riset yang berkualitas dan bermanfaat bagi masyarakat, meliputi keanekaragaman hayati, lingkungan dan maritim, ketahanan pangan, pertanian, dan peternakan, kesehatan dan farmasi, material maju, energi, teknologi, informasi dan komunikasi, pertahanan dan keamanan, dinamika sosial, kemanusiaan dan kebudayaan, kebencanaan, inovasi teknologi dan pendayagunaan iptek, pelayanan, pengujian dan pembinaan ilmiah, serta peningkatan kualitas SDM iptek. Kerjasama keilmuan LIPI dalam negeri meliputi kerjasama penelitian, lisensi teknologi, pengembangan, aplikasi dan komersialisasi hasil penelitian, kerjasama non-penelitian, dan konsultasi kepakaran LIPI. Sedangkan kerjasama luar negeri, LIPI melakukan joint research, workshop, pertukaran peneliti dan beasiswa mancanegara, selain aktif menjadi anggota dan focal point di berbagai organisasi internasional. Melalui kolaborasi bersinergi, menghasilkan riset yang berkualitas, LIPI terus memberikan manfaat nyata, kontribusi berarti, dan turut mewujudkan cita dan asa membangun negeri kita tercinta, Indonesia. Berawal dari cita dan asa untuk membangun negeri, memberikan segenap rasa dan tenaga.
Tujuan dari ekspedisi Nusa Manggala adalah kita ingin mengungkapkan daerah-daerah perbatasan. Kenapa pertimbangannya di luar atau di daerah perbatasan? Karena memang masih banyak hal yang memang belum kita ungkap. Ini yang mendorong kita. Tapi negara memang harus hadir. Menunjukkan negara hadir yaitu dengan cara melakukan kegiatan-kegiatan riset di daerah perbatasan.
Indonesia, negara kepulauan terbesar sekaligus poros maritim dunia. Dengan 10 perbatasan maritim Nusantara, 111 pulau kecil terluar tersebar, menyimpan berjuta potensi, namun belum sepenuhnya tereksplorasi. Dan ini Samudra Pasifik, satu perbatasan maritim di timur Indonesia. Di sanalah 8 pulau kecil membentang, seakan membentuk barisan terdepan. Jauh dari peradaban, terhampar rangkaian pesona alam mengagumkan, menyimpan kemewahan sektor kelautan yang luar biasa, dan belum sepenuhnya terjamah. Hingga sebuah ekspedisi tim ilmuwan Indonesia bergerak menuju 8 gugusan pulau. Di antaranya sebuah kepulauan dan 7 pulau-pulau kecil terluar atau PPKT. Menelusuri, mengeksplorasi satu persatu, menguak potensi yang selama ini terpendam. Apakah benar perbatasan Indonesia Samudra Pasifik kaya akan keberagaman biota laut? Berapa banyak biota dilindungi yang ditemukan di sini? Keunikan fenomena sosial apa saja yang mewarnai kehidupan penduduk pulau terluar? Dan mengapa wilayah kepulauan ini dipilih menjadi lokasi ekspedisi? Ekspedisi Nusa Manggala, kisah 8 pulau terluar. Berawal dari cita dan asa untuk membangun negeri, memberikan segenap rasa dan tenaga untuk menghasilkan pemikiran terbaik bagi negeri tercinta, kami ada untuk Indonesia. Mewujudkan diri sebagai Lembaga Ilmu Pengetahuan Indonesia yang andal, profesional, inovatif, dan berintegritas dalam pelayanan untuk mewujudkan visi Presiden dan Wakil Presiden. Indonesia Maju yang berdaulat, mandiri, dan berkepribadian, berlandaskan Gotong Royong. LIPI memiliki berbagai pusat penelitian tersebar di seluruh Indonesia yang berada di bawah Kepala LIPI, Deputi Ilmu Pengetahuan Hayati, Deputi Ilmu Pengetahuan Kebumian, Deputi Ilmu Pengetahuan Teknik, Deputi Ilmu Pengetahuan Sosial dan Kemanusiaan, Deputi Jasa Ilmiah dan SDM Pendukung Iptek di bawah Sekretariat Utama LIPI. LIPI berperan dalam proses penyusunan dan kelahiran berbagai regulasi dan kebijakan pemerintah terkait aspek ekonomi, politik, sosial, dan budaya. Selain itu, LIPI juga mengelola data dan timbangan ilmiah dalam rangka konservasi keanekaragaman hayati. Menerbitkan tanda pengenal unik publikasi ilmiah terbitan berkala, membuat repositori ilmiah skala nasional, dan memiliki kewenangan sebagai pembina Jabatan Fungsional Peneliti Nasional, serta melaksanakan proses pengukuhan profesor riset di Indonesia. Kini, LIPI juga mendukung percepatan penanganan COVID-19, baik melalui riset berupa kajian maupun produk, maupun pengembangan SDM melalui program Indonesia Memanggil. Untuk mendukung riset, LIPI memiliki 5 kebun raya sebagai pusat konservasi flora ex situ. 2 museum yang memiliki koleksi flora, fauna, dan kesejarahan alam Indonesia, Indonesia Culture Collection yang menyimpan koleksi mikroorganisme terbesar di Indonesia. Infrastruktur riset strategis. Dan beberapa pusat berskala internasional, yaitu Asia Pacific Center for Eco-Hydrology atau APCE. LIPI telah menghasilkan hasil riset yang berkualitas dan bermanfaat bagi masyarakat. Meliputi keanekaragaman hayati, lingkungan dan maritim, ketahanan pangan, pertanian, dan peternakan, kesehatan dan farmasi, material maju, energi, teknologi, informasi dan komunikasi, pertahanan dan keamanan, dinamika sosial, kemanusiaan dan kebudayaan, kebencanaan, inovasi teknologi dan pendayagunaan iptek, pelayanan, pengujian dan pembinaan ilmiah, serta peningkatan kualitas SDM iptek. Kerjasama keilmuan LIPI dalam negeri meliputi kerjasama penelitian, lisensi teknologi, pengembangan, aplikasi dan komersialisasi hasil penelitian, kerjasama non-penelitian, dan konsultasi kepakaran LIPI. Sedangkan kerjasama luar negeri, LIPI melakukan joint research, workshop, pertukaran peneliti dan beasiswa mancanegara, selain aktif menjadi anggota dan focal point di berbagai organisasi internasional. Melalui kolaborasi bersinergi, menghasilkan riset yang berkualitas, LIPI memberikan manfaat nyata, kontribusi berarti, dan turut mewujudkan cita dan asa membangun negeri kita tercinta, Indonesia.
Ekspedisi Widya Nusantara untuk tahun 2018 ini kita angkat dengan topik eksplorasi keanekaragaman hayati dan potensi sumber daya laut di ekosistem pelagis Kepulauan Sangieta Laut Utara Indonesia. Indonesia itu adalah pusat dari keanekaragaman hayati laut di dunia. Kalau untuk di darat ada di Brasil, tapi untuk di laut itu ada di Indonesia. Namun masih banyak perairan Indonesia yang belum kita kaji, terutama dari segi keanekaragaman hayatinya maupun potensi sumber daya hayatinya. Dengan tujuan ada 3. 1, mengungkap keanekaragaman hayati laut di perairan Sangieta Laut. Yang kedua, mengungkap potensi sumber daya hayati laut di perairan Sangieta Laut. Tapi yang membedakan kedua ini untuk biodiversitas itu lebih mengarah ke eksplorasi, mencoba mengungkap apa-apa biota yang belum kita tahu, yang bisa memperkaya keanekaragaman hayati perairan Indonesia. Dan untuk bioresource-nya lebih ditekankan pada biota-biota yang bernilai ekonomis penting. Kita berharap nanti kita akan targetkan minimal ada 2, ikan sidat dan ikan tuna yang memang benar-benar memiliki nilai komersial di dunia. Dan yang ketiga, tujuannya adalah melihat keterkaitan antara biodiversitas dan bioresource ini dengan kondisi fisika, kimia, geologi dari perairan di sekitar Sangieta Laut, mengingat perairan ini banyak dikelilingi oleh gunung laut, dan kondisi fisika, kimia, oceanografi di perairan yang memiliki gunung laut itu sangat spesifik. Secara fisik, kimia maupun geologi itu sangat menarik untuk dikaji. Banyak sekali tantangan di awal kita mau memulai kegiatan ini karena memang lokasinya itu rentan. Tapi buat kami untuk bisa mengkaji potensi keanekaragaman hayati laut Indonesia, apalagi di wilayah-wilayah perbatasan, kita memang harus punya keberanian ya, harus punya tekad yang lebih kuat karena ya itu tadi begitu besar tantangannya. Nah, terutama tahun ini ya saya kira ada banyak kejadian gempa bumi dan gunung meletus, dan barusan adalah kejadian gempa bumi di Palu Koro. Nah, patahan atau sambungan dari sesar Palu Koro ini sebenarnya mengarah ke Laut Sulawesi, dan kita sempat mengkhawatirkan itu akan sampai ke perairan Sangieta Laut. Sebenarnya dari perairan selatan Sangieta Laut sampai utaranya itu di Miangas ya, itu adalah gugusan gunung laut dan merupakan bagian dari Ring of Fire dunia ya. Jadi cincin api bawah laut Indonesia dunia yang melingkar dari Samudra Hindia hingga ke utara Pasifik. Jadi melewati Sumatera, Jawa, Nusa Tenggara, terus ke utara hingga ke Sulawesi, dan dia tembus sampai Sangieta Laut tentu saja, dan ke Filipina hingga ke Jepang. Makanya daerah-daerah ini selalu sangat rentan, dan salah satu yang menjadi target kajian kita adalah Papatua Seamount ya, atau gunung api Papatua yang ada di depan kita sekarang. Sebenarnya kita ada 5 jam di depan gunung api tersebut, jadi kita akan mengarah ke situ malam ini untuk melakukan kajian.
Untuk pelayaran ekspedisi Widya Nusantara 2018, KR Baruna bertolak dari Pelabuhan Bitung menuju Tauna untuk stasiun jumlah 35 stasiun seluruhnya. Setelah stasiun 17, KR Baruna Jaya 8 akan masuk di Tauna. Untuk acara open ship, kita membuka untuk semua masyarakat Tauna. Dari
Okay, Dr. Adi, good morning.
Morning all.
You ready for the substitution?
Yes,
Okay, I believe most of the speakers already in the Zoom room. So, yeah, we already can see Professor Arai and then also Professor Teguh Pristiwadi and then Dr. Augie already here also. We're waiting for Dr. Hagi Yulia Sugeha and Professor Casey then already here with us too. And yeah, and then we have to wait. Mr. Marenda Pandu already here. Dr. Suyadi. Dr. Suyadi already here also, and also, yeah, we're only missing for Mr. Hanung Mulyadi. But can we? Yeah, it's almost 9 o'clock, so we will start the conference for today precisely at 9.
Morning everyone.
Good morning, Hogi.
Morning, everybody.
Good morning, Pak Agi and everyone.
Morning.
Okay.
Good morning, everyone.
Good morning. Hopefully everybody are in the good shape condition. Yeah.
Good condition, Ibu Sofi.
Alhamdulillah, Pak. Okay, Dr. Adi, can we start now? Yeah. In my watch, it's already 9.
Yes, you can start it.
Okay, thank you. Selamat pagi, Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody.
Good morning.
Morning.
I hope you're freshen up today. Yeah, we're going to start our second day of the International Conference on Ocean and Earth Sciences, Icoes, day 2. In today's agenda, today's topic, we will be hearing presentations from the invited speakers and also from the oral presentation from the impact of climate change on marine and coastal biodiversity and its possible strategic plans for adaptation and mitigation. As for our morning session, Dr. Adi Purwandana will act as our moderator for today's morning sessions. In this session, we will be hearing for 5 invited speakers and 3 oral presentations. Okay, Dr. Adi, whenever you're ready, time is yours.
Okay, thank you, Mbak Sofi. I'd like to assist you all to the this session. Distinguished invited speakers and all participants of the international conference on the ocean and earth sciences, Indonesia Science Expo 2020. Good morning and assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, we are now entering the morning session of the second day of the conference, and within the next 3 hours, we are going to hear the presentation from 5 invited speakers and from 3 oral presenters. For the invited speakers, we have Professor Dr. Takaumi Arai from University Brunei Darussalam. His expertise is in marine biology and ecology. The next one will be Professor Dr. Teguh Pristiwadi from Research Center for Oceanography, Indonesian Institute of Sciences, Indonesia. His expertise is in fish taxonomy. The next one will be Dr. Augie Shahailatua from Research Center for Oceanography, Indonesian Institute of Sciences. His expertise is in marine biology. The next one will be Dr. Hagi Yulia Sugeha from RCO LIPI as well. Her expertise is in marine biology. And the last one, we have Professor Dr. Casey Dunn from Peabody Museum, Yale University, USA. His expertise is in marine biology. Afterwards, we will have 3 oral participants. The first one will be Mr. Hanung Mulyadi from Research Center for Deep Sea, Ambon, Indonesia. The second one will be Dr. Suyadi from Research Center for Biology, LIPI, Indonesia. And the last one will be Mr. Marenda Pandu Rizky from RCO LIPI. Okay, before we start the presentation from the invited speaker, I'd like to remind you all that each invited speakers will have around 20 minutes, including the Q&A session. And for the oral presenter, we'll have 15 minutes, including the Q&A session. And I'm sorry, I will interrupt you and informing you during your presentation once you have 5 minutes left. Okay, for the first occasion, I'd like to invite our first invited speaker, Professor Dr. Takaomi Arai. Professor Arai would like to deliver the topic entitled Migration and Habitat Use of Tropical Anguillid Eels of genus Anguilla. Okay, are you ready, Professor Arai? The time is yours. Okay, you may
so,
set it.
Yeah, can I share the screen?
Yes, you could share it.
I don't know how to share the. I don't know.
Okay.
Wait. I already shared the. Wait. This one is slider. Okay. Okay. Selamat pagi semua. Very good morning, everyone. Thank you for inviting for this conference. So my name is Takaomi Arai from Brunei Darussalam. Today's my topic is because I don't know. I also act a host, so someone interrupt to ask me. Anyway, today my topic is migration and habitat use of tropical anguillid eel of genus Anguilla. So this is a brief introduction. The life history of Anguilla is basically is a catadromous eel. It means that the spawning area is located in the offshore. Then they migrate to freshwater for growth. Then after that, they back to the ocean again for spawning. But not all eel migrate to the freshwater environment. Later, I will explain detail. So this is a global distribution of anguillid eels. So far, we have 19 species, subspecies, almost in the world. Of course, some exceptional in the west coast of Africa and also South America and so on. But we can see Anguilla almost worldwide, cosmopolitan species. Then 13 species, as I shade orange color, these species are recognized as a tropical species, which are live in the tropical region. The remaining 6 species are distributed in the temperate region, Atlantic and also East Asian country and also Oceanian country. Then we can find 7 species and subspecies in the Indonesia and also Malaysian region. It means that this area is a hotspot in terms of anguillid species. Then, based on the phylogenetic study, still we are debating Anguilla one of the species, Anguilla mozambica or Anguilla moronensis is a basal species. But anyway, the anguillid eels originated in somewhere in the Indo-Pacific region. That is why this region is very important to study anguillid eels. This is more detail about the diversity of anguillid eel in the world. As you can see, this region, we can see quite many species. It's a quite hot spot. However, the higher latitude, we can see only monospecies. North America is only Anguilla rostrata. Europe country is Anguilla. However, Southeast Asian country and also some African country, we can see plural species. That is why to study tropical anguillid species is very important to understand the nature of primitive form of global migration of anguillid eel. And also, recently, tropical eel are considered as a very important commodity because of the collapse of abundance of temperate eels. So we are targeting for the tropical eels as to compensate temperate species. That is why we need to study a lot of work on the life history and also migration and so on. To understand the life history as well as to minimize the decline of the population. This is a human impact of life history. Throughout the whole life history, eels are subjected by both natural and also anthropogenic impacts such as overfishing, habitat degradation, pollution, parasite infection, and also prevalence, and also global climate change. Most of the global change affect on the larval life history, which are oceanic life, this life history. Then after recruitment, other environmental factor, habitat degradation, pollution, and so on, severe impact for their life. Before starting a migration study, because as I mentioned, there are few studies on the tropical eel. So at first, I just briefly introduced my past study on the maturation and also spawning ecology of tropical species. We conducted in the Java near Cilacap and also west coast of Peninsular Malaysia. We focus on Anguilla bicara, and also Anguilla bengalensis for yearly survey and also historical observation. Then this is detail about staging, a maturation stage for monthly survey. But today, due to the time limited, I do not explain detail. But what I want to highlight is tropical eels are going to spawning for almost every month because we could find a fully matured stage such as stage 5, stage 4, or we could find almost every month. That characteristics are quite different from that of temperate eel. Normally, temperate eel migrate to ocean for spawning during autumn time, late autumn to early winter for almost 6 months. However, tropical eel is going to ocean almost every month throughout the year. This is quite interesting because the facilitated mating between adults of different year class or cohort from and also from a different habitat. So they can meet anytime in the spawning ground. So it accelerates for the spawning. So the tropical eel higher, how to say, can maintain their population compared to the temperate species. Then this is life history of angulid. As I mentioned first, this species are catadromous eel. However, recent study found that not all eel are necessarily migrate into the freshwater habitat. We can find marine eel, sea eel, which is never migrate into the freshwater, but some other population, they migrate between the freshwater, seawater, estuarine frequently. We normally call estuarine eel, very complicated and also facultative or opportunistic migration. Then we consider that is it really angulid? Is it angulid eel or catadromous eel? Now still we are debating. But however, many of temperate eel have a diverse migratory history. We can find all temperate species have such diverse migratory history. However, compared to the temperate species, there are few studies available on the tropical eel. That is why we employ some technique to understand, to determine, to trace the migratory history. This is using fish otoliths, because as you may know, the otoliths perpetually, permanently grows from hatching until die or date of capture. Then we normally, the fisheries ecology and also fish biologists, normally they use otoliths as age determination. But not only that, some element, especially for strontium, also accumulate throughout the life history depending on their habitat. When, as you can see this diagrammatic figure, when the fish stay in the freshwater, the strontium level also low. But once they migrate to the sea, the strontium level also high. Then we can trace the migratory history, especially for diatomaceous fish, anguilla, and also salmon, and also other many diatomaceous fish species. So we apply this technique for further life history analysis. So at first, I just showed this is a tropical eel otolith, which is collected in the Cilacap or Anguilla bicolor. So as you can see, the all sample, we have a red spot in the center region because all eels hatched in the ocean. So strontium level is quite high. But after recruitment to the coastal area, the strontium pattern is quite diverse. Anyway, this is a two-dimensional image of normally we call the strontium mapping. This is a freshwater, completely freshwater eel, which is we can consider the catadromas eel. As you can see, the blue color means that strontium level is quite low. However, other sample, as you can see, this sample is maybe 2 to 3 years. They remained in the freshwater, but they shift. They showed habitat shift from freshwater to estuarine on the high saline water. Then this sample is continuously stay in the estuarine habitat. Otherwise, only short stay in the freshwater. Then they move to marine, otherwise a brackish water environment. Then this sample also showed the habitat shift. And this sample, this specimen showed multiple habitat shift during their life. It means that not only a temperate eel, tropical eel also a quite high diverse migratory history. However, compared to the temperate species, there are few information available. Then this is our diversity study conducted in the Peninsular Malaysia. We actually survey whole Peninsular Malaysia for 4 years. However, we can only find ungulate eel in the northwest of Peninsular Malaysia. And also interestingly, this species composition is quite similar to that of Sumatra. That is why we can share that those eels share the spawning ground. Then the most of the species is Anguilla bicolor, the dominating more than 88%. Then next is Anguilla bengalensis. Then the least species is Anguilla marmorata. This composition is quite similar to the Sumatra Island. But we don't know the habitat preference and also habitat shift. Then we all further study focus on the Penang Island. This is a intensive survey from some river. We collected eel from upstream to downstream. Then this is a species composition. This pie chart, black color means that Anguilla bicolor. The white color means that Anguilla bengalensis. As you can see, Anguilla bicolor prefer to stay. We can find upstream to downstream, but most of sites we could only find in the downstream to midstream. However, Anguilla bengalensis prefer to stay more on the upstream until midstream area, quite cooler temperature and also a high elevation and so on. But based on this habitat study, the diversity and also habitat use study, we do not know. Even we found some sample in upstream, the eel permanently lived in the freshwater, or they move frequently or not. That is why we need to apply this otolith strontium technique for further confirmation of migratory history. Then anyway, at least this ecological study, we found the habitat preference. It is coming from interaction between the intra or interspecific variation and also local environmental condition. Then we applied, this is recently we published a paper. As you can see, this is habitat segregation. The top figure is Anguilla bengalensis, and also the remaining figure is Anguilla bicolor. It means that Anguilla bicolor shows a quite diverse migratory history compared to the Anguilla bengalensis, which is at first we explained the Anguilla bengalensis. All sample we study 41 specimens show the typical freshwater life history. Without no migration to the estuarine and also marine water. However, the broad range of autolysis calcium ratio found in Anguilla bicolor. As you can see, we also found some freshwater residents, but this specimen stayed in the brackish water to the coastal water almost whole life after recruitment. Then this sample shift. At first they stayed in fresh water, but after 2 to 3 years, they moved to higher saline water. Then I don't know why. Then this sample showed at first they stay in the brackish water, but they moved to fresh water. They showed habitat shift. The last one is they shift habitat multiply in the life history. So the autolysis strontium calcium ratio also confirmed that Anguilla bengalensis preferably stay in the fresh water, cooler water, and also higher elevation. However, Anguilla bicolor is quite a diverse migratory history. So it means that depending on the species, habitat preference are different. And also we found Anguilla bengalensis has a more restricted habitat preference. That is why this species is more vulnerable by the anthropogenic and also natural impacts, especially for climate change may severe influence for the habitat degradation of the species. That is why to conserve and also to protect ungulated species, we need to consider this issue by species. Thank you very much. This is my presentation. Sudah.
Okay, thank you, Professor Arai, for very nice sharing dealing with IL's migration characteristic. So I'd like to check. We have maybe we have 1 or 2 minutes left for the Q&A session. Do we have a question from chat? Not yet. Just interested, may I have a question, Professor?
Yeah, sure.
I'm not familiar with IL's migratory characteristic, but I'm just curious, is there any study trying to correlate the impact of ENSO phenomenon, which may be impacting and contributing to the variability of IL's migration?
What factor? You said anthropogenic impact?
ENSO phenomenon.
ENSO, yes.
Yeah.
This is quite a severe effect, especially as I mentioned for this climate change, quite highly impact on the larval transportation after hatching to the transported by the oceanic current. The ENSO also impact because during the ENSO time, the temperature also change. This is highly impact on the mortality rate for the larvae. And also transportation mechanism, especially for transportation route, also different during ENSO. So some population they cannot migrate to the recruit to the coastal area by the ENSO impact. And also the recruitment abundance of grasses also highly fluctuate. Actually, we study with Dr. Sugeha in the Poigal River. She has a good data. Actually, the ENSO is very high impact for the mortality rate and also the recruitment pattern for the unguided eels.
Okay, thank you, Professor,
Welcome.
for a very nice sharing. Okay, since okay, it's the time to move to the second invited speaker. I think for the second invited speaker, I'd like to invite Professor Dr. Teguh Peristiwadi from LSI LIPI. Professor Teguh would like to deliver the topic entitled Ichthyology Research and Biodiversity of Marine Fishes in Indonesia. Dr. Teguh, Professor Teguh, okay, you already joined with us.
Yes, I'm here.
Okay, the time is yours, Professor.
Thank you. My PPT, please.
Would you like us to?
Yeah.
Okay.
I can share my if you want.
Okay. Yeah. Okay. Maybe you could share by yourself.
Okay. Thank you. Just a moment, please. Okay. You can see my
yes,
my presentation.
Yes, Professor. Slideshow, please.
Yeah. Just a moment. Okay. Okay. Thank you very much, Pak Adi. Well, thank you to you because before Pak Wiwit will be our chairman this morning. Thank you, Pak Adi. And I would like to thank you also to organizing committee to invite me to be an invited speaker in international seminar of Icoes. Ohio, Arisa, and good morning, Professor Kase.
Ohayou gozaimasu.
Yeah, and I would like also to thank you all of my friend Pak Ogi and other together here this morning. And ladies and gentlemen, good morning and maybe good afternoon for everyone. This morning I want to talking about ichthyological research and biodiversity of marine fishes in Indonesia. My outline about the introduction, I will give you a brief introduction about Indonesian country, Indonesian water, and what is my purpose. And then also I want to give you about methodology, about ichthyological research and marine fishes biodiversity in Indonesia. And then finally, I will give you about the result of Indonesian fish biodiversity. Well, ladies and gentlemen, everybody knows that Indonesia situated in the tropical Indo-Pacific, and then comprised about 17,504 islands with an overall coastline of more than 100,000 kilometers. So it is very, very long. And then, of course, Indonesian with the other country, like Philippines, like Papua New Guinea, include in the Coral Triangle area. Indonesia is really lying in the center of CTI with other country. And then some studies also remark between Sumatra in the west, New Guinea in the east, and Philippines in the north, really having the highest marine biodiversity, including, of course, fish in the world. Well, with the vast area in Indonesian water, we have coral reef because we have a temperature is very warm here. We have no four season. We have coral reef, we have seagrass beds, we have mangrove, coastal areas, and continental shelf, and also the other ecosystem And maybe this is very special for Indonesia because inside of Indonesian water, we have deep sea, Banda Sea. Until now, we have no idea what we can found in this area. Well, this is my purpose, because this is very, very long motivation for me. I want to know the biodiversity of marine fishes from Indonesian waters. Well, if we are talking about ichthyological research, almost most study of Indonesian water were made for navigational purpose. And of course, this purpose now we can have many research now, resulting now in a growing emphasis of research, including ichthyological research. And everybody knows the scientific research really, in fact, in early days began with the visit of European explorer and naturalist in the early of 19th century. And for Indonesian and around country, for beside Indonesian like Malaysia, Philippines, Brunei Darussalam, maybe Australian also, the predominant researcher of Indonesian fish in this period was Peter Blecker. He arrived in Indonesia in 1942, and then he died in 1878. It is not very long time for Peter to live in Indonesia and to live in the world, but he already make very big work in Indonesia. And then of course, after Peter Blecker dead, this work or scientific work on fish continue by the other scientists. And this is most of my friend in Indonesia, because Indonesia is a very good destination, very good location that we can work together in Indonesia. We have Jack Randall, we have also Jerry Allen, Mark Edman, Martin Gomon, also Professor Matsura and Professor Kimura. He really motivated me and asked me to be try to study of the fishes in Indonesia. And of course, the other of my friend, Professor Motomira from Kagoshima University, Professor Yoshino from Ryukyu University, and then also we have Peter Lass, Paxson, Barry Russell from Northern Territory of Australia. Of course, many other scientists from Japan, Australia, France, US, or the other country of the Europe. Well, this is what I've done already. Ladies and gentlemen, I tried to find any source that have long been published, even by both government agency or other publication, by within country or from international source. I use some checklists, like checklist by Randall and Lim, as editor, and then also Allen and Adrim. He already give us the fish species of coral reef in Indonesia. Also NGOs work like Allen and then Edmar and Raja Ampat and in center of Sulawesi. I use almost all checklists in Indonesia. And then I also try to have a species from the field guide that I have here. and also field report, scientific report, like Snellius, Rumpius, and then Karubar, and the last in 2018, we have collaboration with Li Kongqiang Museum, Raffles Museum. In 2018, we have a collaboration at South Java. In South Java, we make a very, very good collection there. And then maybe next year in April, we have already a checklist of the species that we can found there. Because in this collaboration, we already found 223 species. But this account of species not yet including to my database. So we found about 62 families, and this is preliminary result for you. From the 223 species, we have 85 species unrecorded before. Well, this is status of the data that I use already. Checklist, Allen and Adrim 2003, Randall and Lim 2000. This is a checklist of the South China Sea, and then Allen and Erdmann 2009. This is in Raja Ampat. I also use the data from the visual census from our friend from Bitung, also from Cormap, also from NGO like a Nature Conservation, and then from University, Patimura University, and then also from Regional Planning and Development from Center of Sulawesi. Well, the other data, I also use online data from any museum. I use from Afrobis, Bird Museum, of course, California Academy of Science, Natural Museum, Natural History, Washington, and also JBAF, Global Biodiversity Information Facilities, and also I use from the Fish Base, from Museum National d'Histoire Naturelle from France, any museum, and then also from Japan, Kahaku. Well, this is from online data. In your right slide, we have here like type of the data that I receive from the online data. This data is from Museum National d'Histoire Naturelle from France, and then this is from Fisheries Research Laboratory Mie University, and in Saga Island, close to Nagoya. Well, I also use scientific collection. So I already make a daily collection from 2005 until now. So almost every day I got species from our specimen from the fishermen. Also, I make a market observation. This is also daily observation. So about geographic distribution category species, I follow fish base, and I also follow California Academy of Science. So globally, I can include it into Indo-Pacific, Indo-West Pacific, Western Pacific, West Central Pacific, and circumtropical, Pacific Ocean, Indian Ocean, Atlantic Ocean, and also I give to classified to other distribution. Well, this is the brief review of ichthyological research in Indonesia. So, sorry, if I pull all data, and I make a diagram like this, So in the your left, we have Blicker, Valenciennes, Cuvier, Gunther, Fowler, Temming-Sliger, Lacépède, Bloch, and so on and so on. But in these ichthyologies, big ichthyologies in Indonesia, we have also here Randall. Randall. He is a very, very big scientist on fish. And for the moment, my data, we have about more than 600 species described by Blicker. So Blicker, Valenciennes, Valenciennes, and Cuvier, Gunther, Fowler. This four is very, very big scientists, very big ichthyologists in Indonesia, and they got about more than 38.44%. The 10 ichthyologists found and described about 38.5% of the Indonesian fish. And in the right side, we can found here. I tried to make a simple figure. We can show here the accumulation of the species described by time. Well, in this figure, here we have really a very, very big, increasing. Oh, may I can say that? Describe a species in Indonesia. About in 1825 to 1875. This is really very big increase of new species in Indonesia. Well, this is very, very simple question for us, especially for me. So, how many fish species in Indonesian water? This is very simple question, but it is very, very difficult to respond. And this is the total data that I receive. I work about 10 years already to compile all data. So more about 60,000 data, not specimen, data that I found from my compilation. From the checklist, about 21%. Expedition report about 0.5%, and market observation about 6.6%. And online data, I got this very, very much, about 30.4%. And my scientific collection, this is also about 70.1%. And visual census, about 24.4%. Well, if I put all data, in the map, simple map, so almost all part of Indonesia, all part of Indonesia. I got, this is maybe too far, this one. Maybe I make a mistake about the position. This is also 2 or 3 in the Philippines. And this is from South China Sea. So we can see here the region or area in Indonesia. So Moluccas, from North Moluccas, Center, Sulawesi. This is very, very much data there. And then for the Papua, we have so just in head of head of the Papua, here Raja Ampat, Cendrawasih, and Kaimana, and then also in the Lombok, Bali, and Nusa Tenggara is also very, very much data there. And then also from West Java. Yeah, I think this is a very, very old, very, very old position there. Yeah, because VOC. Yeah, and then also in Kalimantan. Yeah, also Kalimantan. Yeah? West Kalimantan and East Kalimantan. So many data there. Well, so if I check the position, I check the museum where the Indonesian specimen was deposited. Yeah, of course, we have in Museum Zoologi Bogor, MZB in Bogor. Yeah, and we have also reference collection in Research Center for Deep Sea in LIPI in Ambon. Yeah, this is very special here because some of the Rumpus collection were deposited here. Yeah, and then Bitung Research Station. This is my my office now, my laboratory now, and then also in Jakarta. And I heard all specimen from Jakarta is now moved to Bogor. And how about in the world? So Indonesian specimen also deposit in National Museum of Natural History, Washington. Yeah? And then Western Australian Museum. And then Natural Museum of Nature and Science, Tokyo. Yeah? Several years ago, I have a chance to visit there. And then Bernice Museum. Yeah? This is Randall Museum. And then British Museum of Natural History in London, California Academy of Science, San Francisco, America. Well, this is the result of Face of Indonesia. Yeah. So I found about 5 class, 52 order, 324 families, and general about 1,433, and for species, yeah, almost 5,000 species. This data, yeah, this data is 2019, because in 2020, yeah, I update the list because I have the other checklist, and I put in my data. Yeah, we have 5,500 species, more than 5,500 species. But the problem is now I cannot give you the very, very detailed species, number of species, because I'm not yet checked of the synonymous, yeah, synonymize of the species. Well, and then I also categorize my data, yeah, in island, yeah, big island, yeah, big island. Like for example, for Lembe Island in North Sulawesi, I class to Sulawesi Island. Yeah, in Kalimantan, from Derawan Island, yeah, in the Kalimantan, East Kalimantan, I put together in the Kalimantan, and also species I found from South China Sea, I also put in Kalimantan. So I make very, very big class. Yeah. So Bali, we have more than 600 species. Jawa, more than 700 species. Kalimantan, more than 1,800 species. Moluccas, about 1,300. Nusa Tenggara, so almost the same. And then Sulawesi, I have about 3,000 species there. Yeah? And then for Sumatra, we have more 900 species. Of course, of course. Maybe some of you ask me, why Kalimantan have a higher diversity than Papua? Because, as I said to you, I include a checklist of South China Sea to the data of the Kalimantan. Why I include there? Because some of area of Kalimantan is also part of Indonesia. And then also in South China Sea, we have the same habitat, almost the same habitat, like in North Kalimantan. But I'm sure with my new data, my update data, I will found in Papua about 1,800 on 1,900. And Sulawesi also, I found already about more than 3,000 species. So with just a minute, because I have to check synonymize of data collected. And how about families now? So Pomasandridae about 11%, Labridae 9%, Gobidae 4.7%, Apogonidae about 4.5%. If I compare to other area, the top 4 or 5 of families almost same. The difference just the position, just a position. Maybe Pomasandridae will to be a second and Labridae 1, Gobidae, Apogonidae, like this. Just the position is different, but the 4 or 5 family, it is almost same. Well, this is about geographic. So if we can see here, the 3 areas of our species is distributed in Western Pacific, Indo-Pacific, and Indo-West Pacific. And other area of geographic, we have Western Indian Ocean, and then we have Northwestern Pacific, Northwestern Atlantic, and the other also Eastern Atlantic. This is indicate that really Indonesia is a center of biodiversity. The problem is documentation in Indonesia is very difficult to find. It is very rare. Not like our friend from Philippines. Anything there is well documented. So why now I ask to all Indonesian scientists, please keep your document. So with this document, we can show to the world that we are really center of biodiversity. Because Filipina can say it. Filipina can say that they are center of center. So why? I ask to all scientists in Indonesia, please keep all document of biodiversity. Not only data, but also specimen. Well, I try to compare the biodiversity of marine fishes in Indonesia with the other area. We have here Red Sea about 1,000, Andaman about 1,700, South China Sea about 3,000, Filipina about 2,000, and then New Guinea around 2,000, Indo-Australia, books of Blecker, Weber, and Beaufort about 2,778. So really here, we have 5,000 species. So this is really Indonesia have more species than the other waters. Well, and this is my conclusion. In 2008, when I started to compile the data, Indonesian species of fish, about 54.4% of the diversity in the Indo-Pacific. But after 10 years of work, the diversity of reef fishes in Indonesia water represent now about 68% of the fish in Indonesia West and Central Pacific. And this diversity also in Indonesia, about 13.4% of marine fish in the world. Well, for the finish my presentation, I would like to thank you all scientists and technicians at Research Station RCO LIPI. I mentioned Ricardo Y, Mokhtar Jabar, Lapai. Lapai is a technician in a research center for deep sea in Ambon. And of course, I would like to thank Professor Matsura, Professor Kimura, Dr. Kasim Musa, and Professor Rahayu, because they are motivated me. They asked me to work on fish taxonomy. So thank you and terima kasih, Pak Adi.
Thank you, Pak Teguh, for very nice and impressive presentation. Now we have, I'll check the question in the Zoom chat. Okay, we have 2 questions, and actually I have also 1 question maybe after the participant question. So the first question from Dr. Zainal, Professor Zainal from LIPI. Pak Teguh, based on your best estimate, how many Indonesian fish species then? It seems we have the same diversity of fish species with Philippines. Maybe short question, short answer, Pak Teguh.
Okay, I will try to move behind. Philippines, we have 2,145 in this slide. Fish base now, they for Indonesian, they have about more than 4,700. But this is include freshwater fish, freshwater fish. But my data already show we have in Indonesia actually without my new data, we have 4,900 species. And I said that with my update data, we have more than 5,500 species. But I have to check because synonymize in taxonomy studies is very, very important. Yes, Pak Adi?
Okay, maybe one more question from participants, since we have no time left for this session. Maybe I'd like to move to the question from Mas Arif from RCO LIPI. Professor Teguh, do you have any records or data on which species that are endemic, native, and invasive?
Yeah, okay, thank you. This is very good question. In my data, I have no parameter about this. Different with the fish base. Because in fish base, they classified native or endemic or like this. But for me, no. I try to collect the data of any fish, marine fish in Indonesia. So I have the data about the native species, fish species, or endemic species. Sorry, maybe next time because if we work with very, very big data.
Okay, that will be a challenging.
Wow,
For
this is very, very difficult. And then, yes, okay.
Okay, thank you, Pak Teguh, for a very good and nice sharing.
You're welcome.
We have to move to the next invited speaker. I'd like to invite Dr. Augie Shahelotua from RCO LIPI. Pak Augie would like to deliver the topic entitled Notes on Flying Fish Diversity in Indonesian Waters. Pak Augie, are you ready?
Can you? Okay.
Okay, the time is yours, Pak Augie.
So thank you, Pak Adi, and good morning, good afternoon, and good evening for you all around the globe. You see my PowerPoint?
Yes, could you please slide show your PowerPoint?
Okay, I did, but I'm not. Okay, I'll try again. Is it?
Yes.
Is it working? It's working now?
Yeah.
Okay, so thank you very much again. I think I have to start with thank you the organizing for inviting me to talk in this very special event. I really appreciate Dr. Yuli Hagi and her team for working hard during the pandemic to organize this event. So special thanks for you all. And I will have outline for my talk. First, I would like to start with the flying fish fishery in Indonesia. So what is important for Indonesian fishery? And then I would like to make just several notes on the diversity of flying fish. So I divided by the time, early 20th century and then the late 20th century. And based on our result from the study 24 2010, and end up by the summary and conclusion. So all participants, we categorize the flying fish as a small pelagic fish. Flying fish has a streamlined shape, so make flying fish can move fast in the water. So the total length could be reached 45 centimeters, but normally the length of a flying fish below 30 centimeters. Flying fish has a very long pectoral fin, sometimes can reach the anal fin, and caudal fin of flying fish deeply forked, and you probably couldn't see, but the vertebrae numbers of flying fish is around in between 39 and 51. So this is a brief character of a flying fish. So Indonesia fishery, we already have the production data since 1975 of the flying fish production in Indonesia. And if we see for the last 40 years, so basically the production is around 50,000 till 20,000 ton per year. So it's not much change. And the production basically is less than 1% of the total Indonesian fisheries production. So it's not big fishery basically, but we found that the production in Makassar Strait and Flores Sea for the last 40 years, it's extremely declined. So based on the, because there's a very heavy fishery of flying fish in that area. So so the flying fishery of flying fish is not catch the fish itself, but also they collect the egg or roe of the flying fish. That's why probably make the production of the fish flying fish in Makassar Strait and Flores Seas decline significantly. So they use a bale. It's like a coconut leaf. They put the coconut leaf together and then floating on the water, and when the flying fish spawning, they can come and attach the egg on the bale. So on the right side of my slide, you can see that some production of raw of flying fish, and we can use as a food for human. So it's quite tasty if you like to have them. So this is the latest classification of flying fish in the world and also in the Western Pacific. If you see on this map, so we classify, or so ethologists classify, there's 7 families of flying fish in the world, and it's the total number of genus is around 71, and in Indo-Pacific it's about 31. So this is the latest classification of flying fish in the world, globally, and in the Western Pacific. So the highest number of species it's belong to the genus of Chelopogon, and is 31 in the globe and 14 in the Western Pacific, following with Cypselurus and Hirudintus. So that's the 3 very, very high in number of species in the world and in the Western Pacific region. Sorry. So in the early 20th century, Weber and de Beaufort classify the flying fish into 18 species, but until now, the only 5 species valid in name. So this Parex Scotus brachypterus, number 1, and number 3 Exocultus folitans, and number 9 Cypselurus hexasona, and 11 Cypselurus Poecilopterus, and number 15 Cypselurus opistopus. This the only 5 of 18. It's still valid until now. So it is quite a lot of change of the taxonomy, or yes, taxonomy of flying fish since the early 20th century. In the latest, in the late 20th century, again, Parin is an echologist from Russia. He split it, the Cypselurus into 2 genus, so Cypselurus and Chelopogon, and then the number of Chelopogon is more than number of species. I mean, from the Cypselurus. So Cypselurus is only has 6 number of species, but Chelopogon has a 10 number of species now. So this so Chelopogon is I think it's more in terms of number, it's more species number in the world and also in the Western Pacific. So this a very quite big change for the late 20th century, because before, in the early publication, most of the Cholopogon we recognize as a Cypsilurus, but now we have to change the name of, or try to re-identify again of the sum of flying fish. So from 2004 and 2011, 2010, so basically we had a chance to collect flying fish sample from 6 locations in Indonesia: Western Sumatra, South Java, and Lombok and Bali, and South Sulawesi, North Sulawesi, also in around Ambon and K Island. So there are 6 sites that we had a chance to collect from local fishermen. So we just had a sample from the fishermen catch in that area. So so the total is 14 species that of flying fish that we collected and identified. And Makassar Strait and Flores Sea, they had 11 species so far, and following by Ambon and Banda Sea, it's around 10 species. So if you see on the map, so seems like Flores and Banda Sea, Strait, Makassar Strait, they had it's quite a lot of flying fish species at the time. So based on our study from 2004, 2010, 2011. So we speculate that so probably 2 oceanographic event could be influenced for the high number of species of flying fish. So first is the upwelling in this area. It's quite big upwelling during the southeast monsoon, and also probably the Indonesian trough flow very much influenced in that area. Makassar Strait, Flores Sea, and also Banda Sea. So that's our speculation. That I think it's very important for the future study on why the flying fish species is very abundant in that area. So this some notes for the future study. So I think the population structure of flying fish in Indonesia, we need to do that using the very a new approach. It's like genetic or DNA or something like that. So this is very important. We also need to understand which are the dominant species of flying fish that produce eggs for commercial purpose, because the size of the egg is very important for commercial purpose. So it's mean that maybe not only an egg from all flying fish species that we need, but for commercial one, it's only probably some species, not all. We also need the study on the relationship between the biological aspect, diversity, distribution, abundance, age, growth of and physical and related to a physical oceanographic aspect. It's really important, and of course management for sustainable fishery of flying fish. We need to be considered. Ladies and gentlemen, I would like to end up my talk with say that flying fish fishery is important for Indonesia in terms of growth or eggs production. The main fishery of flying fish is in Makassar Strait and Flores Sea. Flying fish taxonomy is revised quite significant, such as genera of Cypsilurus and Cholopogon. High diversity of flying fish was discovered in Makassar Strait, Flores Sea, and Banda Sea. So this probably hotspot of flying fish diversity in Indonesia. And management plan for flying fish fishery is needed to sustain its resources. So thank you very much for listening and attending. Thank you.
Thank you, Pak Ogi, for a very nice presentation. Okay, we are now entering the Q&A session. Okay, we have one question from Professor Hiroaki Saito. Is there any relationship between upwelling index and fishery landing of flying fish?
So thank you, Professor Saito, for it's a nice, important question. Basically, in terms of diversity, we don't understand. It's just our speculation, but we understand that upwelling event is very influenced for the spawning season of flying fish. So because during the southeast monsoon, June, July, August, sometimes September and October, there is the peak of spawning of the flying fish in Makassar Strait and Banda Sea somewhere, and this event is very related to upwelling because during the spawning of flying fish, the fishermen they have a chance to collect the egg of the flying fish in the area. So basically, so this my comment. So we just speculate that's probably diversity also influenced by the upwelling, but we don't really understand the relationship for that. But for the spawning, that's yes, it's very strong relationship. Thank you.
Okay, we have another question from Pak Fransley Hukom from RCO LIPI. Pak Ogi, what should be done about the management of flying fish or flying fish eggs for the sustainability of the flying fish fishery in Indonesia? This is related to the policy made by the KKP on flying fish management in Indonesia.
So basically, KKP or Marine Affairs and Fishery Minister, they already have the fishery plan for flying fish, but they not focus on the egg production or well, they focus on the only fishery, but but in my point of view, that fishery or catching a fish or flying fish it's not really important, because people or fishermen basically has a target to collect the egg. And if you can imagine that if we catch a lot of egg, it means like we cut the life history of the fish. So it's mean like maybe for the long time, so the diversity could be gone. So that's very important. So I think we work together with the KKP or Marine Affairs and Fishery to have a new approach for management of, because we like the management plan. It's more focused on the how the egg fishery. It's not a flying fish fishery itself as a fish, but for egg fishery. So basically, so that's my short comment for Dr. Fransley. Thank you.
Pak Adi, you unmute.
Sorry. Okay, maybe one more question, Pak Ogi, from Professor Sam. Since the egg of flying fish have high economical values, there are so many fishermen in Indonesia exploit this fish intensively nowadays. Is any study on the stock status and the sustainable management efforts on this fish? Thank you.
I have no idea about that, Professor Sams. I have no result from the stock study from the flying fish. But if you see the production of the flying fish is mostly drop, so far. So I think that's probably because the egg production that we collect egg, and now that we understand that the area for the flying fish fishery, especially for egg production, it's going to be in the east of Indonesia. It's like in Papua and Aru and somewhere in south of Maluku. So that's because probably in Makassar Strait and Flores Sea, the production of egg is very low. So they move to the fishermen from the South Sulawesi. They move to Papua, in between the area they call Fak-Fak, Sorong. That's in between Maluku and Papua, and also in the Aru. And so, but we don't have exactly the stock of flying fish data so far that I understand from my understanding. Thank you, Professor Sam. Yeah,
Okay, actually there is one more question, but since we have to move to the next invited speaker, I hope you could answer it directly from the group Zoom chat. Okay, we now move. Thank you, Pak Ogi. Thank you again, Pak Ogi, for a very nice sharing. Okay, we now move to the next invited speaker. I'd like to invite Dr. Haki Yulia Sukeha. Dr. Haki would like to deliver the topic entitled First Study on the Family Diversity of Marine Fish and Invertebrate Larvae in the Northern Indonesian Seas, a scientific report from the EWIN 2018 cruise of research vessels Baruna Jaya 8 LIPI. Dr. Augie, the time is yours.
Hello, can you hear me?
Yes, clearly.
And can you see my slide?
Yes.
Okay. Thank you, moderator. Assalamualaikum warahmatullahi wabarakatuh. I am Hagi Yulia Sugeha from the Research Center for Oceanography, Indonesian Institute of Sciences. I'm really happy to meet you all today. And as you can see here, today I want to sharing with all of you about some part of our study or our recent research by using vessel Barunajaya 8 LIPI around Sanyet Talaut Island. So the title is a first study on the family diversity of marine fish larvae and marine invertebrate larvae in the northern Indonesian seas. Yesterday, you already followed the presentation from two of our cruise members, Mr. Ramdan, related to the phytoplankton, and also Dr. Rozirwan from Sriwijaya University, Palembang. So today, I will not talk about real plankton, but only I will sharing with you about the diversity of the fish larvae and invertebrate larvae that we collected from that cruise. So actually, I'm not the expert for fish larvae, but because I'm the coordinator of the cruise, I have a responsibility to report to all of you. I mean, a kind of scientific report about this study. So that's why in this authors list, not only me, but also included Dr. Charles Simanjuntak from IPB, Hanung from Research Center for Deep Sea, Oni from RCO, Anna, Dia, Ernawati from RCO, and also Edwin from Polytechnic Tahuna, and also Hamsia Ahmad from Halmahera, from the University of Hairun, Ternate, Halmahera Island, and then Aaron from KKP Tahuna, and also another two technicians from Research Center for Oceanography, Johan Picasso and Rihanna. And last but not least, we have another two academicians, a teacher from Samratulangi University, Faculty of Fisheries and Marine Science, Dr. Medi Ompi and Dr. Nego Elvis Bataragoa. But I also want to inform you that two of our members, Mrs. Airen Makakombo and Mr. Johan Picasso, just passed away this year because of sick. So we are very sorry about that. So this is one of our motivation to carry out and to report to all of you about our work. I hope you will enjoy for the next at least 10 to 15 minutes to hear about our recent research. So why important to conduct marine biodiversity in Indonesian seas? You already hear from previous presenter that biologically Indonesia seas has been used as natural laboratory for various study on biodiversity of marine vertebrate, invertebrate, and plants, especially in the Western Pacific region as the center for coral triangle. And ecologically, Indonesian seas had been used as study area for various aspects of physical and chemical oceanography in association with marine biodiversity. However, it's very limited integrated study related to marine biodiversity and its association with oceanographic characteristics. Based on these two things or thinking, and then we conducted this study. So this one fact as an issue in regional and global era about the hotspot for marine biodiversity of Indonesian water. And also I mentioned before that Indonesia is a part or the biggest part of Center for Coral Triangle. And why we study around Sangieta Laut Island? Yesterday, you already heard from my colleague, Mr. Ramdan, why we study in this Sangieta Laut Island. At the time, he mostly explained about the importance of the area as the seamounts area. But also this Sangieta Laut Island located between deep sea basin of Sulawesi Sea and also Malacca Sea in the more southern, and also in conjunction with deep western of Pacific Ocean. And unique geographic character with a part of Ring of Fire. And also because of this is a part of Ring of Fire, of course, promising hydrothermal vent source. And another reason why we conduct study around Sangieta Laut Islands, because in 2010 ago, there was a cruise conducted under collaboration between Indonesia and United States. They called it this Index Shuttle Expedition 2010 to investigate deep sea under the island of Sangieta Laut. And they carry out many unique deep sea organisms, especially for marine invertebrate and also fish. This is several figure carried out from that cruise, and you can see also here there are several deep sea organisms, not only pelagic organisms. But no info about biodiversity in the marine pelagic ecosystem on that area. So they just mostly focus on deep sea with a very high technology, including using ROV, but no info in detail about the biodiversity in the marine pelagic ecosystem. Actually, Biodiversity in the marine pelagic ecosystem is very important. Why? Because mostly all the community in the island, they are fishermen, and mostly they are explore or they exploit the marine fisheries resource from the pelagic ecosystem due to their limited technology in fisheries catch. So the objective of this cruise related to the study is first, to carry out biodiversity of fish larvae in the marine pelagic ecosystem of the Sangihe Talaut Island, and second, to carry out biodiversity of invertebrate larvae in the marine pelagic ecosystem of the Sangihe Talaut Island. And the research cruise, of course, using vessel Barunajaya 8 LIPI, the cruise name Ewin 2018. The cruise team, we have a researcher 20%, a master student 1%, technician 4%, and security officer 1%. Maybe if you come to join this meeting in earlier morning, you have a chance to see the video. But if you are lucky, maybe the host will kindly repeat to showing you in the afternoon. Cruise day about 20 days, and it is on October 5th to 24, 2018. And this is the station of our study. It is covering the marine area around Sangihe Talaut, including Maluku Sea, Sulawesi Sea, and edge of the Western Pacific Ocean. A little bit. And this is the cruise track. We started from Bitung, North Sulawesi, or Manado, or close to Manado, and we port in Tahuna after 17 stations, and then we continue with Leg 2 from Tahuna to back to Bitung with more 18 stations. The sampling year and procedure on this biodiversity study. We use IKMT or ISEC kit amid water troll net, and the net operating was released from the deck of RV Barunajaya 8. The sampling method, we use both of step tow and oblique tow. And this is the situation from the sampling activity. We conduct the study both night and day, but in this photo, mostly night activity. So after we collect the sample, it was in the cold end of the IKMT, and we continue with sorting, and then to divide it, the organism based on their taxon. And everybody have their own responsibility for the taxon. So this is really huge study, but we really enjoy because as our title, this is the first study to be conducted in Indonesia for both fish and invertebrate larvae. And we continue after sorting with onboard morphological observation. Everybody also handling their own sample. In my case, I have a responsible for the young williformes fish larvae. This is Dr. Charles for deep sea larvae, deep sea fish larvae, helping the young researcher from Tahuna Polytechnic University, and also another younger or young researcher who handle the invertebrate larvae. So from the total marine fish larvae, we collected about 3,904 samples, and it consists of marine eel larvae, coral reef fish larvae, pelagic fish larvae, and deep sea fish larvae. You can see here, deep sea fish larvae is the most biggest catch. And from the marine fish larvae, we marine eel fish larvae, we can recognize at least 11 known family, and the dominant family is Cerephomiridae, about 13%, while 17% is unknown family. So as you remember, we just first start with morphological observation in this onboard observation. But after that, we continue in the lab. So in my case, I continue with molecular taxonomy and phylogeny of the Ordo Anguilliformes based on CO1 universal primer. And we, from this study, we can carry out at least three, we can categorize that sample based on the deep sea eel, mesopelagic or marine eel, and shallow coastal eel. We have Dr. Michael Miller here as the expert for this ordo, but he ever published a paper related to the stratification of this ordo based on their habitat in the ocean. But in some part in this study, we can clarify most of his found. And based on this study, we can say that represented by all taxon in Ordo Anguilliformes, these marine eelfish larvae dominancy by deep sea eel. So marine eelfish larvae from each station that we collected, showing very interesting data, where we can see here Sulawesi Sea the most dominant marine eelfish larvae, while Maluku Sea less, and also Western Pacific Ocean. This is only a few stations represented from this Western Pacific Ocean. So eastern edge of deep basin of Sulawesi Sea is more promising for higher species diversity compared to northern edge of Maluku Sea and western edge of Pacific Ocean. That is our tentative conclusion based on this data. And also we try to identify the species composition for each station, and also here you can see that Sulawesi Sea the most higher species diversity, and the highest species found in the sampling station 8 here in Sulawesi Sea, and also in station 10. But also we can see in 22A station there are relative bigger higher species. And why this is important? This 22A station is a Kawio Barat station or seamount station. And another important thing is that we also can carry out the species composition in Maluku Sea and also in Western Pacific Ocean. For marine eelfish larvae, based on their genus, we found that the dominant species is dominant genus is Seriforma, and followed by Gymnothorax. And while for the species dominancy, we found here Seriforma sector is the most dominant species. So another important things we found from this marine eelfish larvae that we collected 2 specimens of the Anguilla marmorata leptocephalus with total length about 28 millimeter, just in the station 22A in the top of the Kawio Barat seamount. So yeah, this is a commercial eel species, and we just thinking about the new evidence of the seamount hypothesis from Professor Katsumi Tsukamoto, that he proposed that seamount is one landmark for the spawning ground of the tropical eel. But if you're looking back the total length of the Anguilla marmorata, actually they are not just recently spawned, because usually when they're found it will be about at least a 3 or 5 millimeter total length based on species. So we suggest this Anguilla marmorata is coming up or coming from Pacific Ocean. They are not spawned in this area, and why they could be around our sampling area. They may follow the Indonesian through flow as a important current occurs in Indonesian water. So I think we need more study for this evidence. So we move to the coral reef fish larvae. From this coral reef fish larvae, we know the 18 family, and from the 18 family, dominant family is Botidae. I will move firstly for this one by one because we have several family next to my slides. And we also collected marine pelagic fish larvae, about 21 known family. And the dominant family is Karangidae, and but we also have an unknown family here, one person. For the deep sea fish larvae, with the most dominant larvae we collected during the cruise, about 32 known family detected during the study, and dominant family is Stomidae with a 19% from all the catch, and unknown family 5%. So we cannot, why we say unknown? Because we cannot detect the family through our morphological observation method. And we move to the invertebrate larvae. We collect 2,982 samples. It consists of mollusca larvae, cephalodont larvae, crustacean larvae, and gelatinous zooplankton. So from mollusca, we have found 16 known family. Dominant family is Cimatidae, 37%, and unknown about 4%. For cephalopod, we have a 2 known family. Dominant family is Enoploididae, 24%, and because some of them is already on the big size or not larvae anymore, so we can adjust their family at least this 7 known family. For the crustacean, we found at least 11 known family, but the most biggest family is unknown, not family. We cannot detect 56% from all the sample collected. It's very difficult to identify this larva compared to the other larvae. And for gelatinous zooplankton, we have an expert here, Professor Casey Dan, a part of the study under collaboration with him, and today he will have a presentation. I hope will be a very interesting also to hear. Our team who dealing with gelatinous zooplankton successfully identify 5 known family, but mostly of them, for 41% is unknown. So that is why we need expert, and so far there is no expert for gelatinous zooplankton in Indonesia. That is why we have a collaboration with Professor Casey Dan from Yale University of United States. So based on that study, we have several conclusions. First, a higher level of fish larva diversity were found in the marine pelagic ecosystem of the Sangita Laut Island, and higher level of invertebrate larva diversity also found on that area. I not need to mention about the number of family. And third, more detailed study using molecular genetic approach will be important in order to describe marine biodiversity status in the study area to the species level. I'm sorry, I cannot showing you all of the genetic data, but I really sure that all of my team now are going to finish their own data, and I hope in the near future, one by one, they will publish the result. We already conducted the genetic study mostly of the taxon. And the last one is, for our conclusion is, marine pelagic ecosystem around Sangita Laut Island were promising as a spawning ground, spawning and nursery ground of various tropical marine organisms. This will be very, very important information for us when we want to dealing with aquaculture or even for conservation of the marine ecosystem in this area. Thank you for your attention.
Thank you, Bu Yuli, for impressive presentation dealing with the result from the EWIN 2018. Okay, we're just in time. I think I have to move to the next session, the next invited speaker, I mean. Okay. Okay, maybe I'd like to accommodate one question maybe before Professor Casey Dan starting the presentation. Maybe the question for Mbak Yuli. Your research is very important. From Bu Yeti, from RCO LIPI, your research is very important. We collect data on almost all taxa of macrobiota, plant and animal, to get to know more about our biological resources. I propose to explore also. I propose to explore also microorganism for next biodiversity study. Almost no biodiversity study for marine microbes in our marine waters. Maybe just a suggestion for Bu Yeti, I think.
Yeah, thank you, moderator. I just a small comment that actually we have Dr. Charlie Esther de Freitas who dealing with microbiology in this cruise. But yeah, of course, because she have her own expert, I think it's better the result will be explained or will be presented by her in next occasion. Thank you.
Okay, thank you, Bu Yuli, for a very nice sharing. I'd like to move to the next invited speaker. I'd like to invite Professor Dr. Casey Dunn. Professor Dunn would like to deliver the topic entitled "A Midwater Perspective on Marine Biodiversity." Okay, the time is yours, Professor.
All right, thank you very much. Can everyone hear me okay?
Yes, we can hear you.
That's great. So I had a recorded presentation, but I found the other presentations very inspiring and made a few changes. So I will go ahead and present my presentation. If I can be allowed to share my screen, I can do that now. Whoops. Just one moment. Okay. Can you see my screen now?
Yes, yes.
Very well. Okay, wonderful. Okay, well, thank you very much for being invited to speak. It's been a real pleasure to hear all of the excellent talks. I really wish that I could be there in person, and I hope that this pandemic ends soon, not only for everyone's health, but so that I can return to Indonesia soon. So I don't need to tell this audience, but is it my main display or do you see my presenter display? Okay. But we live on a very watery world. So it's often quoted that 71% of the Earth's surface is covered by ocean. But in fact, I think that the more relevant number is that 99% of the livable volume on Earth is in the ocean midwater. And of course, the ocean midwater is a vast region of the planet, and it is the space below the surface and above the bottom. And there's many organisms that live there. Of course, not only phytoplankton and fish, but also this very abundant group of organisms known as the gelatinous zooplankton. So as Dr. Hagi said in her talk, understanding biodiversity and pelagic ecosystems is absolutely fundamental to understanding the biology of the oceans. So these are wonderful and fascinating organisms to work with, but also challenging because they're fragile and most can only be seen in the open ocean. And just to confirm, do you see a single large slide or the presenter display?
Sorry?
So
single
can
slide,
you see
single slide.
A single slide. Okay, excellent.
Okay, thank you.
Great.
We can see
Thank
your
you very
slides.
much. Great. Okay.
Very nice.
So Thank you. So I've had the great pleasure to work with gelatinous zooplankton with Steve Haddock at the Monterey Bay Aquarium Research Institute, among other places. And our primary tools for collecting this gelatinous zooplankton, which is so fragile, include blue water diving, where we use a tethered apparatus and we're scuba diving in the surface of very deep water, and we collect the organisms by hand in jars. And this allows us to bring them back intact, which is wonderful for all sorts of studies, from morphology to taxonomy. Aboard these ships, we also use remotely operated underwater vehicles, which allow us to sample down to 4,000 meters or more and bring the organisms up for investigation on the shore. So these are very powerful tools, and there's a wide variety of questions that we address in my lab. I'm an evolutionary biologist, among other things, and one of my primary questions has to do with phylogenetics, including how are all the major groups of animals related to each other. And one of the most surprising results we've obtained is actually that ctenophores, these organisms here, the comb jellies, may be our most distant animal relatives, a spot that was previously thought to be held by sponges because they lack nervous systems and things. So this means either the animals have evolved a nervous system multiple times, or that a nervous system has been lost in multiple groups of organisms, either of which is very exciting. And it just shows that the gelatinous zooplankton is important not only for understanding ocean biology, but biology as a whole. One thing that I think is very misunderstood about biology at this point is the role of taxonomy and biological surveys. So taxonomy and biological inventories are the foundation of all other biological sciences. You have to know what species you have to learn anything else at all. And because we're working with these poorly characterized ecosystems, we often are encountering species that have never been seen before, or that have been seen and not described. So I've described a variety of gelatinous zooplankton species with my colleagues, including Stefan Siebert here, a former postdoc in my lab. Here's one of these species of siphonophores that we have described here. So the ecology of gelatinous zooplankton is very poorly known. And if you think about it, this is incredibly strange. These are among the largest, most abundant animals in the space on our planet that is 99% of the space where things can live. So this is a huge hole in our understanding of biology. The holes in our understanding of biology aren't just about diversity, they're also about ecology. For example, gelatinous zooplankton are often described as trophic dead ends. So the idea is that they're eating many of the same prey, including crustaceans and things as fish, but there are very few descriptions of other things eating the gelatinous zooplankton. So it's thought that the jellyfish are out there. They're these evil aliens that are eating up all of this potential fish food and wasting it. And nothing could be further from the truth. There is incredible work being done right now by Anela Choi and Steve Haddock and Bruce Robeson and others, where they're using actual visual encounter data from these underwater vehicles to study which organisms are eating which organisms. And it turns out that there's a very dense jelly web where energy and nutrients are getting into this jelly web. They're moving around, but they're also going to other organisms. And I think that what has happened here is that much of what we know about food webs is based on gut contents. And if you cut open the gut of a fish, you can see the arthropods and the other fish that it's eaten, but you wipe away all the slime in order to see those things. Well, that slime is the gelatinous zooplankton. It's there. It plays an incredibly important role in open ocean ecology. We're asking a variety of questions in collaboration with Anela and Steve and others on a variety of questions, including how do predator traits determine food web structure? This is a major question. So we talk about food webs, but where do they come from? Why are they structured the way they are? And this is a collaborative project that includes Steve Haddock, Anela Choi, and myself, and some very talented young scientists who are working in our labs on this project as well. And I'll talk a bit today about work that is collaborative among all of us, but that Alex Damian Serrano, a graduate student in my lab, has really spearheaded. And I'd actually like to start with a cartoon that he's made with an animator that describes the question that he's working on. Can you hear the audio okay?
Yes, yes, we can hear your audio. But not for this. We hear no sound.
That's what I was worried of. Here. Well, you know what I'll do? I'll share the link in the, I'll share the video a little bit later with everyone. So he's looking at these organisms, or I'll provide the sound myself. I'll make up the words myself. So he's studying these siphonophores, which are one of the major groups of organisms that we work on. And what's happening with them is that they have these long tentacles with these tentacle batteries here, and they can capture other organisms. And these are highly specialized for the particular prey that they're capturing. And what he has done is he's built a phylogeny of the siphonophores using molecular sequence data to extend other phylogenetic work that's been done in our lab by Kat Monroe and others. And he's looked at the prey of the siphonophores on the phylogeny. And when people have theorized about the evolution of feeding, one of the common features of those theories is that in the course of evolution, you begin with generalist predators that in the course of evolution become specialized on different types of prey. And he's finding that is actually not the case at all. He's finding many cases of the reverse, where you have specialized predators that have evolved to become generalists, to eat multiple types of prey. So this is telling us not only who is eating who and what the food web structure is, but the evolutionary history of that ecology. And so his work so far has mostly used records from the literature of predation. And what he's doing is he's extending this work right now to look at molecular analyses of siphonophore gut contents, where he's he's collecting the siphonophores at sea, and then using DNA sequence data to identify what they've eaten. And he's putting together this beautiful data set here where finally we're getting an understanding of what the siphonophores are eating, building on what has been learned from examining morphologically their gut contents, which was a very productive approach, but some organisms were missed. For example, Physalia physalis, the Portuguese man of war, which unlike other siphonophores, floats at the surface of the ocean here. We can see the fish trapped in its tentacles. And then there's these other organisms here, the Apolemia, which include that species I had shown you that we described. It also includes this wonderful species here that was recently filmed by NOAA. And we can see that these Apolemia are eating a very broad diversity of organisms based on his data. So some of his take-homes include that siphonophores play a very important role in the midwater food web. that siphonophores, these gelatinous zooplankton, can feed on very different types of prey, and that the diets of most species are still unknown, and it's very important to make field observations on these. And importantly, new technologies are filling the gaps and challenging some of our hypotheses. And I want to really emphasize here that the molecular methods that we're using, they're not replacing the morphological methods we're using, they're complementing them. The morphological analyses continue to be incredibly important. So in addition to being a professor in the Department of Ecology and Evolutionary Biology at Yale, I'm also the curator of invertebrate zoology at the Peabody Museum of Natural History. And so in the Peabody, we maintain a collection of millions of specimens of invertebrates from around the world. So we're a large collection facility, but I really see our mission as being much more than going out and making collections and bringing them back. Our goal is to help understand the biodiversity of life on the planet. And when I think about the most exciting places in the world to study gelatinous zooplankton, really the first place that comes to mind is Indonesia. So as we heard early, as Dr. Perstawati noted, Indonesia has more open ocean and deep sea within its national boundaries than any other country in the world. And one of the really incredible things in Indonesia is that much of that deep water is very close to the shore. So usually to study gelatinous zooplankton, we need to get into a boat and go hundreds of miles out to sea. In Indonesia, there's multiple places where it's possible to have breakfast on the shore, go in a speedboat, collect some gelatinous zooplankton, and be back and have lunch, and then study it in the afternoon. That's incredible. There's very few places where this is possible. And I'm so excited to build collaborations with researchers in Indonesia to support their studies of gelatinous zooplankton. And I was really fortunate last year, so early last year, to come and visit. So Dr. Hagi was a wonderful host, invited me to come, and I got to meet many people, including friends who are here at the conference today. And then almost exactly a year later, as this collaboration has gotten underway, Rito and Hanang came and visited the Yale Peabody Museum with these gelatinous zooplankton specimens from the Iwan expedition that Dr. Hagi mentioned. And that was incredibly exciting. They brought their specimens. We worked with them to identify what they had, and then they returned with these specimens. And I just have to say, I'm just really impressed by the enthusiasm and strength of taxonomy and biological inventory in Indonesia. And I just really hope that I have the opportunity to help build further capacity and expertise in this area when it comes to gelatinous zooplankton. So I want to thank my colleagues for coming, and we hope that Dr. Hagi and others could come, but unfortunately with COVID, that's on pause, but I hope that it still happens. So we want to start with collaborations on taxonomy and biological inventory of gelatinous zooplankton. So this, again, of course, is the bedrock. We need to know what's there in order to be able to ask other questions. But I think that this will very naturally seed work in a variety of other areas that are very exciting, including natural history, oceanography, population biology, ecology, and evolution. And it's been fascinating to attend the conference and see the work that other people are doing on things related to this. So thank you very much.
Thank you, Professor Dan. Okay, we are just in time, but anyone has a question, you may write it directly in the Zoom chat, and Professor Dan will answer your question after his session. Okay, I have to move. Now we are attending the oral presentation from the participant. For the first occasion, I'd like to invite Mr. Hanung, but since Mr. Hanung, it seems that he's not present at the time. Mr. Hanung cannot present at the moment. One of the authors of his study will present it. I'd like to invite Mr. Okto Rido Sianturi from RCO LIPI. Mr. Rido would like to deliver the topic entitled the occurrence of harmful jellyfish outbreaks and human stung reported at the recreational beaches in special region of Yogyakarta, Indonesia. Mr. Rido, the time is yours.
Yes, I'm here, Padi. Can you hear me, Padi?
Yes, we can hear clearly.
Okay, so but I just get notification that the host disabled participant screen sharing. Would you please allow me to share my presentation, please? Thank you. All right. Can you see my slide, Padi?
Yes, yeah.
Okay. Yeah. Okay. All right. Good. Okay. Good morning, everyone. Good morning. Yeah. Good morning. So, first of all, I would like to say thank you to the committee to allow me to join this wonderful conference. Awesome speakers and great ideas that I get from this conference. And second, I want to tell you that Mr. Hanung cannot be able to join us and give the presentation because he have to attend another event. So I will replace him for this presentation. So today we would like to share to you about it's actually a short talk about the occurrence of harmful jellyfish in a region of in recreational beach in a special region of Yogyakarta and the stung cases that reported in this area. So basically, I will talk a little bit about jellyfish in general, and then I will share about jellyfish bloom and stung cases in Indonesia, and then I will talk about stung cases in Yogyakarta. A little bit background about of the jellyfish bloom. As we know, is actually we can say that it's exceptional and sudden increase of jellyfish, both in seasonal and seasonal of jellyfish bloom. Sometimes it's actually just their seasonal life cycle. So what we saw sometimes is actually just their normal life cycle when they get bloom, when the station supported for the bloom, and then sometimes we saw the bloom be caused by the anthropogenic activities such as superfishing and then global warming, eutrophication, like Uye Sensei described in this jellyfish spiral. So basically, we are moving from the fish-dominated ecosystem to the jellyfish-dominated ecosystem because of our effort, our human forcing, where we have overfishing, global warming, eutrophication, marine construction, and others. And most of the time, the jellyfish bloom will affect us human in many ways, like fisheries, tourism, power plant, and infrastructure. So, Bros et al. They try to describe the area which there will be a jellyfish bloom, where there are area that in red area that you can see there will be a high certainty there will be bloom in the future, and then a low certainty, and then the stable where the bloom just happen regularly, and then there are areas also that are decrease in jellyfish bloom. But as you can see, Indonesia in this map is categorized as no data. So on worldwide, jellyfish bloom has caused nuclear power plants shutdown in Scotland, Japan, Israel, Florida, and also in Indonesia in 2014 2016 and 2018. And then there's also Aris salmon industry, and also fish farm in Tunisia, and the jellyfish Nomura in Japan. So as Brock shown us in the previous slide, so there's no actually data about jellyfish bloom in Indonesia. So we try to build a database. We try to collect data about the jellyfish bloom in Indonesia. So far there are only few reported cases in Indonesia, and as you can see, I tried to show the places, but what I shown this is just jellyfish bloom. So there are more places that there are jellyfish, but what I would like to show you is a jellyfish bloom in Indonesia. So as you can see, there are blooms, there are stung cases, and there are places that has caused casualty. There are people killed because of this jellyfish bloom. I'll show you in the next slide, this one. So you can see in the left side, in the red circle, you can see there are 3 boys that killed because of the jellyfish. There are Physalia utriculus and Gisaura quincacera that was reported the species that caused this boy died. So despite the occurrence of harmful jellyfish in Indonesia, and some cases happen almost every year, especially in Jogja, but we not yet consider this as ecological disaster. So we have also case in Indonesia. There are jellyfish bloom blocks the power plant water intake in Surabaya in 2016 and 2020. So there was a power blackout in 2016 in this place. And then, as you can see, jellyfish stung beachgoers that killed three boys. And then recently, we just found that jellyfish bloom disturbed fisheries in Jakarta Bay. So what about the jellyfish stung cases in Yogyakarta? So I would like to show you that Yogyakarta is in the southern part of Java. So this place is actually facing directly to the Indian Ocean. So in this case, we get the information from Gelagah Beach, Parangtritis Beach, and Baron Beach. It's a closed beach in the Jogja region. And it was reported that the species that caused these stung cases is Physalia physalis, as you can see in this picture. And it's reported occurred in June to August every year. So there are reported stung cases every year, but stung cases is reported from the stranded jellyfish that they found on the beach. So we get the data from 19th of June to 12th of July in this year. So we get the species. It was reported that Physalia physalis, and then it was thought that it might be related to the strong wind and ocean current during southeast monsoon. So, and in this period, in this almost a month, so there are total 913 visitors that were stung by jellyfish, and the highest number stung actually on the 4th of July. So you can see in 5 July and 12 July, it's actually on Sunday and Saturday and Sunday. So it's on the weekend, so there are a lot of people goes to the recreational beach, and they play on the beach, so they got stung by the Physalia physalis. But if we look at the distribution of age, the visitors that get stung, we can find that there are most of them are actually children and teenagers, 10 years and 11 to 20 years. So it seems like the color of the Physalia physalis that stranded on the beach is the reason these children got stung, because the color is looks so colorful, it looks beautiful, contrast with the white brown and white brownish color of the sand. So it attracts the children to play with it, but little bit they know this nematocyst on this jellyfish still active, even though they have been stranded for a long time. So this is the main reason why they got stung. So then we can say that we try to summarize the factor that cause of these stung cases in Yogyakarta. As we know, in June to October, there is a southeast monsoon wind that came from cooler part in Australia that blows to past Indonesia until to Central Asia. And this is the main reason why this, we think that this is the main reason why this Physalis get stranded on the beach, because if we see the morphology of the Physalis, actually they are on the surface of the sea, so the wind will blow them to the coast and get stranded on the Yogyakarta. To make things worse, this is coincide with public holiday. So actually in June and July, it's a public school holiday. And about 2 years ago, 2018 and 2017 is actually coincide with Lebaran, Ramadan, 8 holidays. So there are a lot of people that going to the beach. And then this year, in this case, is in 2020, it's because of COVID-19. So the beach were closed for a long time, and then due to COVID-19 restriction and lockdown, and then after the beach reopened, a lot of people went to the beach, and then coincide with the stranded of jellyfish, and then that's why a lot of people get stung in this area. And most importantly, we think that the importance of first aid will be a great factor that reduce the case of the jellyfish. So what the rescue team do in to overcome this jellyfish bloom? So they did some first aid by cleaning up the sand from the skin, and then rinse the skin with vinegar, and then they also use a topical painkiller, and sometimes they are using traditional medicine from Jinking. It's small crabs that's abundant in this area, so they will crush this crab, and then we'll use this then we'll put this Jinking on the skin that gets stung by the jellyfish. This is local wisdom that they have been doing for so long. And then they actually the rescue team has been given training about the first aid by Dr. Tri, the picture A. This director of toxicology of Indonesia. So she gives training about the importance of first aid in jellyfish stung, the importance of using a vinegar for the first aid for this jellyfish stung. And also they also put do's and don't poster in Bahasa on the beach, so that people can understand that what should they do what they shouldn't do when they see the jellyfish stranded on the beach. I think that's all that I can say. Thank you very much. Thank you, Padi.
Okay, thank you, Bang Rido. Okay, it's very interesting presentation. I'm gonna check whether we have a question in the Zoom chat. Not yet. May I have a silly question because I'm not familiar with jellyfish. Instead of become a threatening biota, since we found this biota will sting human in many cases, I'm just curious about the economical value of the jellyfish. Could you explain briefly the possibility to taking benefit from jellyfish blooming economically?
Yeah, so there are some edible jellyfish that actually Indonesia has been harvested from 1970. So we've been harvested for almost 50 years. But in this case, Physalis is not one of them. So some of the edible jellyfish is what we say ubur-ubur merah, red jellyfish, that we can find a lot of in Saleh Bay in Mataram, and then we can find another species like Crambionera, and then but in this place in Yogyakarta, we cannot find edible jellyfish. We only found the Physalia physalis. So far we cannot find the economic value from the Physalia physalis, but from other species, yes, we can find it.
Okay, I see. It's depend on the species.
Species, yeah.
Okay, thank you, Bang Rido, for a very impressive presentation. Maybe I'd like to move to the next oral presenters. I'd like to invite Dr. Suyati.
Yes.
Okay. Dr. Suyati would like to deliver the topic entitled Mangrove in Urban Area of Small Islands, Vegetation Health, Potential and Management Challenges. Dr. Suyati, time is yours.
Okay, thank you, Mr. Adi. Can I share my presentation, please?
Yes, of course.
Can you hear me and see my presentation? Okay. A second. Okay, hello everyone. Today I'm going to present our study. The title is Mangrove in Urban Area of Small Islands, Vegetation Health, Potential and Management Challenges. We can see from this map there, just a second, I'm going to start the video. Okay, so you can see me. So we can see from the map that Indonesia, the red box, is the large habitat of mangrove. About 23% of global mangrove are in Indonesia. And Indonesia also good habitat for many species, around 67% of mangrove in the world also in Indonesia. And most of them, or many of them, are endangered species and endemic species. As we know that Indonesia is an archipelago country and 77% of the islands are small islands. And we also have the longest coastline in the world. This means that Indonesia is the good habitat for coastal ecosystem, for example, mangrove. And we can see here the red box. This is the Maluku province. Maluku province is the fourth largest mangrove area in Indonesia. And this is the biggest archipelago province in Indonesia. That we can see that 92% of the territory is the sea and 98% of the islands are the small islands. And many of mangrove habitat located in the urban area. For example, here in the photo is in Ambon and also in Tual. Okay, this is the idea of our research. The mangrove in urban area of small islands. They have a unique characteristics. For example, small islands are vulnerable and low capacity, and mangrove in small islands also have a unique characteristics. For example, a spatial metric or spatial characteristic. For example, the pH is lower than in the large islands. Vegetation also unique, geomorphological and the environment as well. But mangrove in urban area of a small island also face the high pressures, for example, the development, due to the development, especially in the coastal developments and pollution, for example, oil spill and plastic pollution and human population. As we know that 30% of human population in Indonesia, they living in the coastal area. And in here, we also identify some research gaps or research questions. What is the conditions or vegetation health of mangrove in urban area of small islands? And what is the potential of mangrove? Because this mangrove is, just I mentioned before, it has unique and different from mangrove in large islands. And what is the management challenge of mangrove in urban area of small islands? This kind of information is required to support management, especially for urban greening program, not only terrestrial, but also we can use the mangrove for greening the urban area. And secondly is coastal or mangrove management, urban planning and management, and also the conservation actions. Our study sites are located in Ambon or Ambon Islands, represent of main urban islands, and 12 satellite urban islands. Both of these islands are located in Bandasi. As we mentioned before, Pak Augie, before that, Bandasi is the important Indonesian drug flow. This is connected between Hindia Oceans and Pacific Ocean. And this is not only important for biodiversity, but also important for regional and global climate regulations. In our study, we applied some methods. For example, we use a GIS remote sensing, vegetation survey, we use a transects, sampling of plastic debris, we use a quadrat, and interview survey. These all of the methods to address some important points. For example, mangrove cover change, vegetation health, potential of mangrove, threats of mangrove or problem faced by mangrove ecosystem, perception of local people or local communities to mangrove, and management challenges. This is the result. We start from the mangrove area and their characteristics. Our study found that mangrove in satellite urban areas such as Tual, this is not much change. The area is still similar during the recent decades. However, mangrove in main urban area, a small island like Ambon, is decreased by 9% or 0.5% per year in the recent decades. But we also find some mangrove in Ambon, some site increase because due to the rehabilitation or mangrove planting, but the rate of the increase is much lower than the mangrove loss. And the study also found 15 species of mangrove in Ambon and 9 species in Tual. And the most dominant species in Ambon is Rhizopora apiculata and Solerata alba, and in Tual is Bricorea gymnoriza and Solerata alba. But this is not represent whole island. This is only the urban area of those islands. Okay, let me start with next to the mangrove health or mangrove conditions. Mangrove in Ambon also degraded or in the poor conditions. We can see here the density of mangrove is only 763 stems per hectare, and the mean canopy is was only 30%. In contrast, mangrove in Tual, or we call a satellite urban area, the condition is still good. With the mean tree density is higher than 2,000, and the canopy coverage is more than 78%. And we can see here in the table that all sites of mangrove in main urban or Ambon and satellite urban area is this one, satellite urban of Tual, is consistently higher than in Ambon. Let me go to the ecosystem services in terms of carbon storage. We can see here in Tual, they provide more carbon stock than in Ambon. This is in Tual is about 191 megagram carbon per hectare, and in Ambon is 120 megagram carbon per hectare. This is lower if we compare with the carbon stock of mangrove in large islands, but this is still higher, much higher than temperate mangrove, for example, in New Zealand, in Florida, and any country in the temperate regions. Carbon storage in Tual was equivalent with to the 700 megagram of carbon dioxide equivalents, and Ambon is 441 megagram carbon dioxide. This means that a significant carbon dioxide can be stored in the mangrove. This only for above ground carbon, not include the soil carbon or below ground carbon stock. And the economic value we can see here, although mangrove in urban area of small island, they from a small bed, but they provide a significant economic value only from carbon stock. And how about the threat or the problem faced by mangrove ecosystem? We can see here that mangrove loss caused the emission of carbon dioxide equivalent around 110 megagram, or this is equivalent to $7,700 per year. Mangrove health is influenced or affect the carbon storage, of course, because the carbon storage is based on the vegetation, and vegetation health is important for the services of ecosystem. And the next threat of carbon is plastic pollution. We found that in Ambon was more polluted than in Tual. It's only 10%. In Ambon is 23%. And the distribution of plastic also varied. We can see here that landward zone of mangrove is more polluted than seaward zone, and the middle zone is a little bit lower, just around 5%. Different from Ambon, cutted trees is the most prominent trait in Tual. This is around 60%, and Ambon is also 37%. And how about the result of interview survey? We interview around more than 100 respondents, and 90% of our respondents is at the productive age, and they have a good education. Most of them have a senior high school, around 83%. Unfortunately, many of them unemployment. This means that they don't have a permanent job, and their income is relatively low, only 1 to 2 million per month. But 98% of the respondent has a good knowledge about mangrove. We can see here when we ask about the mangrove conservations, most of the respondents say that mangrove conservation is very important, and only 1% they said that not important. And how about the mangrove? And when we ask the second question about the mangrove condition, they said that most of the respondents say that mangrove is getting better. But this opinion of respondents is different from our field data, as I mentioned before, that a mangrove area in Ambon decrease, and the vegetation health also getting bad or decrease. And many of the respondents around here, 43%, got the information about mangrove from friends, and secondly from NGO and televisions. And we can say that the knowledge or the perception of respondent in urban area about mangrove is good, but their participation in protection or in the mangrove conservation is relatively low. They want to participate in the mangrove conservation if there is the incentive from the government or from other organizations. And we can conclude from this study that urban area of small islands are a habitat for many species of mangrove. However, the mangrove area decreased and vegetation was decreased, especially in main urban small island like in Ambon. Mangrove in urban area of small island have a good potential to offset carbon emission through carbon sequestration. This is in line with the study before conducted by Mudiarso and also Donato, that mangrove is important ecosystem to offset the carbon emission. But their study is focusing on the large island, for example, Papua, Sulawesi, and Kalimantan. And unfortunately, the mangrove ecosystem, they face the tremendous threats, for example, mangrove conversions or deforestation, especially in Ambon, and plastic pollution, cutted trees, and sedimentation. Local society in urban area have a good knowledge, as said before, about this, but their participation is low. And we suggest that avoiding of mangrove conversion, pollution, and strengthen the conservation action of mangrove in urban area of small islands should be the high priority strategy in order to mitigate climate change, coastal protection, and biodiversity conservation. And this study is supported by Research Center for Deep Sea and also supported by Research Center for Oceanography. And we would like to thank for all friends and colleagues which help us during the field work and lab work. This is the reference, and thank you for listening.
Thank you, Mas Suyadi, for a very nice presentation. I have to check the Zoom chat question. Not yet question. May I have a question, Mas Suyadi?
Yes, of course.
as we know that the ecotourism has been increasing massively in Indonesia recently, especially in the coastal region, including the ecosystem area, of course. And I think we should guide the local citizens and stakeholders on how to manage the mangrove area so that giving more benefit economically from ecotourism perspective with a minor negative impact. My question is, do we have a guideline practically, maybe from LIPI, or how that can be easily adopted by local stakeholders to open new mangrove ecotourism?
Okay, actually, the mangrove ecotourism in eastern part of Indonesia, in the western part of Indonesia, is the common practice. Not only in the high scale, for example, in the government, but also in the local. In the local community, they have mangrove ecotourism. But in Indonesia Timur, or in the eastern part of Indonesia, I didn't find any significant of mangrove ecotourism there. They use mangrove, only use the conventional things like harvesting the stems for firewood and so on. About the guidelines, actually there is a lot of guidelines about mangrove ecotourism, but we don't have any standard. I mean the national standard of mangrove ecotourism. So they have their own guideline, because maybe different environments, they need the different guidelines. For example, because develop the mangrove ecotourism not only about the ecosystem, but also about the local wisdom, about the culture. So, maybe we need a different guideline for different area. I hope this answers your questions.
Okay, thank you, Mas Yadi. We have another question from Professor Sam. There are mangrove replantation effort in Ambon. How about the progress?
Okay, thank you very much, Prof. Sam. Yes, in the recent decade, there are many mangrove replantation, for example, in the front of Poka and many else in Ambon. But if we see from the satellite images, the area, this increase, as I mentioned before, but it's not significant. The rate of mangrove lot is much higher than the rate of the increase of mangrove. And most of them not increase the area, but just the increase the density. For example, in Poka, the density increase, but the area is not really increased. Yes, we appreciate this program, and we hope that the replantation is still continue. Not only the replantation, but the management, because mangrove cannot grow is only we plant it, but we not manage it. So we have to look after so the mangrove can be survive and growth of the forest. Thank you.
Okay, thank you again, Mas Suyadi, for very interesting sharing. Okay, now we have to move to the last oral presenter session. For the last presenter, I'd like to invite Mr. Pandu, Mr. Marinda Pandu Rizky. Mr. Pandu would like to deliver the topic entitled "The Conditions of Seagrass Beds in Pusomaen District, Southeast Minahasa, North Sulawesi, following the implementation of a community-based coastal resource management program." Are you ready, Mr. Pandu? Okay.
Yes, thank you. Can I share the screen, please?
Okay. Okay. The time is yours, Mas Pandu.
Yes. Can you see my presentation?
Yes.
And the voice is quite okay?
Yes.
Okay. Thank you very much. First of all, I would like to thank the committee of Icoes for allowing me to present the results of my study in 2018. And thank you for all the participants. It was a very nice conference, and I learned many things. Okay, my presentation entitled "The Condition of Cigarette Beds in North Sulawesi," in one of the districts in the North Sulawesi, following the implementation of community-based coastal resource management program. Okay. The outline of my presentation will be introduction, and then we will talk about the study area, the methodology, and then continue with result and discussion and conclusion. Okay, as has been explained by Dr. Aan yesterday, cigarette beds is one of the typical tropical coastal ecosystem with many bioecological, socio-economic, and environmental functions. The cigarette bed is an important nursery ground for fishes and other biota, and it's also one of the carbon sink ecosystem, so it can reduce the carbon emission. And unfortunately, the cigarette beds around the world are mostly declined due to changing environment and human development. And also in Indonesia, which approximately has 150,000 hectares of cigarette beds, which most of them are in the central and eastern Indonesia. And of course, the conservation effort has been done and has been conducted by many of the institutions in Indonesia, whether it was NGOs or government. But unfortunately, again, they were mostly targeted in the more recognizable ecosystems such as mangroves and coral reefs. Seagrass is kind of forgotten, actually. And then between the 1997 and 2003, there was a conservation program called Proyek Pesisir, or community-based coastal management program that has been conducted by Bappenas, USAID, and University of Rhode Island. So it was around 15 years ago, and it was done in now part of the Pusomain district, which the research is conducted. And it was focusing on the implementation of the best practice that has been decentralized. So it was used, or it was conducted by the community in the village, how they can manage their own coastal areas, which also involve many stakeholders, and their goals are establishing marine sanctuary and also establishing the institutionalization, or the result will be the local legislation or village regulation. So one of the expected outcome were the increased knowledge and responsibility of the involved stakeholder in managing coastal resources sustainably, and also of course the environmental status of the coastal ecosystem is hoped to be increased. So the legacy of this project were expected to be observed now through the implementation of the village regulation regarding the coastal ecosystem. So that's why the aim of the study is to obtain the information on the seagrass condition as one of the coastal ecosystem in Pusomain district, and discovering the lasting impact or the legacy of the program CBCRMP or Proyek Pesisir through the observation of people's behavior and environmental condition of seagrass beds in Pusomain district. This is the study area. As you can see, this is the coastline of Tumbak and Bentenan village, which is the village of Proyek Pesisir, and all the stations are in the coastal Tumbak and Bentenan, except for one station, Station 4. It was in the Bentenan Island, the small island in front of the Tumbak village. Most of the substrate were sand and rubble, with a little bit of mud in some of the areas. The methods for the seagrass monitoring, we use a modified quadrat transect, which has 3 100-meter transect per station perpendicular to coast, and then sampling point per 10 meters using 50 by 50 centimeters. Centimeters quadrat. And then we observe the coverage visually and identify the species and the species coverage. And then we analyze the data using mean coverage. And then we analyze the dominance of each species. And then we also categorize the status based on the Ministry of Environment standard. And other relevant data were obtained also during the survey, including the interviews with villagers and on environmental condition and on their behavior to the seagrass environment. The result, we found 7 species of seagrass from 2 families, and it is common also found in North Sulawesi. These species, there's no unknown species. Every species has been discovered before, and then the most species found in Station 9, which has 6 species, and the least were in Station 3, which only has 2 species. And for the status, according to the Ministry of Environment, only 1 station has a healthy condition or healthy status based on the coverage, which is on Station 2, and the other are in moderate status, and only 1 in the poor status, which is Station 3. And the interesting thing about this is these stations are in one the lowest coverage is right next to the highest coverage, which means in one coastal line there is a seagrass bed which has good health, and next to it it has a poor health. The cause of this condition is because in the area where it has poor health, in Station 3, it has fish landing site. So there are big ship anchored there to sell their fish, and it has a oil pollution there. So it might also affect the coverage of the seagrass. On the species dominance, we see that 4 species has quite high dominance, which is Enhalus acaroides, Thalassia hemplici, Syringodium, Isotiphilium, and Simodosia rotundata. Other 3 species are quite low, and this is also common in this area because usually we have two of the highest species dominance between Enhalus and Thalassia, but here we also have a Syringodium, which also dominant because they cover some of the areas that the Enhalus and Thalassia cannot thrive. And this is the legacy of the Proyek Pesisir or CBC-RMP. We found that village regulation on coastal ecosystem were recognized, but there's no enforcement observed. It's a little bit the same with the Pak Dr. Suyadi's presentation, that they recognize that there is a regulation about the coastal management, that you cannot dispose domestic waste to the sea, and then you should not cut the mangrove trees and so on. But it was recognized, but it's not enforced because there was no enforcement. There was no enforcement authority there. And then the domestic waste disposal was still observed, whether in mangrove and the seagrass beds. The oil spillage in seagrass beds from Pajekos, or the big ship for the fishery ship, were observed, and this may cause also the lower dominance, lower coverage in one of the station. And there were no direct recollection of the result from the Proyek Pesisir program, apart from the recognition of its existence. So when I asked about the what do you know about the Proyek Pesisir, they recognize that it was conducted years ago, but there were no other recognition. There were no desire to conserve the coastal ecosystem and so on. So the legacies were minimum or non-existent in today's Tumbak village. So this, as you can see, this is in the back of the village. This is grass bed. You can see there's still plastics there. There's still domestic waste there, and in the seagrass bed that's a little bit far away from the village homes, there were no plastic or no waste disposal like this. And the conclusion, the relatively favorable seagrass condition implied that there's still room for improvement, particularly in conserving seagrass beds in North Sulawesi. But there were lack of availability of spatial temporal data of seagrass condition, and that's why it was hard to know that you have a good growth, or you have a good a better condition after the program procedure or project procedure, because the baseline data was lacking. So we hope that in the future we can monitor the seagrass beds more continually. And the last one, the collaboration and cooperation between stakeholders, including coastal community, local and regional government, academia, and private sector, is one of the utmost priority in order to achieve a mutual understanding in the importance of sustainable coastal environment, particularly in seagrass beds. Thank you, and back to the moderator.
Okay, thank you, Mas Pandu, for very interesting presentation. Okay, I will check, is there any question from the participant in the Zoom chat? Not yet, I think. Okay. Okay, thank you for all invited speakers and oral participants. We have finished all the presentation from the morning session of the second day of the International Conference on the Ocean Science, Ocean and Earth Sciences, Indonesia Science Expo 2020. Maybe I'd like to give the time and place to Mbak Sofia.
Okay, thank you, Adi. Okay, thank you, Adi, for guiding us through all the morning session. Okay, ladies and gentlemen, we're ahead of schedule about 10 minutes, but we will stay on the schedule. We will have a lunch break around an hour. So we will be right back at precisely at 1 o'clock to continue the afternoon session. Okay, ladies and gentlemen, don't forget to stay on the line, stay on your Zoom line. We will be right back, and we will see you in 1 hour. Okay, thank you, ladies and gentlemen.
Thank you all.
Thank you.
Berawal dari cita dan asa untuk membangun negeri, memberikan segenap rasa dan tenaga untuk menghasilkan pemikiran terbaik bagi negeri tercinta, kami ada untuk Indonesia. Mewujudkan diri sebagai Lembaga Ilmu Pengetahuan Indonesia yang andal, profesional, inovatif, dan berintegritas dalam pelayanan untuk mewujudkan visi Presiden dan Wakil Presiden. Indonesia Maju yang berdaulat, mandiri, dan berkepribadian, berlandaskan gotong royong. LIPI memiliki berbagai pusat penelitian tersebar di seluruh Indonesia yang berada di bawah Kepala LIPI, Deputi Ilmu Pengetahuan Hayati, Deputi Ilmu Pengetahuan Kebumian, Deputi Ilmu Pengetahuan Teknik, Deputi Ilmu Pengetahuan Sosial dan Kemanusiaan, Deputi Jasa Ilmiah dan SDM Pendukung Iptek di bawah Sekretariat Utama LIPI. LIPI berperan dalam proses penyusunan dan kelahiran berbagai regulasi dan kebijakan pemerintah terkait aspek ekonomi, politik, sosial, dan budaya. Selain itu, LIPI juga mengelola data dan timbangan ilmiah dalam rangka konservasi keanekaragaman hayati. Menerbitkan tanda pengenal unik publikasi ilmiah terbitan berkala, membuat repositori ilmiah skala nasional, dan memiliki kewenangan sebagai pembina Jabatan Fungsional Peneliti Nasional, serta melaksanakan proses pengukuhan profesor riset di Indonesia. Kini, LIPI juga mendukung percepatan penanganan COVID-19, baik melalui riset berupa kajian maupun produk, maupun pengembangan SDM melalui program Indonesia Memanggil. Untuk mendukung riset, LIPI memiliki 5 kebun raya sebagai pusat konservasi flora ex situ. 2 museum yang memiliki koleksi flora, fauna, dan kesejarahan alam Indonesia, Indonesia Culture Collection yang menyimpan koleksi mikroorganisme terbesar di Indonesia, infrastruktur riset strategis, dan beberapa pusat berskala internasional, yaitu Asia Pacific Center for Eco-Hydrology atau APCE. LIPI telah menghasilkan hasil riset yang berkualitas dan bermanfaat bagi masyarakat, meliputi keanekaragaman hayati, lingkungan dan maritim, ketahanan pangan, pertanian, dan peternakan, kesehatan dan farmasi, material maju, energi, teknologi, informasi dan komunikasi, pertahanan dan keamanan, dinamika sosial, kemanusiaan dan kebudayaan, kebencanaan, inovasi teknologi dan pendaya gunaan iptek, pelayanan, pengujian dan pembinaan ilmiah, serta peningkatan kualitas SDM iptek. Kerjasama keilmuan LIPI dalam negeri meliputi kerjasama penelitian, lisensi teknologi, pengembangan, aplikasi dan komersialisasi hasil penelitian, kerjasama non-penelitian, dan konsultasi kepakaran LIPI. Sedangkan kerjasama luar negeri, LIPI melakukan joint research, workshop, pertukaran peneliti dan beasiswa mancanegara, selain aktif menjadi anggota dan focal point di berbagai organisasi internasional. Melalui kolaborasi bersinergi, menghasilkan riset yang berkualitas, LIPI terus memberikan manfaat nyata, kontribusi berarti, dan turut mewujudkan cita dan asa membangun negeri kita tercinta, Indonesia.
Assalamualaikum warahmatullahi wabarakatuh. Distinguished keynote speaker, invited speakers, ladies and gentlemen. It is my pleasure to be here to welcome you all to this Indonesian Science Expo International Conference on Ocean and Earth Sciences 2020. Indonesian Institute of Science, through its 3 centers under the Deputy of Earth Sciences, namely Oceanography, Limnology and Geotechnology, proudly held the Indonesian Conference on the Ocean and Earth Sciences from today, November 18, for the next 3 days of November 20th. The conference theme is about ocean and earth sciences for better human life with nature. This conference has been initiated in order to address several global issues related to human impact on the natural ecosystem. Those global issues are consisting but not limited to the impact of climate change, marine environmental pollution, natural disaster, and human over-exploitation to natural environment. It is become a great challenge for human beings who live in Earth but continue to take the advantages of the ocean and Earth to be able to combat and to solve the problem. Another mission of this conference is to understand the Earth in its past, present, and future, the energy and resources required to support an increasing global population, the needs and challenge of sustainability in excessive changes. Distinguished ladies and gentlemen, I do hope that this conference will serve as a platform to provide solutions to the problem about impact of climate change, marine environmental pollution, natural disaster, and human over-exploitation to natural environment. I wish you all a fruitful and productive discussion, and the result of the conference will benefit not only for human prosperity and natural environment, but moreover for the sustainability of our ocean and earth. Last but not least, I would like to sincerely thanks to all of you for being able to join this conference during the COVID-19 pandemic. Please stay healthy and be happy during this long pandemic. Again, thank you very much for your kind attention and enjoy the conference. Wassalamualaikum warahmatullahi wabarakatuh. Assalamualaikum warahmatullahi wabarakatuh. Distinguished keynote speaker, invited speakers, ladies and gentlemen. It is my pleasure to be here to welcome you all to this Indonesian Science Expo International Conference on Ocean and Earth Sciences 2020. Indonesian Institute of Science, through its three centers under the Deputy of Earth Sciences, namely Oceanography, Limnology, and Geotechnology, proudly held the Indonesian Conference on the Ocean and Earth Sciences from today, November 18, for the next 3 days of November 20th. The conference theme is about ocean and earth sciences for better human life with nature. This conference has been initiated in order to address several global issues related to human impact on the natural ecosystem. Those global issues are consisting but not limited to the impact of climate change,
berapa tawarnya? Ada bawalnya doang.
Saya pertama kali melihat di sini itu tahun 1980-1981. Itu kalau kita nyebur di sini dari mulai tepi sampai di tubir di sini, ini adalah tubir di sini, itu penuh dengan karang. Paling tidak persentase tutupan waktu itu masih 75-90%. Dan setiap kalau kita jebur ke laut pasti masih banyak sekali ikan, dan kalau kita bandingkan saat ini sudah kira-kira ya sudah jarang sekali kita dapatkan ikan-ikan yang cukup besar. Keadaan terumbu karang saat ini di sini itu sebetulnya cukup memprihatinkan juga, karena ternyata sekarang kalau kita lihat persentase covernya atau persentase tutupan karang yang hidup itu sekitar hanya 30-40% yang ada di sekitar sini. Terumbu karang juga saat ini dimanfaatkan oleh objek wisata, karena banyak sekali sekarang berkembang wisata bahari atau wisata maritim gitu, yang terutama melihat objek-objek di dalam karang itu sendiri, karena karang itu sangat eksotik sehingga mereka menjual sebagai objek wisata. Sebetulnya manfaat terumbu karang bagi kita atau terutama bagi nelayan, terumbu karang itu adalah rumah dari ikan-ikan tersebut, sehingga secara langsung terumbu karang itu untuk nelayan memang tidak ada. Tapi kalau kita lihat bahwa terumbu karang itu sebagai rumah ikan, tempat memijah atau tempat beranak, dan juga tempat mencari makan bagi ikan-ikan, sehingga kalau terumbu karangnya itu bagus, ikannya tentu banyak, dan ikan itu bisa dimanfaatkan oleh nelayan atau oleh kita semuanya gitu. Di samping itu, kalau secara fisik terumbu karang ini seperti pulau-pulau ini adalah terbentuk dari karang itu sendiri. Kalau tidak ada karang itu, saya kira pulau-pulau di sekitar sini juga tidak ada, karena pulau sekitar sini adalah sebetulnya semuanya pulau karang. Dan kalau terumbu karang yang ada di tepi pulau yang agak besar itu berfungsi sebagai pelindung pantai, sehingga pantai tidak terjadi abrasi. Seperti saat-saat ini di beberapa lokasi di Indonesia banyak terjadi abrasi karena terumbu karangnya rusak gitu. Jadi pada prinsipnya penyebab kerusakan karang itu ada dua, yaitu pada umumnya adalah secara alami, yang kedua adalah karena aktivitas manusia. Jadi pada prinsipnya aktivitas manusia itu pada umumnya adalah seperti contoh di sini Teluk Jakarta dan sebagainya, itu karena adanya polusi dari kota Jakarta. Polusi itu datangnya dari zat-zat pencemar dari industri maupun dari rumah tangga.
Di Teluk Jakarta ini masalahnya parah sekali karena yang membuang limbah itu adalah 18 juta orang di daerah Jabodetabek ya, lewat kira-kira 17 sungai Jadi memang sudah jauh beda dengan keadaannya dulu.
Lalu yang kedua, aktivitas manusia tentunya saat ini banyak sekali mereka menggunakan teknik-teknik yang untuk tangkap ya, menangkap ikan, tapi mereka tidak ramah lingkungan. Karena mereka yang pertama seperti contohnya menggunakan bom, dan yang kedua contohnya juga menggunakan sianida gitu. Misalkan untuk nangkap ikan kerapu yang besar yang sekarang banyak dijual di restoran itu yang hidup, itu adalah pakai sianida, dan sianida ini merusak terumbu karang.
Perbedaannya ikan yang ditangkap sama jaring benar-benar segar ya dia. Kalau ditangkap dengan bom, itu ikan karena dia pingsan dulu, otomatis di perutnya kemasukan air jadi ngembung dia, ngembang. Terus matanya pun agak merah. Itu bedanya ikan yang ditangkap sama bom. Kalau dia keracunan kebetulan saya nggak ada ya. Tapi kalau biasa keracunan tuh dia punya ininya udah hancur perutnya. Itu keracunan. Tapi itu banyaknya di ikan baronang sama ekor kuning. Kalau ikan kakap merah sampai hari ini mereka jarang, paling nggak tangkapan dengan bubu. Biasanya kalau baronang, kalau bom itu matanya jadi merah dia. Terus perutnya bengkak, kembung, besar gitu. Itu satu tanda-tanda ikan nangkapnya pakai bom.
Ya kalau tuan bius itu yang anak ikan tuh terus disemprot begitu, terus kambang ikannya. Terus dipindahin buru-buru tuh ikannya hidup. Kalau tidak dipindahin buru-buru ke di air lain, itu terus mati. Tapi kalau kena karang itu obat itu, ya pasti mati. Karang itu ya, dia pasti mati dia.
Saat ini juga ada yang sifatnya alami, walaupun secara tidak langsung penyebabnya juga manusia, yang kita namakan bleaching atau karena bleaching itu suhu air laut naik lalu menyebabkan kematian karang ini. Nah, disebut karena warna karang itu menjadi putih dan akhirnya mati, itu yang disebut sebagai bleaching gitu.
Kalau terumbu karang yang ada itu di Kepulauan Pulau Putri, pulau Seribu sekitarnya, dan tentunya itu udah hanya tinggal 15-20% paling topnya. Ya tentunya itu bagaimana kesadaran menjaga lingkungan oleh para pemilik pulau atau oleh pemerintah, itu saja. Yang lainnya sudah pada habis.
Kepulauan Seribu sih ya parah ya, karena sangat terpengaruh oleh pencemaran dari Teluk Jakarta yang berasal dari darat, dan di samping itu juga mengalami perusakan, entah karena pembauman ikan, entah karena penyalahgunaan terumbu karang untuk bahan bangunan, entah karena dirusak oleh orang-orang yang nggak ngerti yang menduduki pulau-pulau di sana gitu. Ya tentunya karena terumbu karang juga punya fungsi proteksi pantai ya. Kalau terumbu karangnya hancur, pantainya kena berbagai macam problem seperti abrasi, sedimentasi, dan juga intrusi air laut, sehingga sumber-sumber air bersih kemudian hilang dan yang ada di sana cuma air payau yang tidak bisa dimanfaatkan. Di kerugian ekonominya besar sekali.
dari segi proteksi pantai tadi, kita sebagai contoh kan di daerah Bali juga gitu, itu untuk melindungi satu kilometer pantai dari abrasi, itu yang padahal itu tahun 95 itu menghabiskan dana kira-kira 2 miliar rupiah untuk per kilometer untuk membuat ya suatu barier biar tidak terjadi abrasi. Nah, kalau kita hitung dari situ, berarti kalau sebetulnya kalau karangnya tidak rusak, mereka tidak perlu membangun barier itu, sehingga per kilometernya mencapai 2 miliar rupiah.
Pada umumnya, masyarakat di daerah terpencil dan di pulau-pulau kecil itu sangat tergantung kehidupannya dari terumbu karang. Jadi kalau terumbu karang kita punah, maka banyak sekali kawasan di Indonesia yang tidak layak huni lagi karena sumber dayanya habis begitu. Dari segi ekonomi tentunya besar sekali. Misalnya ya, kalau 1 hektar terumbu karang saja bisa menghasilkan ikan 0,5 ton saja setahun. Ya, bisa dihitung tuh berapa ruginya. Kalau misalnya 1 kilo ikan tuh harganya katakanlah $1 ya, kalau kita bicara dolar ya, 500 ton itu berarti 500.000 kilo kali $1, $0,5 juta per hektar ruginya.
Kalau dilihat kerugian memang besar sekali, dan saya tidak bisa nyebutkan berapa besar ini ya secara detail ya. Itu seperti para saintis yang memperkirakan bahwa terumbu karang itu kalau diambil sekejap saja mendapatkan devisa, tetapi kalau untuk dipelihara mendatangkan devisa dari turis, dan itu besar sekali efeknya ya, hasilnya untuk pemerintah. Tetapi juga kalau pengerusakan terumbu karang berlanjut, ini akan terjadi abrasi pantai, dan itu betapa besar nggak bisa diperkirakan lagi karena tidak bisa diulang lagi peristiwa itu. Contohnya sekarang kalau Anda lihat di pulau-pulau dengan adanya tembok di sekitarnya itu kan estetikanya jelek sekali, tapi harus melakukan itu. Kenapa tidak dilakukan? Karena kalau tidak dilakukan tentu akan terjadi abrasi besar-besaran dan pulau hilang. Banyak pulau-pulau yang sekarang di sekitar Pulau Seribu hilang sama sekali dari pandangan kita. Makin hari makin mengecil. Kenapa? Karena ya tunggul-tunggulnya udah pada habis. Ada dipakai untuk bikin rumah, dipakai untuk jalanan, dipakai untuk septic tank, dipakai untuk ekspor ikan hias. Dengan karang-karangnya, itulah yang menjadikan pengerusakan besar-besaran.
Kerugian di tingkat nelayan dengan hilangnya karang, nantinya akan kita lihat bahwa nelayan itu semakin lama semakin susah mendapatkan ikan. Mereka akan berubah, seperti lihat saja ini beberapa nelayan di Pulau Pari ini yang dekat dengan kita di lokasi ini. Sekarang mereka orientasinya sudah bukan nelayan lagi.
Dari pengalaman saya sendiri saja setelah mewawancara beberapa anggota masyarakat di pulau-pulau kecil, itu mereka cerita, terutama orang-orang tuanya, bahwa dulu tuh mereka dapat penghasilan yang bagus dari terumbu karang karena ikannya banyak. Tapi karena terumbu karangnya sudah dipakai bahan bangunan, sisanya dibom, itu sudah jadi kayak gurun pasir, nggak subur lagi gitu, sehingga masyarakatnya jadi melarat dan harus pindah, migrasi ke kota, ya toh, kemudian menempuh gaya hidup yang sama sekali baru yang tentunya tidak mudah bagi mereka.
Sekarang usahakan nggak seperti dulu lagi Bapak usahanya. Jadi ini perahu, dari tadinya perahu nelayan, terpaksa dibikin restoran ikan bakar karena nggak ada jalan keluarnya lagi, macet total. Jadi ini begitu. Jadi 2 tahun berjalan.
Kami mencari ikan di pantai lautan sangat banyak gitu. Sekarang sekarang susah sekali. Sekarang saya mencari tengah lautan, cukup banyak di tengah lautan.
overall saya kira 40% dari masyarakat kota Jakarta butuh konsumsi ikan.
Ya ikan kan nggak banyak kolesterol daripada orang makan steak, menurut saya gitu loh.
Jadi solusi-solusi yang perlu kita lakukan yaitu antara lain adalah pengendalian aktivitas manusia sendiri. Nah, pengendalian aktivitas manusia ini bisa berbagai macam karena kuncinya adalah pengendalian aktivitas manusia. Ya, misalkan seperti tadi untuk mengurangi yang melakukan pengeboman, lalu untuk mengurangi juga yang aktivitas menangkap dengan sianida, ya kita tentunya harus menegakkan penegakan hukum itu harus dilakukan, lalu undang-undangnya juga diperbaiki sehingga orang-orang yang melakukan pengeboman dan melakukan penangkapan ikan dengan sianida itu bisa dihentikan.
Yang paling efektif sih pemerintah mengadakan penyuluhan, cari solusinya. Jadi kalau kita pengusahaan, suara kita kan nggak pernah didengar. Jadi kalau saya pribadi, saya menghimbau kepada pemerintah.
Ya, karena saya sudah merasa bahwa jengkel sekali ya, kemana-mana udah mentok, nggak ada tindakan nyata dari pemerintah. Saya coba waktu itu, tahun berapa itu saya lupa lagi, menulis surat ke Menpolkam Susilo Sudarman, katanya saya dengar-dengar dia concern dengan lingkungan. Saya menulis surat bahwa ini harus ditegakkan ini hukum ini, kalau diambilin terus ini nggak benar ini. Saya pernah menanyakan itu dan tanggapannya juga masih ya ada jawaban tapi ya sekedar jawaban saja.
Artinya kita memberikan dorongan bagi masyarakat setempat untuk melakukan manajemen sumber daya lokal termasuk terumbu karang. Dan itu sudah berhasil di beberapa tempat seperti di Sulawesi Utara, di NTB, di NTT ya. Tapi jumlah pekerjaan seperti itu harus diperbanyak. Kita sudah mulai langkah-langkah pertamanya.
Sebetulnya dalam pemanfaatkan karang untuk perdagangan itu ada 3 kelompok yang saling berhubungan, yaitu satu itu nelayan, pengambil, lalu ada yang istilahnya pengumpul, yaitu pedagang perantara, dan eksportir. Mereka semuanya harus ada sertifikasi. Hanya orang-orang tertentu yang bisa melakukan istilahnya perdagangan karang ini. Kalau tidak, nantinya juga saya khawatir itu bisa rusak. Jadi sebetulnya daerah Pulau Seribu atau Teluk Jakarta dan sekitarnya ini adalah suatu daerah yang sudah sangat kompleks dengan masalah laut ini. Karena apa? Di Jakarta ini boleh dikatakan kalau Anda bisa mencari solusi untuk memecahkan masalah Pulau Seribu dan Teluk Jakarta, berarti Anda bisa menyelesaikan seluruh problem berhubungan dengan laut di seluruh Indonesia. Saya katakan begitu kenapa? Karena di sekitar Teluk Jakarta dan sebagainya, atau di sekitar Pulau Seribu itu sangat-sangat kompleks persoalan di Teluk Jakarta itu. Nah ini mereka jadi solusinya itu juga tentu semuanya harus kompromis antara semuanya.
Berawal dari cita dan asa
Okay, ladies and gentlemen, it's almost 1 o'clock in the afternoon, Indonesian Western Time. In about 5 minutes, we will start our afternoon sessions. But before we go to the afternoon sessions, we will have yes, okay, I will inform you before we start the session with the conference etiquette. Okay, let me read it again to you. All speakers and participants must use a proper ID indicating real name and affiliation. All speakers and participants allowed to give in question to the speaker or presenter via menu chat available in the screen of the Zoom meeting by the following format: name underscore affiliation underscore question and underscore to whom. Please make sure your microphone is in mute position unless you give question or comments during the Q&A sessions. 45 minutes of presentation time will be allocated for each keynote speakers, including 15 minutes Q&A sessions. 20 minutes of presentation time will be allocated for each invited speakers, including 5 minutes of Q&A sessions. 15 minutes of presentation time will be allocated for each oral presentations, including 5 minutes of Q&A sessions. This certificate will be sent through all the participants' email address 1 month after the conference. Okay, thank you, Sylvia, for sharing the screen. Let me check to our keynote speaker for this afternoon session. Let's say hi to Professor Willem Renema from Naturalist Biodiversity Center. Professor Willem Renema, are you ready for this keynote session? Let me check on the, okay.
Yeah, that's okay.
Okay, hello, Professor Renema. I will check with our moderator for the keynote speech session. This will be to Dr. Hagi Yulia Sugeha.
Yeah, hello, Sophie. Thank you. I'm ready.
Okay, we start our session for this afternoon. Okay, Ibu Yuli. Okay, let me check on our participant first. Okay, ladies and gentlemen, thank you and welcome back to our afternoon sessions. Before we start the presentation sessions, we will have the keynote speech session, which will be delivered by Professor Dr. Willem Renema from the Naturalist Biodiversity Centers. Act as our moderator for this session will be Dr. Hagi Yulia Sugeha from Research Center for Oceanography LIPI. Okay, Dr. Hagi Yulia, time is yours to start the session.
Thank you, Master Ceremony, Miss Sophie, for a nice introduction. Hello, everyone. Assalamualaikum warahmatullahi wabarakatuh. I'm Hagi Yulia Sugeha here. Allow me to be your moderator in this afternoon session. But before I continue to introduce our keynote speaker today afternoon, I should let you know that actually, normally the keynote speaker will be put in the earlier session or in the morning or in the first session as a keynote speech, of course. But because of difference in time between Amsterdam and Netherlands and Indonesia, so our keynote speaker today asking the committee to put his schedule on the afternoon. So that's why our keynote speaker today, Professor Dr. Willem Renema, will just give a speech today afternoon.
So as you may see here, our keynote speaker today is Professor Dr. Willem Renema from Netherlands, with his topic about brown water reef as potential refuge for coral reefs in times of rapid environmental change. Professor Willem Renema is a professor from the Leiden Naturalist Biodiversity Center, the Netherlands, and Institute for Ecosystem and Climate Dynamics, University of Amsterdam, Amsterdam, Netherlands. So his expertise on marine biology, and it will be very interesting because today he will not talk only about marine biology, but also about impact of climate change on the marine biodiversity, especially for the coral reef ecosystem. So without waiting more longer, let's give a time to him. So Professor Dr. Willem Renema, time is yours.
Okay, thank you. Could you allow me to share my screen?
Yes, of course, please.
I think I would like to help from Tia, our host, to give the Professor Renema as co-host so that Professor Renema can share the screen, please.
Okay, I think that works. You see my screen? Just checking.
Yes, it's already, Prof. You may continue. Yeah.
Cool. Great. Well, thank you very much. Thank you very much for inviting me for this presentation. Thank you very much for allowing me to sleep a little bit longer. And yeah, it's great to be able to give this talk. Although I must admit, I would have rather done it in Jakarta than here from Leiden. But yeah, there's this situation that we can't travel. So yeah, I would like to talk today about brown water reefs. Many people have coral reefs are very well known, certainly in Indonesia, of course. You live close to them, you well, see them very often. But in at least in perspective, they often are associated, and when people talk about coral reefs, then it's all often about these clear water settings, that they are limited by nutrients, that there's little sedimentation. But actually, and certainly in Indonesia, you know that many of the coral reefs occur in what I used to call, or used to refer to, in brown water conditions, which are conditions where there is more terrestrial influence, there's more sediment, there's more nutrients. And we actually think that these are really important, or can be really important environments for the continuance of the of coral reefs in the future. And as you also probably all know, these coral reefs, and again, especially the coral reefs in Indonesia, are extremely diverse. There's many species of not only coral, but also many other associated taxa, so fishes, sponges, algae. Most organisms that live in shallow marine environments have their highest diversity in Indonesian coral reefs. As such, they already provide a huge value because of, for example, unknown medicine that can be discovered. But they also provide a much more direct value through fishing, through coastal protection, through tourism that is of invaluable asset for the community. So we have to look into what's going on with coral reefs at the moment, and I want to do that with first looking a little bit on what's going on now, and then step a bit back in time and look at the past. Like, can we learn from the past what has happened in the past, then go back to the recent and then say some things about well, what might be actions we could take for future conservation. And another thing that coral reefs are, well, I would almost say known for, or at least many places where I go, then I talk about coral reefs and I say I'm a coral reef researcher, then people say like, oh, they're all dying because of bleaching, because bleaching is the thing, at least in here in the Netherlands, it's a lot in the news that there is another global bleaching event or large die-off because of bleaching. So, but bleaching is something that that has definitely increased in time. So recently there's been, and this is a graph that only lasts until 2016, but actually in 2020 there was another bleaching event in the Great Barrier Reef. This is and in many other places, for example in the Indian Ocean. So bleaching is occurring more frequently, and bleaching is a response to both temperature and light. So then there's an increase in light, increase in temperature, then the photosymbionts of the corals start to produce, become more active. They cause photooxidative stress in the coral polyp, and then the symbionts are expelled from the coral. So, and then when that lasts too long, the coral dies and has to recover from scratch. But if recovery is quick enough, then they can just recover to their original state. But that is all about time, and what is especially true, and this is an example from the Thousand Islands. Just saw a movie that was just a little bit later than this, but what you see is that this mass bleaching, this is one of the mass bleaching events that's also really impacted Indonesian reefs in the 80s. But what you see is that bleaching has a really direct impact, so that the reduction in coral cover is rapid, but the recovery takes a long time. The corals have to recruit onto the well, in this case reef flat, and they have to grow, and that they don't grow that fast. So to get some cover takes a lot of time. And one of the restrictions of current data sets, and again, especially in Indonesia, or maybe even especially in the Coral Triangle, some places have been really well studied, but they are often in Australia, in Japan, Hawaii, where there's long time series, but these longer time series are often not available. And even in these well-studied places, often these time series start following the big disturbance. So this is probably one of the few that actually has a measurement from before the 1982 bleaching event. And so many of these time series actually start measuring after already something big has happened. So you do not know where you're coming from. So what we want to do we want to look into these turbid reefs, these brown water reefs that there's two ways of reducing temperature. Is something that is widely distributed, that's in seawater. So the water is flooding the shelves and the reefs, and when it gets warmer, there's little hiding from that. But from light you can hide, or at least you can occur in environments where there's less light. So you would at least have a little bit less of the light induced stress. You can either go deep, mean, water is absorbing light, so when you go deeper, you have less light, so the corals might be sheltered by depth from the light. And the other one is brown water. But brown water reefs have always, when you read papers about coastal reefs and so on, they usually be referred to as marginal habitats. So marginal habitats that do not that is not really good for coral reefs. But there's really good indications, like diversity is not lower in these turbid environments, carbonate production is not lower, accretion is not lower, so they seem to perform pretty well in these environments. The only thing that next to bleaching, there might be less bleaching, so maybe they are sheltered from bleaching, but another thing that is happening in these environments is often there are also more diseases. Some of the disease factors, like the crown of thorns in the bottom, it's a sea star that is eating corals. There's really good indications that there's more nutrients in the water, that the larvae disperse better. And coral and diseases appear to be a little bit more frequent in these turbid environments. So there's more potentially more other disturbance going on than only the bleaching frequency. But as I said, what we need to look at is like how has this been dealt with in the past. So I would like to take you on a tour a little bit further back in time. We go to Mangkalihat, so in Kalimantan. Samarinda is here. Mangkalihat Peninsula, Sangkuliran is here, and along this road or the side of the sea, there are some really good outcrops of fossil coral reefs. And there's a lot of development, or at least there was a lot of development going on in 2010, we did this research between 2010 and 2012. And so we had a really good access to these fossil reefs that are really well preserved and provide a lot of information on how reefs evolved. And in general, the reefs get younger when going from Samarinda to Sangkuliran. So when you go north, the reefs get younger. Here they are about 20 million years, and here they are about 8 to 6 million years. And the first reef that we saw in this way was what we refer to as the 3D reef. We drove past it and we initially looked at it and thought, like, wow, this is a really typical reef outcrop, very similar to what we see in modern reefs. But well, we will measure it, we will study it and look at the species composition to see whether there are changes in species composition. And we had a group of students in a large project that worked on it together with the Geological Survey in Bandung. And we measured the reef, we sampled the corals and the rocks in the reef, and we came up with a history for the reef. And it still sort of looked like a very familiar side of a reef. So it's a very muddy environment below the substrate. So there's these weird looking large benthic foraminifera that actually stabilize the sediment. On top of these forams, you get colonization of corals, you get a coral reef development with a really three-dimensional structure. There's more sediment and there's the forams again, and then you get another phase of reef development, and then it's smothered in sediment again. So we thought like, all right, this is familiar, this is what we can see in the recent as well. But the surprise came when we started looking at the coralline algae. because these coralline algae indicated a really high depth, and we thought this was, from the modern analog, a shallow reef. And what this meant was that this was a type of reef that we actually do not have, or that is very rare at the moment, in not only in Indonesia but everywhere. And it's a delta formed reef. So it's a reef that develops in the delta of a river on for example sandbars that are there on the side where there's little sediment runoff. And when the river is sedimentate, there's a lot of sediment discharged, like in the Mahakam River, then these are individual reefs that are separated on the different sets of the river. When there's low sedimentation, there would be stacks and it would look like higher and larger structures. But it was a non-known reef. And we did this for the other reefs and I'm not going in detail about these other sites. You can read either my paper, which is about the dating, and Santo Domingo's Nadia's paper is published in Coral Reefs, and that is about the sedimentary environment and the coral composition of the reefs. But what we found was that all these reefs occurred up to 8 million years, occurred in really turbid environments. There were no reefs that were in highlight environments. It was always very muddy. There were large coral carpets, so shallow areas that had very little relief, so that didn't grow into shallow environments to get more light. And only around 8 million years or so, maybe 10, but there was a increase in the relief, and you get these modern type zonations in your reef where you find environments that are in the highlight zone of the water column. So these appear to be pretty young. And we related these reef phases to sedimentation in Kalimantan. So there's the uplift of the central part of Kalimantan, and when you get more uplift, you get more sediment into the coastal system. And these gray areas is where we find the coral reefs. So first the platy ones, then the branching coral carpets, then the highlight high relief reefs, and they all coincide with these periods of lower sedimentation. So there's disturbance. In this case, it's not bleaching but sedimentation. So when they can deal with sedimentation, but at some point they go over the limit. The sedimentation rates go over the limit of tolerance. The reefs disappear regionally, so in the entire basin, and in fact around Kalimantan that's quite synchronized. That's paper we still working on, and the sedimentation rate, and so in fact the uplift of Kalimantan is driving the distribution of reefs in the area. But then we look, and as I said, Nadia Santo Domingo did a really big job in identifying all these fossil corals. And when we look at just species composition is not that different between all the reefs. It's just that the abundance of some taxa, so the abundance of platy corals, of massive corals, of branching corals is very different between the reefs. But the species composition is very similar. So with a similar species pool, you can result into, because of different environments, you can get completely different looking coral reefs. And then there's another thing that we observe there, and that's Acropora. And Acropora, the staghorn corals, that is something that is really striking to many coral reefs. When you see the 3D complexity of coral reefs, it's often related to staghorn corals. But we didn't see them, or hardly saw them, in these old reefs. So we went looking into it. Is this really something that's going on regularly? Is this just here? Or is it a global observation that in these older reefs, there's hardly any, there's no Acropora dominance, really high abundance of coral, of Acropora. And that's basically exactly what we found in the older situation between 16 and 5 million years. We hardly find, we have lots of places where we found coral reefs, but we had no places where there was this high abundance of staghorn corals. In the Pliocene, 5 to 2.2 million years roughly, only one place, Curaçao, where there is high abundance of Acropora, and then only younger than 2.2 million years, actually in many of the places where we do have outcrops. That's a period that we do not have a lot of outcrops, but where we have data in many of the places, we have also this Acropora dominance. And if you go even younger, then it's basically the standard that there are places and time intervals in reef development that there's a lot of staghorn corals. And again, we relate that to disturbance. Disturbance in this case, it's not sedimentation, but the disturbance is the sea level movement during the ice ages. So during the ice ages, sea level can move 120 meters down in the youngest ice ages, and in earlier ones a little bit less. But the rate of sea level rise, the highest that has been recorded, could be up to 15 meters per 100 years actually. So it's really, really, really fast. That are rare events, of course, but have a big impact on the corals, on the coral reef distribution. And most of that has to do with the depth distribution. Most of the shelves and where the reefs occur currently is in relatively shallow environments, but when sea level drops 20 meters, there's not so much habitat reduction. But as soon as there's a reduction of about 60 meters, then the reef environments sort of drop off the shelf, and you have only very small amount of area left where you can develop coral reefs. And if you have a really high amplitude, then there's even less, and they have to, when sea level rises, they have to recolonize the shelf. And you see that species that are really good at colonizing and really fast growing do that faster. And Acropora is just that. It's a species that is basically a weed. It's a taxon that can colonize fast in times of sea level rise. So here again, disturbance is shaping the environment, but it is just making the reefs look different. So Pliocene reefs look different from Miocene reefs. So what about modern reefs? So what's going on? So yeah, this is a picture from the Spermonde Islands, Makassar, where I will bring you to. You have a nice data set there. This is a picture from 1997 when I started doing my work there. And the Spermonde is a really nice area because there's a big city, Makassar, there's a good infrastructure, there's a university we work with. And these reefs occur in a gradient from near shore to offshore and from near the city to away from the city. So we can look like what's going on. And I'm just going to go for the generalized picture. There's reefs. Most of the corals grow on the west side. There's more sandy. This is a reef that's a little bit of an exception, but also quite a bit of coral on the east side. But in general, west side. So we measured corals, coral cover on the northwest side of the reef because usually we had to standardize. And we measure and we have data available starting in 1989 and ending in 2018. The only disadvantage of the data set is that they're not very high density of measurements. So we only have we have some gaps in the time series, but the pattern that we find is quite clear. So when we look at coral cover, then actually the most studies measure coral cover in these upper reef slopes, so between 3 and 7 or 10 meters deep. That's where they measure, and so that's what we did as well. And what we find is that actually coral cover is really high. That in 2013 it's a little bit lower, but it's already in 18 it's already getting higher again. And the algal cover is also quite low. Again, in 2013 it's a bit higher. But but in general, and if you compare these types of coral cover, and of course coral cover is not everything, but if you can compare this type of coral cover with, for example, an average of the Great Barrier Reefs, they have an average of about 20%. So we have an average about 50%. So the coral cover is pretty high in these environments. So that looks like good. But when we start looking in different environments, then the picture changes a bit, and the picture changes especially when we look in the reef flats. And one thing that we find is that there's much less data, and so coral reef researchers do not really they like to dive, so they like to be on the outside, and they don't, and in many cases they actually ignore this reef flat area, but that's where we find the largest change in conditions. So coral cover already in 1997 was not that high, it got a little bit lower even, but I mean it's still not. But what especially is changed is that the algal cover has increased a lot, and when in 1997 often you still had to swim really carefully not to hit the corals. In 2012 and so on, there's a lot of the reef flat, the shallow part, really there's a lot of die-off, many places with really high canopy algae. So this is really big change. And then the question is, has this always been like this? There's a big, but I tried to publish this story and I got the comment like, oh, but it's always been like this. Just because these...
You have remained 3 minutes. Thank you.
Yeah, I will need 5. That's good. But these reefs, they reef the sea level, and there's no accommodation space, so there will be very little corals growing on the top of these reefs anyway. And what we then also have is these reefs from 1928, where we actually see that in 1928, Sarapo was visited by Professor Umgrove, and there the corals are growing even though there's no accommodation space. And in 2018, exactly 90 years later, students visited this island and I asked to take this picture, and then it's all seagrass. So the corals have disappeared from this reef flat, so this pattern appears to be real. This is a detailed, more detailed picture where you see the corals even at low tide sticking out of the water. Something you do not see at all anymore. So again, recovery is high, but because of the disturbance events, the structural complexity is a bit less, and the shallowest parts of the reef have changed a lot. So there's a contraction of the coral zone on the reefs. And then I would like to go to the last part, and it's about like, is there a future for coral reefs? And the discussion at the moment has been a lot about like, do we manage resilience or not? So do we have protected areas? Are they effective or not? And the tendency at the moment is to say like, no, protected areas, because they manage fish densities, they have little impact. The big question, of course, is are these data? I mean, there's little data to really test it. There's very few longer time series. This is one from Kenya, published by John Bruno. And in Indonesia, situations are different. There's more people, there's diversity is higher, habitat diversity is higher, and it are these turbid reefs. So maybe there's stuff different. And another thing that about these studies is that the only action that is evaluated is keeping out fisheries and not doing anything about, for example, stimulating coral recovery and recruitment. And you can do that passively. This is an example from, not from a coral reef, obviously. This is just from a cold tidal flat in the Netherlands, like it is right now. But we have there's a company, Bureau Waardenburg, who has these elements of, they actually made, they're disintegratable, so they disappear. You have it's not plastic. And they stabilize the sediment, and you see that where they are, that you get more settlement of, in this case, plants, but there's also examples of corals, but then you do not see the elements anymore, so I removed it. So I took this picture, and then in Pulau Seribu, the Mars company has a community project on large-scale rehabilitation by using these star-shaped structures, and they attach corals to it, so to actually even already help recruitment, and that has shown that has increased coral cover on the island where they did it first, the Pulau Badi. So maybe just protected areas are not sufficient, but if you do protected areas and some sort of action to help the corals recruit, then at a large scale, that probably will be a way to protect coral reefs, with the realization that these reefs will then actually look different. So it's not a restoration in the sense that we go back to the conditions that to a reef that it was in the 1990s or 1960s, but we go back to a future reef that will look different, that will have a different feel than these past reefs, because conditions have changed and the coral reefs will adapt to that. So, well, thank you very much. It's time for questions.
Yeah, thank you, Professor Willem Renema, for the very interesting presentation. Yeah, we are very excited to know that from 1928, that before Acropora is there, until now the Acropora become a higher in population around Indonesia, of course. And also you mentioned about several important things affecting the coral ecosystem, such as population, species diversity, habitat diversity, and also the turbid reef, of course, in Indonesian water. So I think all the audience now, maybe you have a question. We prepare at least 10 to 15 minutes for discussion or question and answer. And yeah, while waiting for the others, I have already two questions for you, Professor. This is coming from Dr. Fransley Hukom of the Research Center for Oceanography, LIPI. His first question is, Professor Renema, what do you think about the Coral Diversity Center in Indonesia that need to be considered? And second, with regards to the collaborative works between LIPI and NDC, the plan of key expedition is really to be a hot issue for us. Do you have any reason why the area should be explored more? Thank you.
I think that when to the first question on the coral triangle center and the high diversity, yes, I think it's really important. I mean, in Indonesia, around Indonesia and the coral triangle, 70 or 80% of the corals occur, but that's only what we know of, and corals are well documented, but there are so many other groups that we know so poorly. So there's still a lot to discover, and we really, and that sort of merges into the next question, that we should really up the efforts, and it would be really great if we could manage to get that expedition and to East Indonesia, and especially to well, Kai, but there are more areas that you can say that are data deficient, or where there's been very little work done. But Kai Islands is definitely one of them, and so that would be a really great opportunity to reinstate our history of joint activities in Indonesia. Between LIPI and the current work that I'm talking about, we mostly do with Universitas Hasanuddin, and with and we do work a lot with Universitas Patimura in Ambon. But really getting the diversity inventories going, then LIPI would be the partner to work with. So yes. I think, and then Kai is interesting, and is a sort of a compromise between the marine work and the broader scope, with not only having marine work but also some terrestrial work for the botany. And Kai has has been visited by Snellius in 1980s and by Snellius in 1928, so we can actually compare results from what we do there now with what we know from the past. So in that sense, again, it's looking at the history of diversity, making new discoveries, but also looking back into the past.
Okay, thank you, Professor Renima, for your answer. So any other question from the audience? Okay, I think no question. So I have one question for you, if you don't mind. I'm working with fish, actually, some others coming from the coral reef area. And I really interested with your recent explanation about the completely changed from about 20th century for the acropora. And while the acropora is the coral reef building spaces for reef spaces, reef building spaces for coral. So my question is, what's the connection with the associated species, especially for fish species, maybe related to the acropora? If you have any information, that will be important for us, because it means probably we can also focus to study on the evolution of the species that are associated with coral reef, especially for the coral reef with dominantly by the acropora species, because I know, if I'm not wrong, Indonesia is one of the center for the acropora species. Thank you.
Yeah, no, definitely. I mean, acropora is the reduction of acropora will have a big impact on fish, because acropora is also one of the taxa that is generating this high structural complexity on the reef. So there's many places where they can hide from predators, where they reproduce. You often see the fish also going in between the acropora when you swim over the reef. So a situation where you have less structural complexity will definitely have impact on the fish. I would, because I'm not a fish person, I would not be able to say like which taxa are the ones that will be impacted by that most. But yeah, I mean, I can always ask around, of course. There's people who study fish much more than I do, but unfortunately, it's impossible to do everything because it's too much.
Thank you, Professor Willem Renema.
But it would be really great to actually look together on these associations between fish and not only Acropora, but also Acropora and other types of coral to see whether we actually can find a relation with that, and then also with the evolution of the center of diversity.
Okay, thank you. And we have another question. This is coming from Professor Samwat Huizen. I'm not sure whether this is a question or kind of a opinion. Let me read. I think the coral bleaching is the main factor in degradation the coral Indonesia, such as in the El Nino event in 1983, 1997, 2010, and 2016. How can we overcome these problems to protect the corals in Indonesia? This is a question.
I mean, the problem with bleaching is that it is mostly globally induced. What is interesting about Indonesian reefs is, well, one is that recovery is really surprisingly quick. So in 2010, our survey was quite quickly following bleaching. Well, two things there. The one is that there was already a high density of really small colonies, so the recovery potential, the recruitment, and the growth is really high. So it appears that Indonesian reefs can recover really quickly from bleaching. The other thing is that, for example, in the Spermonde, and I have not visited that regularly other areas. I've visited many areas, but not in time series. Only a bit of data on the Thousand Islands as well. But in the Spermonde, these mass bleaching events had very little impact. So there was some local and strange bleaching. Professor Jompa said that in 2010 there was actually bleaching at depth, but not the mass bleaching as you would see in the Great Barrier Reef. So it appears that, I mean, the 1980 bleaching event, the 1982 bleaching event definitely had a big impact all over Indonesia, but the other bleaching events appear to have a bit more patchy impact on. I mean, there are definitely areas where there's a lot of impacts. On Bali there was a lot of bleaching, but it's not that that all reefs have been impacted as severe as others. So, but bleaching is not something that, I mean, the only way to prevent bleaching is basically prevent climate change, and so we all have to be careful with CO2 exhaust into the atmosphere and mitigate climate change. I mean, because that's the driver for these increased El Nino frequencies.
Okay, thank you, Professor Willem Renema, for the answer. And I hope Professor Sam and also Dr. Franz Hukom will be satisfied with his answer as well as for me. And I think we should finish your session as keynote speech for today afternoon. Again, thank you very much, Professor Willem Renema. We are waiting for the future to collaborate with you. So everybody, thank you very much to join this keynote speech this afternoon, and I hope to see you again in the next session. Good afternoon. Assalamualaikum warahmatullahi wabarakatuh. I give back this next session to the MC. Thank you.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Haji Yulia Sugeha and also Professor Renema for the session. Okay, ladies and gentlemen, let us move on to our next agenda, we will have 5 invited speakers and also 2 oral presentations. And this session will be conducted by Mr. Arif Rahman as our moderator for this session. Okay, Mr. Arif Rahman, time is yours to start the session.
Thank you very much, Ibu Sofi. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. Welcome to the afternoon session of this Icoes 2020 for the day 2 afternoon session. As Ibu Sophia said, we have 5 invited speakers for this afternoon session and 2 oral presenters. And for our first invited speakers, I would like to ask Dr. Udi Eko Hernawan from Research Center for Oceanography, Indonesian Institute of Science, to present their presentation on environmental factors that drive the distribution of a colonizer seagrass in the Indo-Malay archipelago, predictions under climate change scenarios. Dr. Rudy, are you ready?
Yes. Let me share my screen. Thank you, everyone. Good afternoon. In this session, I will talk about the environmental factors that drive the distribution of seagrass in Indonesia or Indo-Malay archipelago, and also projection under the climate change scenarios. So to start the talk, I would like to again emphasize the significance of seagrass of Indonesia's seagrass in a global context, because of its ecosystem services. If you look at blue carbon context, recent studies state that the carbon sequestration by Indonesian seagrass is at least 2-fold higher than the global average. And together with mangroves, to a smaller extent, Indonesia's carbon storage roughly 17% of the world's blue carbon. So that's from the blue carbon context, and that's why it helps mitigate climate change. From biodiversity perspective, seagrasses are home to many species. We can find invertebrates or vertebrates in these habitats. In Indonesia, we can find 16 seagrass species. And this habitat also support fisheries production. In the context of Indonesia, many economically important species can fishes, mollusks, crustaceans can be found in seagrass habitats, and it supports small-scale fisheries where many communities in Indonesia they live close to the coast, and so it supports livelihood for the coastal community. One more about the significant importance of seagrass is they help keep the water quality checked. They help stabilize sediment and protect coastal erosion. Also, recent study found that seagrass meadows filter water from bacterial pathogen. I'm not going to talk more detail about this part because this is just an introduction. So in this talk, I will focus on Halophila ovalis. This is a colonizer seagrass. We can find this in estuaries, lagoon, mostly in intertidal or subtidal areas in Indonesia, in many types of sediment, from coralline soft sand to muddy substrate. as a colonizer seagrass, it grows fast, but short-lived. So it's a fast turnover. And one thing for a colonizing species is that for seagrass here is that they have a rapid ability to recover from disturbance. On the other hand, they have relatively low physiological resistance to disturbance. So this is about the Halophila ovalis. So you can see how Halophila ovalis in the knowledge of seagrass. And then talking about the current knowledge of seagrass in Indonesia, there are many gaps of knowledge. If we compare with coral reefs, then seagrass is way behind our knowledge in coral reefs or mangroves, because, well, it receives less attention than the other two. I'm going to talk only two parts: habitat distribution. We still don't know what factors to determine seagrass distribution in Indonesia or Indo-Malay archipelago. Even the basic question of to what extent seagrass habitats are distributed, how much habitat, how much extent of habitat in Indonesia, we don't know yet. If we talk about seagrass resilience and response to changing environment, then it is even worse. I mean, we don't know much about that in seagrass. How do seagrasses respond to climate change or increase in temperature? Will they survive under increased temperature in the Indo-Malay archipelago? We don't know. If we search for literature, there's no studies reporting on that. Experiment studies, there's no study about that in Indonesia. So that's the gaps of knowledge, and we know that in the near future, 2100, our ocean is going to be warmer and then more acidic and less oxygen. In the scenario for RCP 4.5, for example, the most likely scenario, well, the ocean temperature will be warmer and then still more acidic and then deoxygenated as well. In this changing environment, the question is how seagrass will respond. That is the question, and to answer that question, I'm presenting this. This is just a preliminary work, our preliminary work. We still need to do a lot more to explore in detail. But then we use, as a start, we use a species distribution modeling to predict habitat availability of Halophila ovalis under different environmental conditions, especially in the different sea surface temperature. We use Maxent modeling, maximum entropy modeling, and then we use the occurrence data, seagrass occurrence data from OBIS, Ocean Biodiversity Information System, and combined with the Core Map CTI field survey. We include 7 environmental factors obtained from BioOracle: sea surface temperature, the maximum and min one, nutrient, phosphate and nitrate, dissolved oxygen, diffuse attenuation coefficient. It represents the water turbidity or water clarity, also photosynthetic active radiation, pH, and salinity. And we use 3 scenarios: current condition, and then the RCP 45 and RCP 85 in 2100. With regard to projecting distribution under different scenarios in climate change, here we we use sea surface temperature prediction in 2100. So what we found after we did the modeling is that we find that diffuse attenuation coefficient, or DAT, or have the highest percent of contribution to the response variable. It's almost 50%. And the second one is salinity. The other three, or the other five, nitrate, sea surface temperature, pH, salinity, is less contribute in the response variable. So from this table, we can see that diffuse attenuation coefficient and salinity are the most important factors in driving the distribution of seagrass in the Indo-Malay archipelago. And then we go further with, we do the jackknifing. And again, we find that the diffuse attenuation coefficient and salinity are the most important factors in determining potential habitat for Halophila ovalis. You can see here, I mean, the DAT.IAA, I mean, that is the diffuse attenuation coefficient, that stands for diffuse attenuation coefficient, and then salinity and sal.IAA is salinity here. Both in the test and the training data and the test data, they have the same pattern. That diffuse attenuation coefficient and salinity are the most important factors. I cannot. Okay. Now the response curve. For the diffuse attenuation coefficient, DAT.IAA here, we can see that this is the turbidity. With the increasing value of diffuse attenuation coefficient, that means the water become less clear, it becomes turbid. So with the increasing water turbidity, then it is likely we can find Halophila ovalis. So that's for the response curve for diffuse attenuation coefficient. The second one for the salinity, it seems like from this response curve, the Halophila ovalis Halophila ovalis habitat is optimal in high salinity water, high saline water. So we can find here high likely of Halophila ovalis in saline water. And for the sea surface temperature, the mean one, this is just an example. I just provide the response curve for the sea surface temperature. With the increasing temperature, less likely we would find Halophila ovalis. So that's the response curve. And using the environmental variables, then we could model the potential habitat for Halophila ovalis. And this is the map, the spatial map of seagrass Halophila ovalis distribution with the current environmental variables. We can see that in the western part of Indonesia, we can find a lot of Halophila habitat with high probability in these Riau Islands. Based on our field survey, for example, in Bintan or others, we can find a lot of Halophila in this part here. and also in many parts of the in the eastern part of Indonesia, like in Kepala Burung, Raja Ampat, somewhere. So this is the potential current distribution of Halophila ovalis. And then we project with the increasing temperature under RCP 4.5 and RCP 8.5 scenario in 2100. We can see that the area that was previously green, that means high probability of finding Halophila ovalis, like for example here in the Western Indonesia, in scenario RCP 4.5, then it's become lower. And then in the northern part of Australia, then it becomes greener. That means they most likely become a habitat for Halophila ovalis. And it becomes worse under RCP 8.5, and we find a potential habitat loss in most of Indonesia, Singapore, and Malaysia. So how much in percentage that we might be lost in 2100? Under RCP 8.5, potentially Halophila ovalis habitat would lose about 83%. And under RCP 4.5, the habitat loss would be about 26%. And in the northern part of Australia, under RCP 4.5, actually they will lose. It is likely there is a possibility of habitat loss, but they also gain habitat, and it's 168%. This indicates a distribution shift of Halophila ovalis. So, but then from this result, I have to stress that this is just preliminary results. So we need to test this possibility with other methods as well, which is still ongoing. And the consequences of habitat loss or species loss here is that in seagrass, this is a colonizer seagrass. Halophila is a colonizer seagrass, and loss of colonizing species in seagrass meadows would impact the resilience of the whole system. It might result in failed seagrass community succession, because in transient seagrass meadows, the presence of colonizing species in seagrass meadows is very important. If there is no colonizing species in transient seagrass meadows, that would hinder the process of seagrass community succession. And of course, with the species loss or habitat loss, then we would lose ecosystem services. So in summary, we found that turbidity and salinity are the most important factors in determining habitat availability for Halophila ovalis. We also indicate a potential distribution shift or latitudinal shift of Halophila ovalis that will likely to occur in 2100. And then there will be potential habitat loss for Halophila ovalis in 2100. And this loss may have significant ecological impact and may lead to loss of ecosystem services. Thank you.
Thank you very much, Dr. Udi, for the very interesting presentation. We still have about 2 minutes for question and answer, and I already see 2 questions from the chat. The first one from Cory Lipi. What do you think about sea level rise impact on habitat loss or gain in Indonesia?
Yes, of course, it will change the habitat, the bathymetry, the coastline, and it will change the potential habitat availability. Yes, it will have potentially it will have impact on habitat loss and gain in Indonesia. But we in this modeling, we haven't included that variables. That is why we still need to further do some calculation, do some further work to get a more robust result.
Okay, thank you. And then for the next question from Professor Sam Watuyan, you mentioned that the Halophila ovalis distribution prefer the saline water. Would you explain why Halophila more distribute around Bintan rather than in East Indonesia water such in K or Tanimbar Island? Thank you.
Yeah, I mean, Bintan is just an example. It doesn't mean that there is no Halophila in other areas as well. So it's because it's the salinity is locally, it can be locally specific. So I believe there is also Halophila in K and Tanimbar as well. I have visited K Island and Tanimbar, and I also find Halophila ovalis over there. So Bintan is just an example.
Okay, thank you very much, Dr. Udi, for such a wonderful presentation. Now, I would like to move to the next invited speaker, Dr. Michael Miller. Are you here? Dr. Michael? There you are. So you're still mute, okay.
Yes, I'm here.
Yeah. So the next invited speaker is Dr. Michael Miller from the University of Tokyo, and he will deliver a talk about the biodiversity of eel in the Coral Triangle. Now, the time and stage is yours. You might share your screen. Dr. Miller, can you share your screen?
Yeah, I'm trying to. Would that do it?
Yes, we can see it.
Well, hello to everyone around the world. It's nice to be part of this interesting symposium. And we're a little bit ahead of schedule, so maybe I should talk a little slower. But I'll be talking about the question of whether the high biodiversity of eels in the Coral Triangle will be affected by climate change. And I'm presenting today on behalf of our colleagues at LIPI in Indonesia, many of which who are part of this symposium, and my colleagues in Japan. So as everybody's been talking about during the symposium, the Indo-Pacific region and the Coral Triangle in particular clearly has the highest marine biodiversity in the world, partly because of the many coral reef areas. And it also includes a relatively high number of anguillid eels compared to most parts of the world, and many marine eel species, in fact, far more species. So marine eels are very diverse in their habitat use and shapes, sizes, and colors. And remarkably, they're found all the way from coastal intertidal zone all the way down the benthic slope to about 5,000 meters, and also in the pelagic zone, in the hadal zone and the mesopelagic zone. And their leptocephali are all found in the upper surface layer of the ocean, and they grow to much larger sizes than most fish larvae. They have diverse body shapes, sizes, and head morphology of the different family and genera. But most marine eels are never seen because they're mostly hiding during the daytime, and so their ecologies are very poorly known. And how climate change might affect them has not been evaluated. So today I'll start by just talking about some basic climate change factors that everybody's familiar with already. What are the possible effects on anguillid eels? And more specifically, look at the question of the high biodiversity of eels in the Coral Triangle based on larval collections. And the main focus is a global comparison of a number of species from Leptocephalus catch studies. And then consider a little bit about what are the possible effects on marine eels, such as coral bleaching, that's already been talked about in this session. So CO2 has been steadily increasing on planet Earth for the last decades. And this affects how much heat is trapped in the atmosphere, as everyone knows. And so in the last decades, since the 1970s, global surface temperatures have been increasing. And this increase from about the same time has been occurring in ocean surface temperatures around the world. And of course, there are ocean atmosphere factors such as El Nino, where the normal condition of trade winds blow to the west, and you have normal conditions of rainfall in the Coral Triangle. Whereas in El Nino conditions, those trade winds are weakened and resulting in eastward flow, and the rainfall shifts to the east. And so you can have droughts in the Coral Triangle during El Nino. And as other speakers have talked about, the Indian Ocean dipole can also affect surface temperatures and rainfall, such as more rain in Indonesia in the Coral Triangle, or less rain, or cold versus warm conditions. And of course, when you have cloud cover and rain, it affects the sea surface temperature in these areas. So as have been the focus of several presentations, high water temperatures can cause the coral bleaching of shallow reefs. But how might all these types of climate change affect different types of eels? But there are only 19 species of anguillid eels or freshwater eels worldwide. But for marine eels, there's more than 900 species worldwide. So first, how might these things affect freshwater eels? And in the Coral Triangle region, there's at least 6 species of anguillid eels, which is probably the most of anywhere in the world, including these species listed here. And as previous speakers talked about, all anguillid eels have the same basic life history. And also marine eels, they're spawned in the ocean. They have the larval growth stage of the leptocephalus, and then they metamorphose into glass eels. But in the case of anguillid eels, they have a yellow eel growth phase in freshwater and estuaries. But the silver eel migration stage that migrates downstream to the ocean, this may be the stage that's affected by climate change the most, because they might be affected by rainfall or changes in fresh water. And some indication of this comes from a fishery for tropical anguillid eels in Poso Lake that flows into Tomini Bay. And there's a set of weirs on the outflow there. And Dr. Sugiya analyzed catch data from these weirs and suggested seasonality of downstream migration. And so looking at the catch during 2001 and 2002, during August and September, the catches dropped off to nothing. And this was confirmed by a Barunajaya survey for leptocephali in Tomini Bay in 2002, showed that there was no spawning in September and October, confirming that. So if tropical silver eels may stop migrating in the dry season, such as periods of no rain from El Nino, this could disrupt the downstream migration of silver eels. And this could occur all across the Indonesian archipelago, where there's different patterns of seasonal rainfall and monsoon systems. To shift to the biodiversity of marine eel leptocephali, collaborative larval sampling between Japan and Indonesia has indicated that the highest biodiversity of marine eels occurs in the coral triangle. And this has been discovered from cruises on the Barunajaya 7 of LIPI in Indonesia and the Hakuomaro of Japan, where surveys were conducted around Sulawesi Island, off Sumatra, and a separate survey was conducted in the Sulu Sea and some of the surrounding areas. And this shows the IKMT being fished on the Barunajaya 7, where the plankton is sorted and leptocephali are pulled out. These are some anguillid larvae, but the vast majority are marine eel leptos. But for anguillid eels, these are the stations that have been sampled, and collections of small leptocephali revealed that there are local spawning areas in a variety of different areas. So these species do not conduct the long migrations typical of other temperate anguillid eels. But these surveys in the Indonesian seas are part of a larger sampling effort by Professor Shimoto and his colleagues to find freshwater eel spawning areas. And Indonesia was an important place to discover these short distance spawning migrations in collaboration with Indonesian scientists at LIPI. And of course, most of the leptocephali were of marine eel species. And so the study in conjunction with this conference will look at catch data of number of species of leptocephali worldwide, including the Atlantic Ocean and the Indo-Pacific, and compare to the Coral Triangle. And if we look at the number of species of leptocephali, it's pretty obvious that the surveys inside the Coral Triangle collected very high numbers of estimated number of species of leptocephali. All were above 100 species. And even just one survey in Tomini Bay collected more than 100 species. This is much higher than East Asia, the North Pacific, or Indian Ocean, South Pacific, and clearly higher than surveys in the Atlantic Ocean, except for one intensive survey in the Gulf of Mexico. But it's very clear because this Western North Atlantic total is the total number of eel species known from any type of research, including adults and everything. And this is lower than the number of estimated species just in the Sulu Sea cruise survey alone. So this suggests the highest biodiversity of marine eels, which is very poorly known, by the way, is probably in the Coral Triangle. And it's easy to understand why, with the structural complexity of the whole region and all the coral reefs shown by red here. But what type of marine eels might be affected? And one obvious thing to look at that we've heard about already is coral bleaching, because this can occur over wide areas, as this prediction map shows for the April-May 2016 event, which included possible coral bleaching in the Coral Triangle. And the Wusian et al. Paper documented that coral bleaching occurred in wide areas in 2010 event and 2016, because all of these white spots across the region were documented cases of coral bleaching. And so this coral bleaching could affect eels because there are obviously eels living in the coral reefs and surrounding areas. And some coral bleaching events can be totally devastating, which would change the community structure for a while. One type of eels that live in coral reefs are the moray eels. So they could be heavily affected when corals are dead, because predators obviously need prey. But there are many other habitats around coral reefs in the Coral Triangle, which include benthic habitats or slope habitats. And this is important because some species of marine eels are burrowers, so they need the soft sediment, and it's unclear how they would be affected by changes in community structure. Other burrowing species include the congridae, that include the garden eels, which actually live and feed in their burrows. So these garden eels, they feed on plankton that's drifting by. So it's quite possible they are not going to be affected by increases in water temperature unless zooplankton populations are also affected. Other marine eel species live on the slope and down deeper. So they may be too deep also to be directly affected very much. And the mesopelagic eels that live in the midwater, maybe they live too deep to be affected by surface temperature increases. So it's most likely that the effects on eels come from community structure changes from coral bleaching. And an interesting study on northern Sulawesi Island looked at 15 years of satellite images and discussed different habitat trajectories after disturbances, including sea level fall during El Nino or coral bleaching. So it's easy to imagine various trajectories of the habitat would directly affect what kind of marine eels can live in each of these habitats. And the final way that all marine eels might be affected, including anguillid eels, is it could affect their food source. And the food source of leptocephali was actually a long-term mystery, which is still a little bit not completely understood, but it appears that they feed on marine snow. And marine snow is present throughout the world's oceans, and it comes from particulate matter and exudates. So biological production, aggregation of particles all come together into marine snow. But most importantly is marine snow includes TEP, or transparent ectoparticles, that are exudated from phytoplankton and bacteria. But these TEP compounds include carbohydrates, which are simple sugars. And inside marine snow, this would probably be very excellent food component for leptocephali. So if there's not enough marine snow, or the marine snow condition is low, there could be early larval mortality and affect survival. So the eukaryotic phytoplankton are known to bloom during high nutrient conditions. And warm water is not good for high nutrient conditions and phytoplankton blooms. So warmer water could reduce the survival of leptocephali due to the quality or amount of their food. So I'll stop here and say that obviously there's a lot to be learned about eels and climate change in the Coral Triangle or anywhere in the world. And we almost don't know anything yet. But I'd like to give special thanks to Professor Shimoto and Dr. Wusian, who established this long-term collaboration to study leptocephali and eels in Indonesia. And I think it's been a really successful international collaboration. And obviously, international collaboration is good. So terima kasih. And I'd also like to thank for the use of various photographs of eels and coral reefs from the internet. And I finally say, let's all hope that life on Earth survives climate change. Thank you, and let's protect biodiversity. Thanks.
Thank you very much, Dr. Miller, for a very interesting presentation. And now I see your presentation generate some question already. So I would like to start the question answer session by reading the question that already in the chat. The first one is from Dr. Augie. What do you think regarding the diversity of marine eel and the Indonesian through flow and coral triangle? Do they have a strong relationship?
Well, I think it probably means that there's high number of species throughout the region, because water flowing through the Indonesian flow through into the Indian Ocean means that the leptocephalus, the eel species, in the Indian Ocean off Sumatra and Java are probably almost the same as they are farther north in the Coral Triangle. So what we don't know yet is if there's any regional differences in species. And I should point out that these numbers of leptocephali species, which are estimated from morphology, and Dr. Sugiya may be gathering information from DNA, but these are maybe higher than what are estimated for some groups from actual adult eels, because adult eels are very difficult to capture without using ichthyoside or things like that. And most species are not collected. So these currents, like the through flow, may be sort of mixed species throughout the region.
Okay. Now for the second question from the Dr. Eric. Do you think deep water eels be affected by oxygen shortage rather than by temperature rise?
Well, that's a good question. I know that some parts of the world have these oxygen low areas. I'm not familiar with if that occurs in the Indonesian seas or the Coral Triangle. And maybe there's no data about how tolerant or not deep water eels for that might be. But in general, eels are pretty tough creatures, and they might be less vulnerable than most types of fishes or other organisms. So I don't have any direct information about that, actually.
Thank you very much. We still have time, so I will move to the next question from Dr. Sam, Professor Sam Wardrijan. So how big the influence of climate change to marine snow, the food sources for both freshwater and marine eel larvae or leptocephali, especially around the Coral Triangle regions of Indonesia?
Well, that's the critical question for early larval survival, because imagine a big Anguilla marmorata may spawn like 50 million eggs at one time. So all those larvae in one place need food. And I couldn't include it in my presentation, but there is clear evidence that water temperature could have a huge effect on the production of marine snow. Because at the Bermuda Time Station series in the Sargasso Sea, they studied the alternation of eukaryotic phytoplankton with the cyanobacteria Prochlorococcus, which is present everywhere. And under warm water conditions, low nutrients, Prochlorococcus is a dominant phytoplankton species, and they probably do not produce much marine snow. But then when you have nutrient pulses of colder water, presumably, you have the eukaryotic phytoplankton blooming, and they would presumably produce marine snow in much greater quantities and qualities, which would be good for leptocephali. And this is hypothesized for the Atlantic eels, that this alternation between eukaryotic and prokaryotic phytoplankton could influence the levels of recruitment every year for the Atlantic eels in the North Atlantic. So warmer water temperatures may really reduce larval survival. But of course, some larvae would survive, but it could become a bottleneck for the abundance of recruitment, for particularly anguillid eels that would spawn a lot of eggs at one time.
Okay, thank you very much, Pramila, for the wonderful presentation and answer. And now, although we are still 5 minutes ahead of time, I would like to invite our next presenter, Dr. Davin Setiamarga from National Institute of Technology, Japan, and he will presenting about the DNA-based studies in natural history research of marine biodiversity. So, Dr. Davin, you might share your presentation and you might start.
Right. I'm going to share my slides. All right. Okay, I think you guys can see my slides, yeah?
Yes, very clearly.
All right. So thank you very much for the chance. Today I'm going to talk, I'm Davin Setiamarga. I'm originally an Indonesian, but I'm now a tenured faculty member at the National Institute of Technology, Wakayama College. So in the programs and abstracts, it's written only the first part of my affiliation, only National Institute of Technology. My full affiliation is actually National Institute of Technology, Wakayama College. There are about 51 colleges, 51 National Institute of Technologies in Japan, and I'm the one in Wakayama. So I'm going, it's very interesting actually to listen to the talk since today, because actually, of course, I didn't have any meetings with them. I didn't have any discussions before preparing my slides and my topics. But at the end of the day, everyone is actually talking about, well, very related things and emphasizing and putting forward similar take-home messages. So I'm going to start to talk first about natural history research using cutting-edge methods. So of course, the science of natural history is already very ancient. I'm taking pictures from, these pictures are all taken from the internet actually. It is all cave paintings. As we can see here, even before we have taxonomy or systematics as scientific fields, humans already, of course, by instinct, classified living organisms, because of course you have to classify if these certain animals will kill you, or if you can eat these certain animals, or if these certain animals will kill you if you eat it, and so on and so forth. So it's basic instincts of human actually. And of course, later on when it's become systematized as first, of course, being properly systematized and become a scientific field of classification. Usually we use a lot of different morphological characters or other physiological characters to differentiate to different living or to differentiate various living organisms. And then so basically we classify them into different groups. We put everything with similar characters into one group, and then we put those with different characters into different groups. So, but of course, natural history does not stop only at classification, but also studies the interaction among the organisms and the interactions between the organisms with their environment, and also, of course, how they are organized into populations and communities, and of course, all being studied at various different levels, from the molecular level to ecology and also morphology. And of course, the core, the heart of natural history is sample collection. You go to the field, you collect samples. These are actually some of the pictures, of course, I took from the internet, but this is actually I took by myself when I went to the field and collect my own samples. And of course, after you got the samples, then you start dissecting them, taking pictures, making sketches, and basically measure them, and so on and so forth. And then you voucher them in the and make them into collections in the museums. This is the collection in my own lab, actually. So yeah, so this is you voucher the samples, and it becomes an inventory of, or a record of the biodiversity you checked from a certain place in a certain time. But of course, there are problems. Number one is there's always an anthropogenic bias in biodiversity research and conservation. So of course, everybody has seen a lot of pictures about animal rights, about protecting animals, you know, with pictures of cute animals being abused and so on, like this very cute little kitten here. But I haven't seen any poster like this. But cockroach is a metazoan also. Why would you protect kittens but not cockroaches? So because of that, this is a paper by Trudet et al. In 2017. He checked the number of occurrences records being registered to GBIF, and then he found out that many animals are actually being overrepresented, like over here, the one with like avians, the birds, overrepresented, mammals also overrepresented. But insects, spiders, gastropods, the snails are very, very underrepresented. Well, one of the reasons is, of course, we have bias. So because of that, because even inside the data, because even inside the papers and records about biodiversity, most of the records sometimes only focus on a small subset of species. But why? Because those species are more likely to raise fundings, or are considered or are considered ecologically more important than others, or of course sexy, very very important organisms, or because we eat them. So nobody cares for everybody wants to do research about tuna, but nobody wants to do research about some tiny little small neglected snails in the middle of nowhere in an island somewhere. All right. So yeah, and then of course, although invertebrates are comprise 97% of animal diversity, research on invertebrates are usually very, very it's meager when compared to vertebrates and of course fishery species. And number two of the another problem is, of course, sometimes we have a very biased and skewed old practices in natural history research. When you sometimes people would go to the field and collect organisms every month, and collecting thousands and thousands of individuals from the field, and every single individual got pickled and put into bottles and then put inside the museums. It's of course it's conservation, but conservation of dead bodies. We are not conserving the living individual. So we're basically removing the individuals, the sample individuals, from the gene pools. And of course, sometimes, but of course, the problem is, of course, I do understand that some people would like to get new or fresh samples in order to understand what's going on. I mean, the most current or the most recent condition of the organisms. But also at the same time, people like me doing molecular work, sometimes it's very difficult for us to work using old pickled museum samples. And of course, recently, a lot of people really love to do molecular methods. I think Casey in the morning mentioned about taxonomy, morphology is still very important. But nowadays, everybody, rather than dissecting, other than doing taxonomy, a lot of people would like to do molecular works because, of course, it's practical and easy. So one of the new fad is DNA barcoding. Everybody, samples collected from the field. You sequence a part of their DNA, and then you use the sequence, the DNA sequence you obtain from the samples, and then you use it as a query to interrogate a database, and then to basically identify the sample based on the sequences. And of course, recently, of course, everybody really would like to love environmental DNA. So basically what you do is you don't have to collect the actual animals. What you need is only to collect the soil, the water. So basically the environmental matrices where the organisms live. However, of course, both problems need, both new methods, both DNA barcoding and environmental DNA, need database registered DNA data of properly identified samples. So if you don't have a properly curated exhaustive database, when you sequence something, even if you get the sequence and then you interrogate the database, you won't be able to ID your organisms collected from the field. So even environmental DNA, you still need the blast part where you have to identify the sequences by interrogating the sequences you obtain from the environmental samples to the gene data bank. The problem is, of course, BLAST is actually not developed for taxonomic identification. It's developed to identify an homologous sequence of the same gene from different organisms, not to identify the same different individual from the same species using a similar sequence. But of course, it can be used. The algorithm can be used for taxonomic identification, provided that the database is exhaustive enough for the taxonomic group you are working on. Now, this is an example. This is the picture of my niece about 2 or 3 years ago. I took it when I went back to Indonesia. So say we do DNA barcoding from her. We don't know what kind of organism it is. So we collected some samples and then we sequenced. And of course, the problem is, and then of course we blasted. We interrogate the DNA database. The problem is of course, whether there is a representative sequence of the correct taxon of my niece in the gene bank or not. If yes, then she might be identified as a human. But if no, then she would be identified as a chimpanzee. Probably it still looks similar, but still, she's not chimp. If there is no primate data on gene bank, probably she'll be identified as a mouse, as some mammals, other mammals. So when you do environmental DNA, for example, the problem with environmental DNA is that you don't collect the actual organisms. So you only get sequence data without ever seeing the actual organisms. So when you blast it to GenBank, and then there is not enough representative of the taxa you are looking, then you might get some unknown result saying that your animals are your organisms are unknown. But the problem is it really unknown, or is it really a novel species? It's probably just because there is no representative data on GenBank. This is a paper in 2009 from my academic advisor during my PhD. So they were one of the first to do environmental DNA for invertebrates using CO1 sequence. As you can see here, the sequence data will be further utilized by construction of dedicated database. So there was no, the database at that time was not exhaustive enough. In order to further clarifying sequences from unknown species. The problem is, of course, it's like 11 years already, and then we still do not have an exhaustive enough or unbiased database yet. So basically, then we have a vicious cycle here. When you do conventional methods of environmental monitoring, you need samples, and then you get invasive oversampling. Okay, then you do non-invasive molecular methods, but you still need samples because samples in the museums are not useful. So you ended up collecting samples again. Okay, so you do non-invasive molecular methods, like you just need to clip a part of the body from the individuals you collect from the field, and then you release them back, or you do environmental DNA. You need a reliable database. In order to build a reliable database, you have to do genetic data, which you collect from the field again. So it's just go on and on and on, because you will keep collecting animals from the field for the name of science, and then you basically removing the samples from the gene pools. So one of the an ongoing project I'm doing in my lab with various collaborators is to do museomics. Basically, we are trying to sequence properly curated vouchered samples in the museum. Although I mentioned that sometimes old museum samples are very difficult to work with molecularly, we are trying to use develop methods in order to build a reliable sequence database resource for future non-invasive environmental monitoring studies. So basically, what we do is we sequence museum collections, and then if of course, if possible, we would like to also try to sequence holotypes and paratypes, but usually it's very, very difficult, because most holotypes and paratypes, when they're collected like about 2 centuries ago or a century ago, they're all in formalin or already like in a very bad condition. And of course, because we use museum samples, we try not to get new samples. But of course, if you go to some places that is not yet explored, you ended up collecting new samples. But still, at the end of the day, we are trying to build a robust reference database, so that in the future you can just simply do environmental DNA and you don't have to actually do invasive environmental monitoring studies. While Indonesia, everybody has been saying the same thing. Indonesia is very diverse, marine biodiversity #1 in the world, and so on and so forth. And then we have, you see a lot of new species being identified in Indonesia during the last 10 years. It's difficult. For example, like some new mammal species, insects it's of course it's very understudied, so it's not really a big wonder if you find a new species of insects. But I mean, coelacanth or mammals, they are really so it's Indonesia is actually really a treasure box of biodiversity. So Indonesia needs to be more active. But in order to do this, Indonesia is a lost world. But in order to understand our biodiversity, Indonesia needs to be more active in doing basic natural history research in order to explore and catalog our own natural resources. And Indonesia needs its own natural history repositories and databases. We cannot, for example, in order to figure out what's going on about our own animals, we have to go like across the globe to find the samples, for example. We have to have our own collection in Indonesia. And this slide, actually, I took this slide when I was invited as the symposium Cendekiawan Kelas Dunia in 2017 or 2018. So apparently the government is trying to increase research, increase the TRL, technological readiness level of research. So here, of course, you have to do more applied research, and then you rather than publishing your results, you're expected to make products and so on and so forth. But I mean, all of my research, all related to molecular taxonomy and molecular evolution, all TRL 0, not even 1. But of course, because I'm doing fundamental, non-applicative basic research, it's called fundamental, it's called research dasar is called fundamental because it's the foundation of the other applied researches. If there is no foundation, it'll just topple over. It will just topple. And of course, the biggest problem with the common view is that basic and fundamental sciences won't make money, which is somehow true. But of course, it's a scientific research. It's not about making money. It's about making new discoveries. Everybody love hilirisasi in Indonesia. But this is, I always say this in my own dialect. Now nu hayang dihilirkan mun hulu nasaat. And of course, a lot of people mention that, oh, scientists only publish papers. Of course, it's our job to publish papers. It's not our job to do research and development for the industry. The industry cannot be lazy. They have to read our papers if they think they need our research result. It's completely outrageous if the government asks basic research scientists to not publish papers, but develop new variant of kerupuk udang, for example. It's not what basic science, that's not what taxonomy should do. So my rant about the policies ends here. I'm going to introduce some of the research going on in my lab at present. I'm now studying about this mangrove slug. Mangrove slugs collected, the Paramoyonchis tumidas, collected from Central Java, the northern coastal area, mangrove forest located in the northern coast of Central Java. So we are doing population genetics and molecular systematics. I'm doing this together with Dr. Madasibero in Diponegoro University, and then I do this research with IPB. So basically, we collected samples from the seas of Banda, from the Banda seas, and then we did DNA barcoding to see if there the presence of invasive species. This is quote unquote applicative research. So basically, I collaborate doing research with ITS and Atmajaya, isolating new bacterial species. Isolating bacteria with biomineralizing capability to be used in concrete development. So we did molecular phylogenetic analysis, we did XRD, we did full genome sequencing. And this is the research I'm doing together with the National Museum of Science in Tokyo, Kahaku in Tokyo. They have a lot of collections of Ceramaster japonicus and patagonicus collected from the seas of Japan. We found out that what was in Japan, there was thought that there were two species. We thought that there were two species of Ceramasters in Japan, but our molecular studies indicated that there was only one species. And this is also a research I do together with a lot of people. So basically, I'm sequencing full mitochondrial genomes of various octopuses, and then did molecular phylogenetics, and then do divergence time estimation. It's very interesting because we found out that most of the extant species of octopuses actually evolved during the Cretaceous period. This is the end of the Cretaceous period, when a meteorite hit Earth and dinosaurs went extinct. So, but also during the Cretaceous period, Gondwana land split into two subcontinents. So a lot of dynamic geological events that might explain why the octopsis radiated during the Cretaceous period. And I just published this in the preprint server in bioRxiv. So using proteomics and transcriptomics, we try to identify the core set of homologous protein domains being used to build the shell matrix proteins in conchiferan mollusks. Thank you very much. That's all.
Thank you very much, Dr. Davin. Now I would like to open Q&A session. If there is any question, please type in the chat and I will read it for you. Okay, if there isn't any question, I would like to move on to our next invited speaker, which is Dr. Didier Aurel from Mediterranean Institute of Oceanography, France. And the speech is about the possible adaptive responses to climate change in corals. So, Dr. Didier, are you here right now?
Yes, do you hear me?
Yes, I can hear you.
Okay, so I try to share my screen. Okay, is it okay? Do you see the slides?
Yes,
Yeah, I think
I
so.
can see it.
Okay, thank you. So thank you very much for giving me the opportunity to talk today. So I will do a kind of mix of results and synthesis opinions on the possible adaptive response to climate change in corals and the importance of specific and genetic diversity. So I will go quite fast on the introduction slides because there have been already very interesting talks in this symposium. So we already talked about a lot about corals and climate change. So what we mean about corals and what I will talk about today is cnidarian belonging to hexacorals, clearactinian, which are of course engineered species. And as we have seen, tropical coral reefs are support of high biodiversity with ecosystem services such as fisheries, coastal protection, agriculture, and so on. The context about what I will talk today is climate change. So we already talked about this today with anthropogenic drivers of climate change, such as greenhouse gases, which induce warming. And I want to underline here the importance of extreme events in warming and also acidification. So just here you have, I took just these two pictures of the one IPCC report, which shows the inferred and projected trends in warming in the Pacific and Indian Oceans. I will not detail it, but we expect the ocean to go warmer with more extreme events as well. So one consequence for corals of warming is bleaching events. So this corresponds to the disruption of symbiosis. A lot of corals live in symbiosis with zooxanthellate algae, and this disruption can have very negative consequences for corals themselves and for coral reefs. So we had already examples of bleaching events today, sorry. I said this morning, but it's morning for France. You can see this map for the mass bleaching event of 2015-16, where the red dots indicate locations where you had important bleaching and blue dots where you had no substantial bleaching. So it's really an important phenomenon worldwide and with differences depending on location. But we also observe differences among species to the response to bleaching. So here, for example, this publication by Terry Hughes, one of his famous publications about the bleaching in the Great Barrier Reefs in 2016, and what they observed there, it was that you had a more important effect, more bleaching for fast-growing, branching tabular species compared to more massive species, for example. So I don't detail the picture on the right, which shows that the more it is, the higher you have a shift in the composition of coral communities. So So it's important to, when you study the response of the coral reef to climate change and to bleaching, to take into account that not all species will respond in the same way and to identify species. So of course, it's quite obvious that you can differentiate fast-growing branching species from massive species, so it's not the problem. But in some group of corals, species definition and identification can be a problem. So what I will talk more about today is the case of the Pocillopora species complex. So for Pocillopora corals, based on morphology, there has been around 17 species which have been defined. So they have been defined based on morphology, macro or micromorphology, but when genetic analysis began on these species, the picture became quite more complicated. And I will refer it here to the article by Pauline Gélin, Hélène Magalon, and their colleagues, which was published in 2017, where they used two kinds of genetic markers to study these species. First, they used the mitochondrial DNA with a locus named ORF, and the differences in sequences among corals with this marker allowed to define what can be called primary species hypothesis. So we can see different group of sequences, and on the basis of this group of sequences, you have a first definition or first hypothesis of species limits. Then, as this is a single marker, we can try to test this species hypothesis with other markers, independent markers. Here it was microsatellites, and they're allowed to define secondary species hypothesis, which can be coherent or not with the first primary species hypothesis. Based on the combination of these two information, Pauline Gélin and their colleagues proposed at least 18 secondary species hypotheses among 14 morphotypes which were used in their dataset. So there are two phenomena which can be invoked to explain the differences between morphology and genetics. First, it is well known that you have morphological plasticity. That means that a given species can have different morphology. And second, you have cryptic lineages, which means that for a similar morphology, you can have different species. I want to illustrate this with preliminary results in Indonesia. It comes from the Lenguru project and the Lenguru expedition, which took place in northern Papua with colleagues from LIPI. And so here you have the main location of the Lenguru area. And here, just to give you an idea of sampling sites, I won't detail sampling sites today, it's very preliminary. And we collected, in fact, colleagues from LIPI collected some corals in this area, and we had a focus on the Pocillopora species complex in this area. So we worked with our colleagues from LIPI, Muhammad Abra and Rizky Satria, who came in Marseille to work with us on the genetic of Pocillopora, but also two corals. And regarding Pocillopora, we sequenced the mitochondrial mitochondrial locus ORF, sorry. And here you have a tree of the sequence to obtain. So in red, these are reference sequences from GenBank, which include the sequences published by Pauline Gélin, the previous article, and in black, the new sequence we got from Lenguru. So you see, for example, here under PSH5 that you have sequences from Lenguru, which are identical to previously published sequences in GenBank. So we can use the previous sequences to propose some species identification in Lenguru. You can see that you have several groups of sequences which correspond to different species hypotheses. These species hypotheses had been defined previously again. And in Lenguru, for what we have analyzed already, sorry, we have at least 7 species which are present, which are the species hypotheses indicated in blue here. What we are working now is the comparison with morphology, and Abba is working on the morphology, and we will integrate all these results. But we have already an interesting diversity of Poecilopora corals in this area, and what is very interesting for us is that it's in sympatry, and so we can study if there can be some hybridization in this area. And just to mention, so it's very small, so it's not easy to see, but we have a new mitochondrial lineage, which is a sequence we never observed before, a little bit different from other ones, and that's an interesting topic, objective to study if it could be a different species. So coming back to climate change, why is it important to study species type? I will present some results from a project we had with F. Toulza and Guillaume Mita in Perpignan about the response to thermal stress in Poecilopora, and where we compared Poecilopora colonies coming from New Caledonia, where temperature is more stable, and Oman, where temperature is much more variable. So colonies were collected and submitted to experimental stress in aquarium, and we studied the response of these corals to thermal stress with the sequencing of transcriptomes, I mean all expressed genes, just before compromised F. And we checked if our colony belonged to a given species type based on mitochondrial ORF sequencing, just as before. So here is one result we got. So it's not maybe not easy to analyze. So each line here corresponds to one sample, one colony, which was submitted to stress or control. And we compare with this matrix the response of the different colonies. So here, when you have dark blue, it means that the response is very similar. So on the diagonal, it's the comparison of a colony with itself, so it's the same response, of course. But if it comes more light and even white, it means that you have a very different response. And I mean response, it's the transcriptome, the whole expression of a different genes. So we have samples from New Caledonia up and down, samples from Oman. In New Caledonia, we thought we had sampled the same species, but in fact, with the same morphology, we got two different species hypotheses, 4 and 5. And in Oman, which is maybe not very surprising, we had a different species, which was PSH 12. What we observe is that in New Caledonia, so in the same site, we have two different species with different response to thermal stress. For example, if you see the samples from New Caledonia from species hypothesis 5 here, they have a very correlated response, which is very different from the response of species hypothesis 4, and which is, of course, different from the response in Oman. So we have a difference of response for close species in sympatry. And also we observe that there is a difference between Oman and New Caledonia, which was expected because here we can have two effects, species effects and an effect on the origin of colonies with maybe an acclimatization of colony to their environment. And also I don't develop this here, but what we observed, which is very interesting, at least to understand the adaptation of these corals, is that we had a higher plasticity in Oman for gene expression in a situation of thermal stress. We can go further on these topics. Just to present the preliminary results we have working on samples from Oman, because in Oman we had in fact more samples than that and more mitochondrial types. So we can test if when you have different mitochondrial sequences, they correspond to different species with new kind of marker. Here we used what we call RAD sequencing, which produce a lot of loci, which are called single nucleotide polymorphisms, SNPs. And here we got more than 130,000 SNPs to study the differences among these mitochondrial lineages. And here you have a tree where each branch corresponds to one coral posilopora sample, and the color corresponds to the mitochondrial type. And what we get here is that at least for these mitochondrial types, which are quite similar on the tree, we can see that with SNPs, we don't separate them on the basis of this mitochondrial lineage. So here, the differences on mitochondrial DNA do not seem to indicate that these are different species. And it would be very interesting to apply these kind of methods to the diversity of posilopora samples we observe in Indonesia, and in particular in Indo. So again, what about climate change? So there is a diversity of response among species, I've shown it, but there is also a diversity of response among population and among, I mean, population within species and among individuals within population. So there has been already previous works showing that the effect of a previous stress can influence the response to new stress, but there can be also a question about the possibility of genetic basis for differences in thermotolerance in corals. This is very important because if you have genetic differences in the response to thermal stress and to bleaching, this could be the basis for a future adaptation to climate change. So we have now the tools to do such genomic studies of adaptation to thermal condition, and here we can do to local adaptation to local thermal regime. And we can use high throughput sequences. For example, we can detect SNPs in with different techniques. We can detect SNPs in transcriptome with RAD sequencing, as I've shown, or in whole genome sequencing. And then we can use this data to test if we can see a correlation between a genetic type, a genetic variant, and some environmental conditions, such as temperature, if we compare populations which have with different thermal regimes. So I don't want to detail this today, but there is already different work who have shown that we can detect local adaptation in corals. And there are signals of local adaptation to thermal conditions in different species and populations. Just to mention that when we do this, we have to be careful about false positives. And this shows that there is adaptive diversity in coral population, which I would say it's quite obvious we would find this. But what is interesting is that we can maybe identify populations which are more linked to thermal conditions than other ones. So there is a possibility of adaptation. Maybe it's quite obvious as well, but it will depend on several parameters: the rate of change versus climate change versus the maximum possible rate of adaptation for corals. And other parameters who will influence this is the connectivity among population and possibly other pressures which will impact these coral population. And what can we do with this? So for me, as I'm a population geneticist, I'm not an ecologist really. So my main interest is that it's a very interesting case study for the evolution of adaptation in marine environment. I mean, the marine environment is very open, and we can here have a very interesting model study of how a given species can adapt to different thermal conditions in the sea. But there are also some other proposals which have been proposed these last years based on these genomic studies to use this for the management of coral populations. So I will just mention two articles. This first one by Palubi proposed a quite simple model, maybe not so simple in fact, about assisted migration, where they propose that if you transplant some coral colonies from one thermotolerant population to a less thermotolerant, you can help the evolution of a less thermotolerant population. And this could be used in management. And there are even some more, I would say, extreme proposals, such as proposals for the assisted evolution of corals, or even direct genetic modification. A very recent article who used CRISPR to test if it's possible to modify corals genetically. I won't go further on this, but it raises a lot of questions about the spatial scale and what will be the importance of these techniques if we still have warming. And from a personal point of view, for me, the most important is to work on limiting warming in the next decades. So as a conclusion, species and population diversity are very important in the response to climate change. And we have a very interesting case study in Posilopora corals, and where we can use genomic approaches to first study species limits and to study adaptive diversity. And what I would say is that the Indonesian archipelago is an important area for to study coral evolution, because as we've heard already today, it's at the heart of the coral triangle. And it's a contact zone between lineages, and we see also this in Posilopora. And we also have different environments where we could implement similar studies to what I've shown for a study of local adaptation in coral. So I think my time is done, is over, sorry. And just to mention, it's a collaborative work, and here several names of people who worked on this during the Lenguru expedition and during the work lab we made after that. Thank you very much, Terry McAfee.
Thank you very much. It is very interesting presentation. Now we still have plenty of time for question and answer session. If you have any questions, please type in the chat in Zoom, and I will read it for you. If there seems to not no question yet, so probably I would like to ask a question or two.
Yes.
So I'm quite interested in one of your research on that you said that coral reef in Oman has a higher plasticity against climate change. Could you explain what type of adaptation that they have, so they seem to be able to handle climate change stress much better than the others? And can you also detect that type of adaptation in their genomic data?
So yes, in Oman, I don't have all the details, but briefly, what we observe is that if we submit the colonies of Posilopora in Oman to thermal stress, we can see that there is a variation in the expression of genes, and this variation is much more important than in New Caledonia. That means that they are able to change their gene expression faster and on a more important way than in New Caledonia. That's the first kind of observation. And the second kind of observation is that we have some what we call front-loaded genes. That means that we have some genes which can which are important in the response to thermal stress, which are expressed before the thermal stress, like a kind of what we could call pre-adaptation. So I don't have in mind all the genomic functions which are involved in this response, but I put at the end of my presentation a list of references, and you can find the article by Brenner and the collaborators where you can find the molecular functions. Oh, sorry, I don't hear you. I see that there is a question.
I forgot to unmute.
Yeah.
Okay, thank you. So there's another question from Dr. Eric. You mentioned that adaptation might compensate temperature rise, but is there a threshold beyond which adaptive responses will become impossible?
Bonjour, Eric. So yes, it's a very important question, and I guess there is a lot of uncertainties, a lot of things we don't know, and this is one of the main things, I guess, to know if even if we have genetic diversity and genetic diversity in the response to climate change, up to which level the species can adapt is unknown, I guess. For me, it's really unknown. That's why I think it's there are two messages, I guess. The first is we have to keep the most diversity we can because it's kind of insurance facing climate change. And the second main thing for me, as I said in my talk, is to really work to limit warming as much as possible. But the threshold, I don't know, and it may depend on the species and the population.
Okay, thank you very much. We still have time for one more question. It's from Professor Sam. He said that he asked the same question before, but he thinks that the coral bleaching also the main factor in degradation of the coral in Indonesia, such as in El Nino event in 1983, 1997, 2010, and 2016. So his question is, how can we overcome these problems to protect the corals in Indonesia?
Yes, so I'm not an ecologist, so I'm not really the best to answer how we can protect, but I would say it's a little bit the same answer than before to Eric. I would say that from the point of view of population genetics, is that we should protect diversity as much as we can. So that's one answer. The second point, which is quite general, is that I mentioned the fact that there is not only bleaching, climate change, which can impact corals, and we see it in the Mediterranean as well, is that you also have overpressure. And if we can limit overpressure, such as destruction, direct destruction, pollution, and so on, it's important as well. And again, my last message is let's work on limiting warming. Oh, I see a very interesting question.
Yes, thank you very much. We still have a little bit more time because this question sounds interesting. So I would like you to answer it. So, is it from Dr. Devin? Is it adaptation or is it acclimatization? Is the ability to withstand temperature rise already present genetically, or is the selection process still going on? This is quite interesting.
Yes, this is very interesting, and it's the main question and one of the most important questions for the experiment with Oman and New Caledonia. You're right, you're completely right. So the answer is not easy. For what I've shown for the comparison between Oman and New Caledonia, it can be both. It can be genetic adaptation or acclimation, right? People from Perpignan, Guillaume Mita, Jérémy Vidal-Dupiol are working to study acclimatation with, for example, the study of epigenetics. That's the first answer. Second point is that for the studies of local adaptation, what I've shown in the last part with literature, we study the genetic differences which are linked to different environments. So we are at the genetic level, and it's really here genetic adaptation. But you're completely right. In fact, when we talk about adaptation to a given environment, probably there is a mix of genetic adaptation and acclimatation.
Okay, thank you very much, Dr. Didier. And I would like to move on to our next invited speakers, Dr. Nina Yasuda from University of Miyazaki, Japan, and her talk will be about the genetic structure of the threatened coral species in the northernmost territorial habitat, the potential refugia in relation to climate change. The time and the stage is yours, Dr. Nina. You might share your slide.
Thank you very much. I would like to share my screen first. Okay. Thank you very much for introduction. Today, I would like to talk about some marginal coral habitat where it is very far from Indonesia, but very, very interesting phenomena is occurring in this area. So I'd like to show our data to get some knowledge. So as you already know, corals are threatened due to elevated water temperature. So the most of the corals in Indonesia is suffering from the bleaching event, coral bleaching event. It is caused by elevated water temperature, like this. So then, what is going on in the northernmost edge of the coral species range? Because Japan is situated further north from this area, so we have a much colder water temperature. So in this region, the sea surface water temperature has been increasing, of course, due to the climate change. And along Kuroshio Current, which is the south of Japan, which is started from the Philippines, the water temperature has risen by 1.21 by 100 years, which is more than twice than the world average. So there is a rapid increase of water temperature in this region. So what we have observed in the last 30 years is something like this. This is an example of Yokonami area, Shikoku in Japan, as you can see here. So in the early 1990s, there has plenty of sea algae bed, sea algae and seaweed bed like this. However, in 1998 and 1997, there has been a dramatic decrease of this population. And then in 2000, it looks like almost desert and nothing has been left. It's associated with climate change. And then what happened in 2013 and now is that there's plenty of corals colonized in this area. So the sea algae has been completely changed and replaced by the corals, and corals are expanding in this region. This is not actually in that economic area. So many of the temperate area in Japan now we can observe this kind of situation. And if we focus on individual species like Acropora hyacinthus, that in Indonesia you also have this species in your country. So distribution of the species has been expanded since 1980s like this. So the northernmost area has been Wakayama. However, in 2000, we could observe further north in the western area. In 2016, we could observe the species up to here. So apparently, the corals are migrating to north associated with climate change. So this is a paper introduced in Nature, the corals marching to the pole. So it looks like corals are escaping to north and expanding their population. So this fact gave us some idea that if this kind of temperate area in Japan can be used as a refugia to protect or preserve some coral species. So here I would like to use the word refugia as defined by Kepler et al. 2012. Refugia is habitats that's components of biodiversity that reach to persist in and can potentially expand from under changing environment condition and facilitates the persistence of the species under anthropogenic climate change. So we are interested in if the temperate area in Japan can be used as a refugia, coral refugia in face of climate change. So I would like to explain what is a mechanism that can cause this kind of coral distribution expansion. So this is the figure that shows the expansion mechanism. So on the left-hand side, you can see some pre-existing coral communities and crucial and other current system, associated current system, where disperse, transport a coral larvae. And the alive larvae in the temperate area could survive because elevated water temperature, and then they can propagate. So basically, larval dispersal may mediate the coral expansion and extension of the species. However, when we think about the refugia, we should be careful because it is possible that only a small amount of larvae will be able to alive and adapt to the northern habitat. So it is possible that they have lower genetic diversity in such temperate habitats. So in this case, if there is some slight change of environment conditions, they would easily locally extinct. So it does not offer, it cannot offer any safety refugia in this area. So I would like to explain a little bit about the importance of genetic diversity. What will happen if genetic diversity is high is that even if there's genetic environmental change, some of them can survive and recover again, replenish again. However, there is a low genetic diversity, then they can easily locally extinct because if all the genotypes are weak against the heat or cold stress, then they cannot survive. So genetic diversity reflects the vulnerability of the population. So it is also said that marginal population has lower genetic diversity. So the aim of the study is to examine the potential role of temperate coral habitats as refugia in era of climate change. So if we can save some coral species for example, in Indonesia, can be saved or survive in those kind of colder area where there's no effect of coral bleaching. So we examined genetic diversity, gene flow of corals, including recently colonized, recently appeared population. So first, we tried to reveal in the case of Acropora hyacinthus, which is abundant and dominant corals, both in temperate and subtropical areas. We collected more than 1,000 samples from all over Japan like this. And yellow circle means that they are recently colonized population. So those kind of population has never been observed before 1980s, but recently propagating. And this is a result. This study has been conducted using microsatellites. We actually found at least 3 hidden species, which is not detectable by morphology. And on the bottom, you can see on the bottom left, you can see its distribution of the 3 lineages. So as you can see, the lineages showing green is predominated in temperate areas and expanding to north. And we still don't know whether the red and blue lineages is also migrating north. So we need some monitoring to monitoring if these kind of different lineages are also migrating north. However, what we observed in temperate area is green lineages. So we focus on green lineages in this study. So we also employed some oceanographic modeling, which is directly test if the larvae of corals can migrate to different area between different areas. So we calculated 27 years oceanographic data, and we released a small particle, LIPI, from different sites. So on the right-hand side, you can see the result of the connectivity matrix. So x-axis means sink population, and y-axis means source population. And on the as you can see in the red square, this this square means the direct migration from subtropical area to temperate area. So there are some possibility that some of the subtropical corals, corals can be migrated to temperate area, but the possibility to reach is much limited than within the subtropical area. So direct dispersal from subtropical to temperate is more limited, physically more limited than we expected. However, it is also still possible. So this is a result from the assignment test based on the genetic data. And we could observe that the larvae are gradually migrating to north. So as you can see, the pre-existing temperate coral population is important source for the recently migrated population. And this is a result of the genetic diversity. We could observe very interesting result from this data. On the left-hand side, you can see the subtropical population. On the middle, you can see pre-existing temperate population. And right-hand side, you can see recently colonized population. As you can see, the genetic diversity is gradually decreased as we go to the north. And recently colonized population always has much lower genetic diversity than the pre-existing temperate area. So which means that recently colonized population is more vulnerable to environmental change. So they can easily extinct at this stage. So how about other coral species? We also examined two other dominant coral species in temperate areas. One is Acropora solitariensis, and another is Acropora cf. Both two species are also dominant in temperate area in Japan, as well as you can see in Indonesia. So the distribution of these two species is like this, and the distribution has been relatively limited before 1932, but in 1955 to 1977, people became observed that these two populations in western side of Japan, like this, and in 1980 to 1995, we could observe new population in Shikin area. And now we can see further north like this. So we sampled from different areas, including Taiwan, which is outside of Japan, to compare the genetic diversity. In this study, instead of using microsatellite, we actually also use microsatellite, but we also use some novel population genomic methods, which is called MIGSEC. This is a genome-wide SNP analysis, but very, very easy and cost-effective. We will employ 2 PCR and then you use some MiSeq and get a lot of data from out of that. So genetic diversity was actually, this is the result, and genetic diversity of the corals was unexpectedly comparable. So even in the northernmost site, like Tateyama area and Goto area, so I'm sorry, it's still in Japanese, but the northern habitats still have relatively high genetic diversity, even comparing to Taiwan. So there is no decrease in this species. And the another species also have relatively comparable amount of genetic diversity in the northern habitat. So it actually means that not always the northernmost habitats are vulnerable to environmental change. So we also examined some composition of symbiotic zooxanthellae genotypes, which is deeply related to the survival and environmental adaptation. So we use the ITS2 data from the northern habitat as well as the other temperate areas. So this is a result. So the blue lineage showing the northernmost site. So and the others are showing yellow and orange. Because we examined the two species, we showed in different color, but both two species has distinct genotype of symbiont only in the northernmost site, which is very, very weird result. So if we show on the map, the distribution is something like this. So as you can see in Tateyama area and the Goto area, the genetic genotype of the symbiont composition is completely different from other side, even though the genetic structure of the host corals are comparable to the different northernmost and other areas. So this is like this. Only two northernmost sites have distinct symbiont genotypes. So there might be some special adaptation to the coldest environment, or also it is possible that the northward migration of symbiont species is lagging behind. So there's still not enough symbiont of the orange and yellow type in the northernmost site. So we are still investigating the seawater samples of these areas. So this is a prediction of the coral cover increase in temperate area in Japan, which is used as a statistical model. So after examining the correlationship with the water temperature and other many environmental factors, there was a strong correlation with the coral area and the lowest water temperature during the winter months. So this is like this. So based on this data, we predicted the increase of the coral area in this in Japan. It's something like this. As you can see, the red part that is expected to increase coral area in the future. So what do these data tell us about the potential role of temperate coral habitats as a refuge in an era of climate change? So we think not all corals can escape to the north. It is very reasonable. But some coral species have lower genetic diversity and distinct symbiont composition in the recently migrated populations. So in terms of conservation of corals in face of climate change, pre-existing temperate habitats may play an important role, even as a source area, as well as the relatively stable habitats for corals. But of course, not all species can be saved in those areas. Of course, there are so many some endemic and highly diverse corals in the south, like in Indonesia. There are plenty of very important species. So not all species can be conserved, but some of the species can be preserved in these areas. So, and also, if we take it, look at the viewpoint of evolutionary novelty, the source of evolutionary importance as a origin of diversification and speciation, the current northernmost habitats may be of high value because they have distinct, they're using distinct symbiont, and they are surviving with a very low genetic diversity, which may be threatened by local extinction, and try to adapt in a very rapid way. So monitoring this site along the climate change would be would give us some new insight about the evolution of corals. And I would like to thank for everyone, and especially for Dr. Hagiuria for inviting me to speak here today. And I would like to thank for all the funding and the lab members who contribute to this work. Thank you very much.
Thank you very much, Dr. Nina. That is quite an interesting presentation. Now we will open the question and answer session. We already have one question here in the chat. From Dr. Willem, do you think that with continued warming, there is a northern limit to the distribution of corals in Japan?
Yes, it's a very interesting question. I think yes. I think the Tateyama area, which I show as a northernmost site, would be the one of the limit, because after that, further north, there is a cold, strong current from the north, which is called Oyashio. So even with the global warming, I think for corals still have difficulty to migrate to the north. And I think it is also, I didn't speak about anything about ocean acidification in this slide, but after beyond the 2050, it is possible there will be some effect of coral acidification from the north. So northern part has more easily affected by the ocean acidification. So I think Tateyama area or something like around that area would be the one of the limit of the corals. But still, other areas have expansion of corals in temperate area in Japan. Thank you very much.
Thank you for your answer, and we still have time, so I will read one more question from Dr. Didier. He asked about, what about the clonal reproduction in newly colonized populations?
That is also a very interesting question. Thank you very much. Actually, for the 3 species we examined, we could not observe the higher rate of asexual reproduction, although we still show some asexual reproduction in this species, because it probably because we worked on the table corals. I only show table corals in this presentation. We also examined Acropora purinosa, which is the branching type coral, Acropora corals, and we observe a really high rate of asexual reproduction all over Japan. So I think the morphology is more important. It has more effect on the asexual reproduction rate in the temperate area. Thank you very much. Rather than the newly colonized or not.
Thank you very much. We actually have a little bit time, so if anyone still have a question, we can still open one more question. Okay. If not, thank you very much, Dr. Nina, for presenting such an interesting.
Terima kasih.
Yeah, sama-sama. So I would like to move to our next session. That's the last of the invited speaker session. Now We still have 2 more oral presenters. For the first presenters, it will be Ms. Rianta Pratiwi from Research Center of Oceanography, Indonesian Institute of Science. Her presentation is about the ecological studies of shrimp Venus monodon fabricius in the Segara Anakan Cilacap, Central Java, Indonesia. For Ms. Rianta, the time and space is yours. You may share your slides now.
Thank you, Arif. Wait for a while. I will share my screen first. Can you see?
Yes, we can see it.
Everybody? Okay. Thank you very much. First of all, I wish you Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. In this occasion, my name is Rianta Pratiwi. In this occasion, I will talk about my study with the title, The Ecological Studies of Shrimp Venus monodon fabricius 1789-88, Penide in the Segara Anakan Cilacap, Central Java, Indonesia. I'm sorry. Segara Anakan Lagoon, or SAL, is the large lagoon located along the southern coast of Java, Cilacap district, Indonesia, fringed by the only large estuarine mangrove forest left in Java. SAL is a semi-enclosed lagoon, which consists of the central lagoon as primary and bounded by mangrove forest and tidal land or intertidal, which provides important roles for fisheries. The lagoon is of high ecological and economical value because of its richness and diversity in living natural resources, comprising commercial important fish, crab, shrimp, and mangrove species. During the last decades, the coastal habitats have changed in these regions as a result from increased urbanization and expansion of agriculture and aquaculture activities, leading to problems such as rural encroachment on agricultural land, reclamation from swamps, seed deposition in the lagoons, and a decreased fisheries catches. The Venus monodon is the important commercial shrimp from SAL and other species of Indonesian waters. The time SAL is facing some ecological problems, sedimentation, illegal logging, a high rate exploitation, the degradation of the mangrove forest, and areas have been converted into shrimp ponds. Venus monodon have been taken seasonally to enable some of the basic morphological characteristics of the species to be measured and for the habitats requirement and local geographic range to be documented. The present study forms a part of a wider program of the management of SAL and its mangrove conducted by BPKSA, Cilacap, and our interest on the commercial marine species in the SAL, for example, Venus monodon, have long been studies. I'm sorry. The aims is the first to examine the effects of environmental variables, rainfall, water, temperature, salinity, pH, DO, nutrient, and turbidity on the adult more than 25 mm Venus monodon, and the second to combine this result with information of the immigration of juvenile CL3 up to 25 mm as a mean of interpreting changes observed in the structure of the juvenile shrimp population throughout the year. This 12 months, we assigned from December 2060 up to January 2070, ecology studies of the shrimp Venus monodon was done in 4 zones of Segara Anakan Lagoon. This area of the present study, which is divided into 4 zones, included the Segara Anakan Lagoon, the river Ciberem, Citandui, Kalimantan, Muara II, dan Karanganyar in the northern part of Legan. 1 year to examine its population structure, growth, and morphometric relationship. Sampling procedures for serum penis monodon cuts by Ancho is a traditional station method. The morphological relationships in terms of carapace length, CL in millimeter, and total length, or TL in millimeter, of male and female shrimp, which were significantly correlated at this level of significance. The number and size shrimp cuts on every month depended on location in the south, state of tide, and depth of water. The profile results, which were representative of the total population, monthly changes in population structure were examined by taking the sum total of all shrimp cuts over all stations. There is 53 stations. The growth in weight was subsequently calculated from the CL weight relationship. This is the analysis data we use to determine the CL and TL relationship. Straight line were fitted to the raw data for the female and female, and you can see that the equation and the related carapace length to weight of shrimp. We use the allometric equation. This is the location. We have 4 zones. This is the first 2, and then zone 3, and the 4 zones. For the result, morphological relationship, measurements were taken of 6,854 individuals. That consists of male is 3,377, and female 2,384 captured in the south from December 2060 to November 2070. To determine the CL and TL relationship, straight line were fitted to the raw data for males and females separately using the method of least squares. This is the straight line is for male, and the dashed line is for female. The relationship for male and female were found to be represented by equation. That's a female with the TL equal 15,825 plus 3,225 CL, and the male TL 25,462 plus 2,897. To relate the carapace length to weight of the shrimp, the allometric equation of the W equal E CLB were fitted to the raw data using log regression. This is the curve that for W 10 minus 3, it's for male, and then W 10 minus 3, 1,050. 520 is for male. The male and female penis monodon, the equation giving the best fit to the data were found to be like that I said. And the female 31.15 plus minus 2.03 millimeter in the sample had a large mean CL than the males. The males is 30.11 plus minus 1.46 millimeter, indicating a faster growth by females. The males were slightly but significantly greater in both total length and weight than female based on intercept. This is due to the morphological feature of the bodies. The size of adult, differences in the size of adult shrimps, CL more than 25 millimeter, Therefore, reflects change in both sides of stream present in the salt at the time immigration and the strength of the stimulus to migrate. A large member of relative small size stream, mean CL with ranging from 28,606 plus 3,310 millimeter in December 2060 to February 2017, it to be 30,670 plus 3,514 millimeter. It's found to be abundant in the salt during the wet season of 2060 up to 2017. It is suggested rainfall was very important factor for migration of adult Venus monodon. Population structures, as shown in figure 4, Venus monodon were present throughout the year. The population consists of small number of large individuals from July to December. There is 554 individuals, and from February to April to 110 individuals. The size distribution at this time of study range from 17.10 millimeter to 59.00 millimeter CL. All belong to sub-adult and adult stream categories. The habitat, the interesting record of Venus monodon from salt had been collected from a depth of 5 millimeter in Majijing Rock, an area close to Indian Ocean by Nusawiri. A number of juvenile banana stream, merguensis, Venus merguensis, another mangrove-dependent species, were also captured, suggesting that juvenile of Venus Peneidae stream species, primarily Venus monodon and Venus merguensis, are common in salt mangrove. Since the lagoons receive considerable sediment particles from upland, the stream capture appears likely to vary in size. This may have resulted from local movement of adult, juvenile, and pelagic larvae of both stream species, Venus monodon and Venus merguensis, due to the turbid water with clay particles. Influence of habitat, the stream Venus monodon is also borrower, but borrowing activity is size dependent and increase as the animal grow. The table 2 shown the stream of Venus monodon occurs in both habitats. Other streams were found varying number, and although large catches were occasionally taken in water of 10 up to 50 meter depth, streams generally favor the soft mud or silty clay. The density of stream in the second type of habitat was considered consistently lower, implying the habitat preference selected by adult stream, Venus monodon substrate immediately in front of mangrove rings. This is the table that seen the distribution of adult Venus monodon into different habitat types in the salt lagoons. This from months December 2060 up to November 2070. This is the substrate, silty clay substrate and sandy clay substrate. There's the individual, it's a total individual, and this is the measurement of CL in millimeter Venus monodon with habit in clay, silty clay substrate and sandy clay substrate. The conclusion, mangrove habitats of shrimp population are tightly linked in many regions as it was in salt. Analysis of commercial shrimp catches have shown that the salt support of a small permeate shrimp fisheries, the main species scouts being, in order of important, Venus monodon, Venus merguensis, and Venus hemisulcatus. The surrounding mangrove areas in the salt indicated that the areas being a nursery habitat for juvenile Venus monodon, more than the measured 3 up to 25 millimeter CL. Solid evidence still recommended to be investigated and failed to be studied in detail, although intuition and limited correlation result point to an existing relationship between mangrove forest and fisheries. Pennes monodons have different preference of tolerance to sandy clay and silty clay substrate. Here's a part from stream abundance and reproductive success, recruitment of cross larvae, growth and survival of all the historic state of stream Pennes monodon is suggested for further investigation. I think that's all. And wassalamualaikum warahmatullahi wabarakatuh. Thank you.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Ms. Yanta, for interesting presentation. So I would like to, we still have about 2 to 5 minutes left. If there's any question, please type in the chat column of the Zoom meeting. If there isn't any, may I ask one question? I see I think I read in one of your slides there is that rainfall is important for the migration of adult Pennes monodon. Could you explain a little bit why the rainfall is important? Is it because a lot of waters will facilitate the movement, or is they prefer to move or migrate during low salinity or something like that?
Yes, there's a migration from Pennes monodon because there is many rivers in surrounding the Segara Anakan Legend. Then also during the during the high rainfall, every month is more than 100 millimeter rainfall. That's why Pennes monodon, it can flush the larvae with the rainfalls. That I think that's my answer. Right, Arif.
Okay, thank you very much. If there is any question? If there is no question, thank you very much, Ibu Deanta. I would like to move on to our last but not least oral presenter for today, Mr. Wawan Kosasih from Research Unit for Clean Technology, Indonesian Institute of Science in Indonesia. His topic will be scaling up of the enrichment of omega-3 content of lemuru fish oil by enzymatic reaction. Mr. Wawan, the time is yours. You might share your slide now. Mr. Wawan, can you hear me?
Yes. Can you hear me?
Yes. You can start to share your screen.
Okay. I tried my slide.
Yeah, can you
can see my slide?
No, we cannot. I cannot see your slide. Mr. Wawan.
Yes. Can you see my slide?
No, it's still in my computer screen. The slide is not yet on our screen.
Why? I don't know. This tablet problem.
Could you minimize your and try again?
I don't know.
If you have difficulties, maybe you might want us to show your presentation instead.
You just can help me my presentation.
Yes. There you go. Sophie, thank you very much.
Can see my slide?
Yes, I can see it
Okay.
now.
Okay, thank you, Pak Arif. Before
Excuse
that,
me,
I want to.
moderator. Can you ask him to make louder for his volume? We cannot hear him well. Thank you. His voice.
Thank you, Bu Yuli. I'm sorry to interrupt, Mr. Wawan. Can you speak a little bit louder? Because it's the volume is very low in our speaker or headphone.
Test.
Yes, now that's better.
Okay. Before that, I want to introduce myself. My name is Wawan Kosasi. I am from Research Unit for Clean Technology Indonesian Institute of Sciences, LIPI. My topic may be different from the previous speaker. My topic is more about the benefit of fish and the process improving the quality of the fish, especially fish oil. My topic is scaling up of the enrichment of omega-3 content of lemur fish oil, Sardella sp by enzymatic reaction. Why test?
Yes, we can hear you.
Prepost slide. My outline, introduction, methodology, result and discussion, and last conclusion. Next slide. Introduction: Indonesian as a country which have large marine resources, including abundant marine fish biodiversity. Marine fish is byproduct of the fish processing industry containing omega-3 fatty acid, which have higher economic value. Hydrolysis of fish oil using the enzyme lipase can increase the content of free fatty acid. Omega-3 fish oil omega-3 will be released from glycerol, forming free fatty acid. Next slide. There are two methods for hydrolysis fatty acid. hydrolysis using chemicals, and hydrolysis using lipase. Lipase also called triacylglycerol hydrolysis, code number AC3113, is an enzyme that can be biocatalyzed in the hydrolysis reaction of triacylglycerol to glycerol and fatty acid. Next slide. Lipase enzyme have two characteristics. The first, random hydrolysis, example of the lipase enzyme from Candida cylindriaceae. The glycerol, after the hydrolysis, result the glycerol and three fatty acid, pre-fatty acid. And the other one, specific hydrolysis, example lipase enzyme from Aspergillus niger, can result the two monoacylglycerol and fatty acid, pre-fatty acid. Next slide. The aim of research, enrichment of omega-3 content with enzymatic reaction from lemon fish oil using commercial lipase enzyme. Next slide. This is a methodology. Fish oil enzyme reaction using 1 liter reactor, 160 gram fish oil, add 80 milliliter enzyme, and add 130 milliliter hexane, and add buffer 800 milliliter. Into the reactor, flow nitrogen gas low pressure for release the oxygen. Next slide. Sample preparation for test result reaction enzyme 0.5 on 0.125 gram of sample, add 0.5 milliliter of BF3 in 14% methanol, incubate at 55 degrees Celsius for 1.5 hours, and add 0.5 milliliter of saturated sodium carbonate solution and 1 milliliter.
Hello, Mr. Wawan. We cannot hear your voice. It seems like he's disconnected. We still have time, so we will wait a little bit until he reconnect back. Please be patient, everyone. Okay, welcome back, Mr. Wawan. I see your name here. Can you start unmute your computer or?
Okay, next slide. Test.
Yes, we can hear you. So which slide? Okay.
Okay, this is GCMS result data before restriction enzyme. The lemurupis oil have 14 free fatty acid. Next slide. And then after hydrolysis will be 23 free fatty acid. This is next slide. Next slide. This is a calibration curve for calculate the concentration of omega-3. Next slide. This is effect temperature of enzymatic reaction ala EPA and recovered EPA recovery lemurupis oil. This can see the temperature optimum is 50 degree for ALA or EVA. Next slide. This is the effect of agitation of the reaction enzymatics on ALA and EPA recovery in lemurupis oil. The optimum agitation is obtained at okay, this is a chromatogram of the effect of enzymatic reaction time on the equation of ALA, EPA, and DHA. Next slide. This is a effect of enzymatic reaction time of the content of ALA, EPA, and DHA. We can see the time optimum is 24 hours reaction enzymatics ALA, EPA, or DHA. Next slide. The conclusion, the enrichment of omega-3 lemurupis oil has been successful carried out on 160 grams of fish oil using a 1 liter reactor. The optimum condition for temperature, agitation, and enzymatic reaction time were 50 degrees Celsius, 150 RPM, and 24 hours reaction, respectively. Okay. Next. Thank you. Thank you, Pak Arif.
Okay. Thank you very much for the presentation. It's quite interesting because it's quite different from what we have been talking all days, but although there's a relationship in there. So if there is any questions, I'm opening the question and answer sessions because we still have plenty of time, about 5 to 7 minutes before at the end. If there's any question, you might write them in the chat box in the Zoom meeting. Now, if there isn't any, may I ask one question, maybe? I'm quite interested in the omega-3 content of lemuru, but I'm wondered why lemuru? Why lemuru fish? I mean, have you ever compared the concentration or maybe the other lipid or the omega-3 contents of lemuru compared with the other species of fish?
Okay, thank you for question. Why lemuru? Because lemuru very much in Bali Strait, and lemurupis oil many in byproduct for in factory in Banyuwangi, just for eat meal for chicken and fish, not for omega-3. Raw material very much in Banyuwangi. So I use lemuru fish oil.
Thank you. Have you ever compared the concentration between the omega-3 content of lemuru with other species, for example, like layer or other type of species that are common in Indonesia?
Sorry?
Have you ever compared the concentration or content of omega-3 in lemuru with other species like kakap or kerapu or other species that usually used to make fish oil?
Oh, yeah. The first one, lemuru is easy for getting fish oil, and compare with other fish, lemuru higher than compare with kakap or maybe other fish which a lot of in Indonesia, because if use fish high price, not economic, but using lemur fish oil very economic.
Okay, thank you for your answer. We actually have a little bit more time. If there are anyone who wanted to ask any question, you might type in your chat box in Zoom. Okay. There's one question from Professor Sam. He asked, how about tuna? Maybe in relation to the omega-3 content.
Tuna higher than lemur fish oil, but tuna high price for production omega-3, but lemur fish oil very cheap in Indonesia.
Thank you very much for your answer. If there's any question left. Okay, apparently there's one more question and we still have a little bit more time. So from Ibu Hagi, from your study, which one is more expensive, the price of lemuru fish from the fish market or from your lemuru fish oil?
We use raw material byproduct in factory in Banyuwangi for byproduct for fish, packing fish. So fish oil lemuru and very cheap. 1 kilo, not more 5,000, I think. That's all.
Okay, thank you very much. It has been an interesting presentation. And that's bring us to the end of this afternoon session. Thank you very much, everyone, for your wonderful presentation and very meaningful interaction in this session. I would return the time and stage back to our MC, Ibu Sophie. The time and stage is yours.
Thank you very much, Mr. Ari, for your guidance for the presentation. Okay, ladies and gentlemen, I think that's wrap for the afternoon session. Now, I would like to inform you that for tomorrow, we will start a bit earlier. Tomorrow, I think the presentation will start at 8 a.m. and we will be starting for the keynote speech at 8:30. So before we close our session for today, let us take a group picture first for the recording purposes. I think Daddy can help us to guide through the photo session.
Okay, everyone. Thank you, Mrs. Sophie. I get to take a photo session. Please wait for waiting another participant to turn on the camera. Mr. David. Okay. 1, 2, smile. 3. Please wait. To save the file. Okay. 1, 2, 3. Okay, thank you.
Okay, thank you, Mr. Daddy. Okay, ladies and gentlemen, thank you for your attention from the early morning up until this afternoon. We will be seeing you again tomorrow morning at 8:30. Okay, thank you very much. Stay healthy. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you, everybody.
Thank you, everyone. See you tomorrow.
Have a nice rest. Keep a good health.
Bye.
Bye-bye.
Bye-bye.
Hi, Sam.
Bye,
Hi, Mike.
Hi, Yuli-san.
Bye.
Bagus.
Bagus.
Yeah, very good. Bagus.
Your presentation is very bagus.
Yeah, very nice.
Nih,
Terima kasih, Mike Sang.
nih. Sama-sama.
Ya, sama-sama. Yes.
Okay.
Ratu Setiono, terima kasih.
See you tomorrow.
See you tomorrow, Mike Sang. Waiting for you tomorrow.
Yeah.
Eric, thank you for today. Please join us tomorrow. Ibu Yeti, terima kasih. Pak Sam, Pak Furkon,
terima kasih.
teman-teman semuanya panitia, selamat beristirahat. Sampai bertemu lagi besok. Assalamualaikum.
Waalaikumsalam.